superfamily members, TNF, APRIL, and BAF, that are present in the healthy control and absent in people with HIV. And what we saw is that between the AMs and the CD4 T cells, there were very strong interactions. Okay. And between the AMs and the B cells, there were very strong interactions. And again, between AMs and B and T cells in the BAF pathway. In contrast, you can see that in the light, which is an anti-inflammatory TNF superfamily member, this was enriched in people with HIV, and they had interactions between AMs and CD8 T cells in this pathway. So there are very different networks that seem to be operant in these two different healthy individuals. And you can see this across a number of different networks. So I'm just giving you some highlights here. In some of the chemokine signaling pathways, again, both of these interactions are present in both the groups, but the types of interactions are quite different. So here in the healthy controls, we see strong interactions. So the thicker the line, the stronger the interaction between this M1 like AM3 population and CD4 T cells. In people with HIV, this is now with those immune activated CD8 T cells. We also see in co-stimulatory pathways that there is much more AM3 CD4 T cell interaction, but in the people with HIV, their macrophages are interacting with T regulatory cells. Okay. And similarly, in other sort of co-inhibitory checkpoint pathways, we can see that there's an AM3 CD4 interaction in the healthy controls. But now, in the people with HIV, their AM4 macrophages, which are those M2 like macrophages, they are showing interactions between not just the CD4 and CD8 T cells, but T regulatory cells. So there's a very different network. And I think we're overall can conclude that there's very poor interactions between the M1 and CD4 T cell in terms of the crosstalk, and much stronger interactions between this M2 like anti inflammatory cell and T regulatory and other kinds of T cells. And this is just to show that we can validate this by flow cytometry and see that a number of these cytokine and co-stimulatory pathways are in fact defective in people with HIV. But something that we were particularly interested in is when we look closer by flow cytometry and really characterize those AM populations, we found something quite interesting. So here's the healthy control, and you can see that they're dominated by this population 1 that's showing up in the UMAP, whereas the people with HIV are really dominated by this green population 2. So what are these populations? In fact, what we find is this population 1 that's in the healthy control is your canonical TNF, IL-6 producing M1 like macrophage. But this population 2 has very high expression of IL-10 and this molecule CD200 receptor. Okay, and that's shown here again, um, uh, summarized graphically. Even at baseline, people with HIV have high levels of the CD200R expressing alveolar macrophage. And that continues to be high after MTB infection, whereas they don't have very good TNF responses. So, what is the role of the CD200R 200 interaction? This is something we're very interested in pursuing because it's been shown that these interactions can be quite immunosuppressive in autoimmunity and cancer, but really hasn't been studied in the context of TB or HIV. So we are really interested in pursuing what happens with the T cell AM interaction in the BAL as well as the peripheral blood in these people. What is the signaling pathways downstream? And is this how we're able to inhibit the pro-inflammatory TNF response and prevent good responses to TB? So to quickly summarize this before I end with a very short glimpse into what we're doing next, we think that people with HIV on ART at baseline already have some kind of immunosuppressive environment that prevents them from optimally responding to MTB. Because when you expose them to MTB, you now have this aberrant TNF response, you have an immunosuppressive environment, potentially with the CD200 R200 interactions, which is pushing towards an IL-10 anti-inflammatory response. And this poor crosstalk between the AM and T cells is likely leading to dysfunction in the CD4 and CD8 compartment, which prevents good control of MTB. And potentially, this might expand HIV reservoirs as well, we don't yet have evidence whether there is residual HIV in these people who are on ART, but that's something we're addressing now. What we're also doing is collaborating with Cheryl Day, who is at Emory, who has done, who has been recruiting people with HIV and TB in Kenya, in Kisumu, Kenya. And you can see here that in people who are pre-ART, here's their CD4 counts, and this is showing what their CD4 counts are during the course of ART. So this is 2 months, 4, 6, and 12 months. And you can see that their viral load comes down very nicely. And so now we want to apply the knowledge that we've gained from these ex vivo studies to a situation where we can look at both people with HIV or without HIV, with latent asymptomatic TB, which is what I'm showing you here, or people who have actually progressed to active TB. Because what we can see is that the antigen specific, the MTB specific T cells are not reconstituted to their functional capacity even after ART. So here's the HIV negative population. You can see they make a lot of interferon gamma positive CD4 T cells in response to either PPD or ESAT6 specific T cells. That's an ESAT6 is an MTB specific antigen. But then even at pre-ART and as well as 12 months after ART, we see a much weaker interferon gamma response in terms of the frequency of these T cells. So this is something we really want to look at more carefully and really evaluate what is the phenotype and function of these MTB specific T cells in people with and without HIV, with latent TB or active TB, and can we really start to get a feel for how the T cell response in people with HIV is responding to TB in vivo. I think I mentioned that we also have evidence from our collaborators that there's this lower oxygen consumption. So there's a change in the metabolic profile as well of the alveolar macrophages. And so something that we're really interested in pursuing is trying to understand, are the AMs from people with HIV immunometabolically altered in a way that they now cannot respond to the sort of energy intensive activities that you need to make an M1 macrophage and make antimicrobial responses that would control TB. And we have some evidence from our single cell data that, in fact, there seems to be a dysregulation of OXPHOS versus glycolysis in people with HIV. So, again, this is something we're very interested in pursuing. And when we take some of the sort of immunometabolic bioinformatics analysis that you can do from single cell data, we in fact have some evidence now that these AM4, remember the M2 like macrophages that I told you are present in people with HIV, in fact, they are enriched for pathways like lipid metabolism. Okay, so these M2 macrophages have more lipid metabolism, whereas the M1 like macrophages, as expected, are much more glycolytic. And this is very interesting when you think about the context of TB. So we know that in TB, so just take people without HIV who have TB, we know that they have this early glycolytic response in the early stage, and they downregulate OXPHOS. But then later in infection, they upregulate lipid synthesis, upregulate OXPHOS, downregulate fatty acid oxidation. And this promotes this kind of foamy macrophage that's very characteristic of macrophages in TB granulomas. And now bacteria are capable of using fatty acids to replicate and survive. Okay, so this is very advantageous to the pathogen and leads to this necrotic formation that's really characteristic of TB. So, what we think might be happening, this is something we want to test both in humans as well as in a non human primate, is that this aberrant signaling also leads to an aberrant immunometabolic profile that's promoting synthesis. And that this might promote not just a faster progression to TB, but potentially severe TB that's seen in HIV infected people. And this, somehow is related to ineffective TB control. And so we are now really proceeding to look at the granuloma itself in non-human primates at the spatial level. We want to be able to compare monkeys that have been either co-infected with SIV, either received art, naive, and really test some of these hypotheses, which is are the granulomas in monkeys who are co-infected significantly different immunologically without HIV? And can we start to dissect these processes? And this is the last. I just want to give you a glimpse of the kind of really pretty data that we can get from non-human primate granulomas that have very human-like characteristics. And one of the things we've been very interested in is the ring you see here of IDO. IDO is a very immunosuppressive metabolite that's to suppress T cell functions. We've seen that this IDO is expressed in this ring of CD206 macrophages, but not in the CD68 macrophages. And you can see that here in the overlap. Overlaps really nicely with these M2 like CD206 positive macrophages. So one of the things we are really interested in is this what is keeping the T cells? And you can see here the T cells are in the outer granuloma. They're not entering into the macrophage area where the TB is actually contained intracellularly. So it's ideal preventing spatially the ability of T cells to interact with the right AMs, their antimicrobial functions. So this is something that I think the spatial organization of the granuloma and in the lung in general is really critical for our understanding of responses. And so this is something that we're continuing to do. We want to compare the spatial architecture using these multiplex approaches as well as spatial transcriptomics to correlate the architecture of the granuloma with immune function of those T cells and ask whether this is important in the context of HIV. So finally, I'd like to acknowledge all the different people and collaborators who have contributed to this. The people in my lab who've directly contributed to this are Prashant Kanyasile, Chris Stanzen, who's continuing the work on C2, number of collaborators in pulmonary, and then granuloma project, which is actually quite a separate project. We can tell you more about that another time. A number of collaborators that we're working with to do that. Finally, of course, want to thank the funders and the audience for your attention. Thank you. I think that's one of the ongoing questions that we don't have an answer to. What is really the role of antibodies? Is there a role for antibodies in protective immunity? It's becoming clearer that certainly there's a bigger role for B cells than was previously acknowledged. So I think there's B cells are doing a lot of regulatory things and crosstalk that I think really needs to be understood in the context of granuloma, particularly in these tertiary lymphoid structures that we see on the outside of granuloma. There's T B cell interactions, there's B cell APC interactions, and are they contributing in some way to protection? Antibodies, I think, is a bigger question. We've always seen that there's a lot of antibodies in people who have TB, people who have asymptomatic infection. And while they might be biomarkers in some way, it's really still not clear to me that they're contributing to, they might be contributing, in fact, to the pathology. And maybe some of this is through immune complex formation or other ways in which antibodies can be contributing. But I think in terms of a vaccine, that's something we're very sort of interested in general, in the big picture. If we think about a vaccine, yes, perhaps inducing a better antibody response would contribute, but I think T cell response is going to be. But perhaps if you could engineer a vaccine that made antibodies that aren't necessarily present in natural, unnatural engineered antibodies might have in be fantastic if you could immediately neutralize TB, even get into a granuloma. So from a vaccine perspective, if we were to think about antibodies, we have to think out of the box. Antibodies that aren't present, perhaps present in people who are resistant TB or who clear TB. We still don't know if there are people who naturally clear. There are people who are in endemic areas, healthcare workers who are highly exposed to TB who never get TB. And so maybe there are some resistance. Oh, MSCs, we haven't studied. Yes. Yeah, these are all under treatment. So the cells are fresh, the valves are, these are all people who've been on TB more than 2 years. So they're all stable and they're otherwise healthy. So we're trying to do that. It's actually so we're collaborating with HIV virologists to do that. But probably it's kind of a controversial area. Um, some people believe that the residual HIV is not present in the lung. But I think it's also because nobody has really studied very much in the context of HIV. So they're looking at in the gut, blood. And so I think but because there's so few potentially infected cells, we have detected a few alveolar macrophages with HIV proviral DNA. But we have not been able to, detect any kind of antivirus. So it's possible that there isn't live virus, maybe there's viral protein. But again, viral proteins can have a residual inflammatory effect, dysregulate the immune response, but it may not be actual live virus, or if it is there, be very hard to find. And so we are going to look at people and see if we can. But yeah. Hi. Yes. Hmm. So we have, so yes, by prediction, there is some. So there is some glycine and glutamine metabolism that seems to be increased, but the signal is very low. So I think what we really need to do is to do some metabolomics from the bowel and like confirm it that way. So now that we have evidence that there is some dysregulation, I think we can move on to kind of trying to do that more experiment. And we could do that in the monkeys as well, which would be a little bit more tractable. Yes. Could you? I mean, these are naturally infected, so I don't know what HIV strain they have. Um, I think the M tropic, T tropic now is blurry, right? In the natural population, I don't think people think it's such a dichotomous situation. I think that a lot of those were like lab-based assays. I think when you actually look in people, you can see macrophage and T cell tropic, things happening in many people. So it's difficult to say. And as I said, people haven't studied the lung in HIV, so it's very little data. HIV infected people. Yes. Yeah. So, yes, I agree. So, I think it's a different question though. So, people with HIV pre-art will have so much that their response to TB is going to be very different. So, intellectually, we're very interested in this idea that they're healthy otherwise. Why are they still, and then the other issue is that right now in the US and in most parts of the world, everyone's on ART. So it's really to recruit patients pre-ART. And so we have patients from Kenya. We're going to look in their profile several years ago. It's impossible to get BAL from people with HIV who have not treated. I mean, I don't know even ethically if that we thinking about, but certainly we can go back to archives pre-ART and we're doing to see if we can see similar. Those are the detailed studies we have to do. We need more funding to do it. So we've got all these PBMCs frozen. We can design, 30 color panels. That's our hope. Antigen activated profiling, transcriptomics. There's a lot of things we'd like to do. Thank you very much. Yes, everyone.
Now I request our chairpersons to kindly present Mintu to Professor Jyoti. Now we proceed with Professor Deepankar Nandi's talk.
So this one is yeah. Dr. Deepankar is BSc from Bombay University, MSc from MS Baroda University, completed his PhD with Nobel laureate James Ellison the point at UC Berkeley, and then postdoctoral training with Professor John Bonacco at University of Cincinnati. Since his joining how do I 80 at just this way. His 23 PhD students to his credit, and another 50 or 100 students to come from his work. Dr. Deepan. All right. Okay. Thank you very much. Let me start off by thanking the organizers. And so without further ado, I'll get going. So I'll talk on interferon gamma. Some aspects of interferons have been covered, and I will talk specifically about the responses of gamma that are modulated by nitric oxide and how we got there. And so I've modified the title a little bit. It's yes to no, and here no stands for nitric oxide. All right. So I come from the Indian Institute of Science, and this is on Founders Day, which is March 3rd, which is and the founder
is Jamshedji Tata over here. And this is the old biochemistry department where we were till 2011 and then moved to a newer place. This is the biological sciences building, which houses several of the departments right now. This is a collage of my lab. One of the students had made this. It tells you about the diverse things that we do. And in further details, it's over here. So we work on microbial responses. We have both E. coli and Salmonella as a model. With regard to host responses, we study T cell biology. We also have an aspect studying inflammatory responses, and I'll talk mainly about this. In addition, we also do Raman spectral analysis, and actually that's done in collaboration with Professor Umapati. Interferons were discovered several years back by Isaacs and Lindelman. There are three broad classes, type 1, type 2, and type 3. The type 1s are the mainly antiviral ones, and those, of course, got very famous during COVID because patients who had lower amounts of type 1 interferons were more prone for severe COVID. The one that I'll be talking about is type 2, which is mainly involved in cell mediated immunity. And then you have a third class of type 3 interferons. Those are important at mucosal surfaces. And so these are type 1 interferons, a little bit about them, but they are produced mainly by T cells and NK cells, and they act on other cells. And as I mentioned, they are mainly involved in cell mediated immunity. And if you remember Balu Pulendran's talk, he talked about BCG inducing type 2 interferons, which is interferon gamma, and which affects other sort of responses. And so it was first identified as a macrophage activating factor. So they are known to activate macrophages, and that's what he had also talked about. CD4s, BCG specific CD4s producing gamma, which act on macrophages, which are important in modulating responses. Um, so the canonical pathway is the JAK-STAT pathway, where you have tyrosine kinases phosphorylating transcription factors, and these transcription factors move inside the nucleus. And then you have what's known as primary responsive genes, and then which in turn, through IRF1, turn on secondary responsive genes. Um, like many things in immunology, interferon gamma is important, but also have deleterious effects. As mentioned, they are important for immunity against intracellular pathogens. And so if you lack interferon gamma or its receptor, often you come down with BCGosis. And because BCG is often one of the first vaccines that's given to people, much to the dismay of parents, and BCG is a live attenuated vaccine. So the kids come down with disease, and it's a major problem in some cases, and so interferon gamma is very important for protection against intracellular pathogens. However, very high amounts also make you prone towards autoimmune diseases. So like many things in immunology, we talk from both sides of our mouth, and we should be aware about proper balance. Okay, so how did we start off? We were interested in trying to find interferon inducible genes, and historically, these have been very important. The MHCs, for example, are gamma inducible, and they are, clearly, very important. So this is a particular, but you need a good model system. So what's the model system that we've used? This is H6. It's a hepatoma cell line, and this was used a long time back to identify gamma-inducible proteasome subunits. You can see this is minus gamma, you don't see LMP7 or LMP2, and with gamma, you see robust induction of LMP7 and LMP2. Over here, you induce them, you immunoprecipitate with an anti-sera that recognizes proteasomes, and you run them on 2D gels, and you're able to see this. So we had a model system. So one of the things is these were known. Can we identify some other novel ones and signaling pathways? So this is pretty much the model system that we have. These are H6 cells. We grow them in 96 well plates. We treat them with gamma and then incubate for different time points. And then we process them, and there are different readouts that we use. And you'll be seeing some of the readouts that we use. So this is one of the first ones that came through. So in the absence of gamma, there's very little nitride being produced. So what happens is NO gets produced, and NO gets quickly converted into nitrite and nitrate. There's a very easy assay for nitrite, and that's the Grice assay. That's what we use. And you can see in this case, in a very dose dependent manner, nitrite is being produced. As nitrite gets produced, you also see a drop in cell number. So this is a very simple system that we used for several of our assays. When I first started the lab, we were not, I wasn't very aware of NO, nitrite, and ROS. And Professor Ram Sharma over here actually played a very critical role in telling us a lot about this. And we are very thankful to him for his suggestions. And this is actually a picture of Professor Ram Sharma with Jyoti and Anujit when the centenary celebrations of our department were there. So one of the first things that we found was nitrite is being produced, and these are two students very long back, where we used this as a bioassay to try and find for to score for gamma in a biological assay for gamma in secretion. So this is a very simple system, and when we first had it, it's very useful, and we keep using this. So we've done a lot of studies with this. Jyoti and Bani, for example, have published several papers on this, and Shravanti has also done a lot. So what we showed over here is NO modulates a subset of responses. Some of them are independent. So, for example, the MHC ones are independent of NO, but the others are. So, for example, cell cycle arrest, growth arrest is dependent because you can block it with NO inhibitor. Okay, so a little bit about NO. So nitric oxide was discovered several years back, and Furchot, Ignaro, and Murad got the Nobel Prize for various discoveries of nitric oxide. And it's important over here to realize that NO gets produced uh, via arginine. So you have arginine being converted to citrulline and a nitric oxide. And you have an enzyme over here which is responsible for this, which is NOS or nitric oxide synthase. There are three forms of NOS. You have the neuronal NOS, you have the endothelial NOS, and then the one that we study, which is the immune or iNOS. There are different effects of NO. NO has, either it can act readily on transition metal ions. For example, cyclic AMP, cyclic G, soluble guanyl cyclase activation increases cyclic GMP, and then there's a phosphodiesterase that breaks it down. And what Viagra does, for example, is to block that, and that's why you have prolonged erections. And then you have indirect effects and especially and also formation of peroxynitride. So NO has also applications. So one of the ones are to to take nitroglycerin, keep it under the tongue, and so what NO does is it relaxes it, so it lowers the blood pressure. And you also have arginine supplements, for example, are now given to sportsmen to improve their performance. So you have higher NO. Higher NO leads to better, even a 10%, even small increase in performances can make a difference between a silver medal and a gold medal. And NO detection was also used in breath during COVID and so on. Okay, so this is once again our activation system. And so in the first part, I will just briefly cover an interesting observation that we had. So this is interferon gamma at different hours. And what we found out over here is if you compare the untreated with gamma, so gamma, the ones treated with gamma are more yellowish in color. And over here, as most of you are aware, there's phenol red, which is a pH indicative dye. And so the question is, why is this media turning more yellow? And what does that reflect? So one of the possibilities is that you have more cells growing in the presence of gamma. So you have more conversion over there, and that's not right. Because in fact, with gamma, what you see is the cell number decreases. There is not much difference in the cell morphology over here. So what's happening? So there are three aspects that I'll try and cover in this talk. First is to decipher the molecular mechanisms of cellular acidification. I'll summarize this part because most of it is published, but I will talk a little bit about the implications about it. The second is, the signaling aspects and possible therapeutic interventions. So I'll discuss these parts. So with regard to the first part, I'm going to summarize this part. So what gamma does is it increases acidification. So what the conversion that you're seeing, the yellow color is because of more acid production. And what we have shown is that there is more lactate production over there. And in fact, this lactate production is dependent on nitric oxide, reactive oxygen intermediates, and a key signaling player, which is the HIF1 alpha. So we used this model also to study tumor cell lines that do not respond in a similar way to gamma. That means they induce MHC, but they are not responsive. They don't produce NO. You don't see the lactate production. So what we showed was the gamma activation actually reduces the growth of these tumors if you add exogenous potassium lactate. And this is important because some tumors are responsive to gamma, some tumors are not responsive to gamma, and in terms of tumor therapy, this becomes important over here. And this was the model that we had shown over here. And so while we were doing this work, we realized that gamma seems to play a very important role in anti-tumor therapy. So you have some tumors that are responsive, some tumors that are not, and this is with both with regard to checkpoint inhibitors as well as CARs. So why do we see this difference? Is there some way by which we can convert the ones that don't respond into responsive ones? And one of the suggestions over here was maybe you could use some of the signaling mediators that we had learned as well as lactate, because somehow seems to reprogram these cells and they make it more sensitive to gamma. And so this part has been published. So for those of you who are interested in it, should take a look at it. Okay, this was the next set of question that we asked is how many genes or what are the genes that gamma induces in a NO-dependent manner? So there have been a lot of studies with gamma with regard to global gene changes, but is there a subset of genes that are regulated by the NO produced over here? And so the experiment was done. So this experiment was done with raw and we've done it plus minus gamma and plus minus a NOS inhibitor, which is N-methyl arginine. And bulk RNA seq was performed over here. I'm again going to summarize these parts. So typical of what others had seen, you have transcription factors being induced, the G proteins, cytokines being induced. You also have proteins conjugated by ubiquitin-like proteins, this ISG-elation, which is important for post-translational modifications also being induced. You have signaling proteins like the STATs and the SOCs, which are suppressors of signaling over here. You have the glucose transporters, and you also have cell surface receptors, and of course, the most important ones, the antigen processing genes, which was originally the ones that we had studied. So this becomes important over here because there is a new type, a class of diseases that we are now aware of, known as interferon gammopathy. That means you have patients showing very high amounts of interferons. And these lead to pathologies or hyperinflammatory responses. And it turns out that some mutations in proteasome subunits are also a subset of interferon gammopathies. This is a cartoon that sort of shows you, or these images that shows you what happens. These are patients with mutations in interferon gamma inducible subunits, which lead to hyper inflammatory responses. So basically, since proteasomes are changed over here, their substrates are changed, as a result of which you have metabolic changes, which again lead to some of this. Okay. Um, so coming back to NO, so we found out that like NOS1, NOS3, which are the other NOS subunits, were not induced. But the NOS2 and the CYBP, which is an important subunit for NADPH oxidase, also gets upregulated. There were modest number of NO-regulated genes, but this is something known. But is there something really new that we could uncover? And so this led us to a set of genes which are important in NAD metabolism to be modulated with gamma. And so we followed this up because this led to some novel aspects over here. Now, it turns out that NADH ratios play very important roles in our metabolism. And as most of you know, that NAD is important for epigenetic transformation. So over here, you have histone deacetylases, which play important roles. NAD levels also decrease with age, and boosting NAD might be important for anti-aging and metabolic health. So with these aspects in mind, we sort of targeted this, and we wanted to validate whether this indeed was happening. And you can see again, this is in a kinetic dependent manner. With interferon gamma, you have higher amounts of NAD that's produced. So we started off with the RNA-seq, and now we are able to validate this. And this increase is in an NO-dependent manner, because if you see over here with gamma, you see an increase. If you have cells treated with gamma and LNMA, where you block NOS, you see it's reduced. Okay, so there is expression now, but does it have any function? So to address function, we address it two ways. One of which is to try and block NAD production, and the other is to add NAD and try and see. So you can inhibit, you can increase NAD, and then try and see if it has any functional effects. I'm going to summarize this part over here. So blocking NAD, for example, lowers NO because NOS, is a complicated enzyme, has lots of requirements. So if you block NAD, for example, it lowers NO and increases cell survival. And on the other hand, if you add NMN, which is a precursor which gets into NO, it increases NO at suboptimal concentrations. So the fact that gamma enhances NAD may have important implications with regard to metabolic reprogramming of cells. More importantly, nicotinamide riboside is actually a nutritional supplement and may improve mitochondrial function and so on. So this is again an important aspect over here. So this part of the study again has been recently published. So those of you who are interested can also take a look at it. So I'll finally move to the last part of the study. So we've set up these models. We have found some novel signaling and important genes that are important important in this process. So is it possible that we could make use of this model system to try and screen and identify novel inhibitors that might have potential translational effects? So that was with the idea that we started this, and there's always a need to find more novel ones, and that's what. So again, we use the same model system, except that we screened a library over here, and this is what's known as the LOPAC library, which has a bunch of pharmacologically active compounds, and this was a collaboration with Professor Deepak Saini, who had access to the library. So it's a huge bunch of compounds, and that we screen over here. 16 plates, 80 compounds per plate, and so on. It involved a lot of work over here. But finally, we came down and then we put the data together and we segregated the data into three parts. One is we were able to identify NOS inhibitors that were already present in the library. So that told us, that the assay seemed to work. We also identified a lot of steroids that inhibited NO production. So steroids are known to inhibit NO production. So that again was consistent with what we were expecting. And then we had some novel hits. So these novel hits, we bought the compounds and then tried to validate and see if indeed it was doing something. So we now came down to about four over here. So the first one is pentamidine, which is an MDA glutamate receptor agonist, which is used against leishmaniasis. The other is an antibiotic, azithromycin. The third is roliprom, which inhibits phosphodiesterase, and it has some roles in cyclic AMP. And then the last one was ornithine, which blocks thyrodoxin reductase, which is again used against rheumatoid arthritis. So then we tried to study if any of these would have any in vivo effects, and the model that we used over here was the DSS model of colitis. Over here, where you feed mice and the mice drink DSS and they develop colitis. And then you could treat with the compounds and then try and see if you're able to block over here. Can we move to the next slide? It's not responding. Stuck. Okay. So oh, your battery is running low. battery low. एक मिनट में नहीं अभी भी नहीं हो रहा है. Okay, so it's okay. Okay. Um, all right. Essentially that's what we did, and then we treated them. Yeah, I'll be done. I'm almost done. Okay. And so they work in vivo also. And so pentamidine and auranofin were the ones that showed the maximum effect. So I'm going to summarize this part. So what we have done is over the years we have developed this system to study NO-dependent gamma responses. We have used this system to try and screen for inhibitors as well as potential signaling molecules, and then we validated some of these in vitro effects that we have got in vivo. And so this is the publication and of the application that we have done. And all this work has been done. Avik has been the main person who has done the work, and you can see this is his screening set of plates that he has used because a whole bunch of them. So I'd like to thank students, the funding agencies, mentors, institutes, colleagues, and so on. So this shows you members of the lab at the same place, same location, but different people. And this is in January 2020. We had no idea that COVID would be coming. And then this week that we got, and then the lab has also changed with time. And this is a more recent here. Uh, thank you so much. I'd like to thank you for your attention.
Making it so taking patient. That's what we need.
Good, good, good. So with regard to yeah, yeah. Oh, that's a good question. I didn't share a lot of the data, but others have also done this. So the knockout mice are much more sensitive to bacteria. So in fact, that's the best evidence that it does play a very important role because NOS2 is very specific for the iNOS mediated. Other NOSes also, but this one, NOS2 is expressed mainly in macrophages and neutrophils, and these mice are Yeah, okay. Okay. So that we have not distinguished between that. So what Professor Rao is asking is you have NO, but NO also combines with superoxide to form peroxynitride, which is highly potent, right? So that delineation we have not done, but we have shown peroxynitride also plays a role, at least in our system, because you can block with specific peroxynitride and you're able to rescue. Yes. So, very interesting also. Yeah, so yeah. So for sepsis, yes. Okay, right. So with sepsis, you have to be very careful because you have two different forms. You have the initial hyperinflammatory response, and then you have a subsequent, lowered inflammatory response. So, of course, it's again what happens again during infections. At a later point in infection, you have pretty much a sepsis-like condition that we are talking about. So, with NO, again, when you give it, where you give it, how much you give it, again, you have to be very careful. But you're right. I mean, NO does play an important role in blood pressure, and so it can be having other effects also. It's sepsis is a lot more complicated. Yeah. Very nice talk. Definitely uses that. Good. Yes. So let me try and answer your first question. So the first question has been answered in our system. Done is we have blocked with quenchers, and then try to ask, is there oxidative stress? Oxidative stress was blocked, right? So we have done the reverse experiment, which is you block ROS, using either quenchers, catalase, and so on. Then you see NO. Whereas oxidative stress is lower, you see a partial risk. So in our model, NO is upstream and the ROS is downstream. So and that has been shown. It's quite clear in our model. I always thought. So in our model, NO is upstream, NO is the main player, and then subsequently it turns out. In fact, CYBP, the NADPH oxidase, and so on, are also upstream. So that's what in this that's the way it is. Yeah. Yes, yes. So what happens is both of them are anti-inflammatory. They, so they in the autoimmune they help because there's high amounts of inflammation. In the sepsis model also it helps, but the amount is limited. You see a more dramatic phenotype in the DSS. So that's why. But sepsis is it's multifactorial, is much more complicated. But yeah, so that's why I showed only the DSS model just to make the point that in vivo, that these seem to work. Yeah. So sure. Thank you. So on
Thank you, professors, for so graciously leading this session with such clarity, depth, and guidance. Now, I would like to request our respected chairpersons to kindly present our esteemed speaker, Professor Dipankar Nati, with mementos as a gesture of our heartfelt appreciation for their enlightening and inspiring talk today. Thank you very much, professors. Now I would like to invite Dr. Ambak Rai to kindly come forward and present token of appreciation to our respected chairpersons. We are now moving towards our next session of technical talks, and it is my pleasure to invite Mr. Nagaoka Keiji, who is General Manager from Life Sciences Sales and Department, representative of Sony Corporations, to deliver the technical address.
नमस्ते मेरा नाम नागाओका मैं जापान से आया हूँ so I am stop sorry now I'm using English so I'm general manager global sales come from Sony headquarter So today, please allow me to begin the presentation with an overview of Sony business, maybe around 2 or 3 minutes. Then I ask Dr. Abhishek, our field application members, to talk about technical details. Okay. So this is Sony's business overview. As you know, Sony are doing various kind of business. So now we are belong to entertainment technology services, this group. So this group, how can I say, including Bravia and mobile phone, also digital still camera. I can say some electronic products. So uh, this is a reason why we belong to Sony this electronic equipment business group. So we can utilize many Sony's technical assets into flow site business. Because many people ask us why Sony start this flow site business. So many people, at the first stage, many people doesn't know Sony has this flow site metal business, honestly speaking. But as you can imagine, I believe, so it's a Blu-ray disc technology is one of the very representative example. So you can imagine it very easily. So how we control this laser technology, laser. So flow cytometry is core technology, also laser. Also analog digital conversion data processing technology, also same kind of project. So we can utilize our Sony, how to say, historical technical assets to flow cytometry business. Then now we have started this business from 2012. Yes, thank you for every its customers and the stakeholders support. Now we have say enriched our lineup, as you can see. So first, so we start from SH800, and also next MA900, such kind of compact type cell sorter is very, how to say, user-friendly product. So many shipments around all over the world. Then I need to emphasize that Sony is a, I say, world fastest introduce of spectral technology into this flow site meter segment. So you can see this center. So we introduce this spectral technology into cell analyzers first. Start from SA 3800. Then 2019, we launched ID 7000. It enables multi-parameter analysis. Then today, I am very happy to introduce with you. So about FP 7000, very advanced spectral high parameter, multi-parameter cell sorter. So this FP 7000 this year start shipment. So later I ask my colleague to explain this what is FP 7000. Okay. So this is Sony global operations that support our business. So needless to say, we running the business all globally. But today, I want to emphasize this Indian business absolutely. So last year, we have started Indian business. Now, also, with together with our partner, Spinco and Biotron as our distributor. Then also, I need to say thank you for my customers. We already successfully shipped MA900, also ID 7000 for aims. So I'm very happy to how to say thank you taking this opportunity. Yeah. This is almost my last slide. So as I say, our how to say machine now many researchers globally using our machine. So this year, so this three person got Nobel Prize. The left side, Sonoma, and the center, the Osaka University. Both of that is our important customers. So we Sony are very how to say more than happy to contribute such kind of advanced research. So absolutely, Sony will continue to make effort to contribute Indian cutting edge okay, future researches. So thank you very much and धन्यवाद.
So thank you everyone for listening, and myself Abhishek. So I take care of the application part of Sony in India. So along with the Japanese technology and the information here in India, we are also present with our partners so that all scientists across India can get the full support. So this year we are trying to explain about our latest launch, that is FP7000. It is very intuitive cell sorter with more capability and multiple nozzles capability, exchangeable sample line, and this instrument can accommodate six-way sorting. So with all automation, it enables user to like initiate the instrument very quickly without any manual intervention. So the key aspect or the key feature of this instrument are there like multiple type of nozzles, 70 micron and going up to 130 micron. Your user can themselves change the sample line. Uh, then samples are agitated uh, thoroughly, and so then you have like sample loader where you can have like multiple type of tubes starting from 0.5 micron going up to 15 ml tube. So this gives a liberty to use different type of tubes. And when we talk about the sorting, you have option of from 2-way, 4-way, 6-way, and even all sort of plate can be used now, going from 96 up to 384 well plate. With our technology, we provide you a precise unmixing for the highest data quality. And here, since all the detectors, the maximum number of detectors in the industry, set 10 nanometer apart, give you a complete spectrum. Well, so that there is no gap and you get the full coverage. And this high quality data is you receive is along with the weighted least square method, which is the best method as of now, and this further gives you a finer resolution of your data. So here is one example where we are able to resolve between closely placed dyes in all lasers, violet, green, yellow, green, and red. So you can have like a BV421 E-fluor 450. Normally, through conventional cytometer, it cannot be separated. But with the help of this spectral flow cytometer, these dyes can also be separated easily, and these population can be sorted. So even most of the people in India, they are now looking at the smaller particle size detection and sorting capabilities. So we have showcased that using the Gigamix beads, we are able to separate 100 and 300 nanometer particle sizes. And these are the peaks available here. And here you can see post sorting data where 100 nanometer peaks are here in this and 300 nanometer beads are here. Another key feature is like use of beads single stain control. So normally people have this concept of like where the fluoro the fluorophore presence on cell and beads. So here we can see the intensity. We have showcased that the intensity is more or less similar. So that also reduces the use of cells, and in case of like limited sample type, yeah, this method becomes a very precious, and user can utilize single stain beads to start their experiment.
Same way for the gain settings, so our spectral references have similarity, and even at different PMT voltages, you get a clear signal, reducing the overlap. So this is one of the highlight. And the system is completely automated, starting from your it starts calibration, fluidics check, optics alignment, laser delay, and drop delay, and this whole process is manual interference free, and you can directly use the instrument once it is ready. So this robust fluidic system ensures that you are getting a six-way sort without fanning issues, without challenges, and directly onto the your collection tube. Same way, the single cell deposition system on your plates, let it be 96 well plate, let it be 384 well plate, a particular angled and peltier based ensures that cells are with minimal stress, and you get directly onto your well. And this also, we have index sorting option, and so that you can even identify from which particular population you are getting your cells. The user interface is so user friendly that and it is similar to ID 7000. So those users who have been using ID 7000 earlier, they can directly import their panel and use it on FP 7000 without any changes, and they can acquire data and sort very quickly here. And all menus are like so well represented that even on the upper tab, as well as when you click right click, you get majority of the options that user require. So overall, in a nutshell, I can say Sony is innovative, reliable, and capable. So on these three parameters, we access ourselves and we provide all these capabilities of our instrument to all users. We can be yes, thank you.
Thank you, sir. As we now move forward to the special talk of the day, it gives me great pleasure to invite our distinguished chairpersons to kindly come forward and take the charge of this session. We are honored to welcome Professor Pavan Sharma, who is adjunct professor at IISR Bhopal. Professor Sharma is a renowned immunologist and educator, highly respected for his extensive contributions to the field of infectious disease, host pathogen interactions, and immunoregulations. He is former senior research scientist at ICGEB New Delhi, specializes in medical biotechnology and infectious disease like TB, malaria, leishmaniasis, and MBSs. With decades of research and development and mentorship expertise, he offers strong expertise in parasite immunology and biomedical research guidance. Accompanying him, we are delighted to welcome Dr. Uma Kanga, who is additional professor in the Department of Transplant Immunology and Immunogenetics at All India Institute of Medical Science, New Delhi. Dr. Kanga is a distinguished scientist whose research in immunogenetics and autoimmune disorder has significantly advanced our understanding of immune mechanism in health and disease. With our esteemed chairpersons now on the dais, I would like to request them to kindly take charge of this session and invite the speakers for the special talk.
Thank you very much for the generous introduction. And how nice to have you both. May I also thank the organizers of this meeting to have included me. You know, at this stage, you don't go to every meeting. You know, the title of the presentation is I am 99 past. I was born on 2nd October, which happens to be the birthday also of Mahatma Gandhi, and therefore it's a national holiday every year. And you see, my mother died 8 days after my birth, and I had a stepmother. So I have had a very difficult childhood. Luckily, I got the academic and athletic roles of honor from my college in Lahore. I'm a Punjabi, as you know. And then I got a scholarship to go to France, where I worked in the laboratory of Jacques Monod and got the Doctor of Science, or a DSc type of a degree from Sorbonne. I got a fellowship also from Germany, Humboldt Fellow, and there I spent 1 to 2 years with Fyodor Lunin in München and Tübingen. Coming back, I was lucky to get selected for associate professorship of the newly coming institute, the All India Institute of Medical Sciences. And I joined on 10th August. In the morning, Professor Keswani had joined the Department of Anatomy. So I was the second faculty member to have joined the AIMS. But unluckily, AIMS did not exist in the form that you see today. It had no building at all. So we used to teach in the nurses' hostel building. And then eventually my lab came in being in the 1965. So my next session for research started in 65, and I used to work on mechanism of action of growth promoting and developmental hormones. In 1970, Howard Goodman, who was the chief of immunology at WHO, came and asked me to take up the presidentship of a Immunological society that they wanted to start or establish for India and Southeast Asia. So I was reluctant. And they said, look, India has the world's largest number of lepers. Do you expect Americans to come and solve your problems? I felt so ashamed that I had then no hesitation in signing the agreement, and that is how immunology started in India. Now, could you please, I mean, have I to say?
this one.
अच्छा अच्छा ये इसको कर देना oh no no
Oh, let me take. This one is first. This one second. Second PPT.
next now so after in late 70s early 80s when I was a Nehru fellow I started working on mission oriented research and have a problem तो
Next one.
पीछे हाँ ठीक we have several products which have gone the whole way of not only discovering the basic research and then taking it through the various stages to make it a product which is acceptable by the DHS and then give it to a company because without that it will never reach the public. So one of the products that we have passed on is Basant, which is I mean, I was asked by our OBG department, gynecology department, that they receive many women with their normal vaginal discharge. So can I do something for it? So I started working on the vaginal infections. And so this is a product that I made, which is inhibits the gonorrhea, candida, including the quality of these vaccines is that they also act on antibiotic resistant drugs. So many of them were, non-treatable at that time. And we gave this formulation, which was quite effective. And next slide, please. This, strangely, this formulation is also effective against AIDS virus, which at that time was causing a lot of anxiety. And Manoj carried out even a clinical trial in USA on HIV-1 at the early stage of infection, and it was effective. Also, this product was effective against the human papilloma virus, which causes in women the you see this is the sorry the Basant on human papilloma virus. It's of course quite troublesome, and our Basant is also effective on it. And it's not only in laboratory that we tested it, but eventually we had to take it to human trials. And at Aligarh, they asked me to do something. They have many cases of HPV coming. And with them, we tested 11 cases. All of them, without exception, became negative from positive status. So it was rather good that we developed this product. As I said, vaginosis was the problem on which our colleagues at AIMS in OB and JY were concerned. And what is interesting is that, in fact, originally we made the basant for this purpose, because they were saying that it's a very, the smell is foul and it's a uncomfortable thing for women to have. And women who used to come to their department, a very large number, 90 to 95% of patients were having vaginosis or abnormal vaginal discharge. And our basant was effective on each one of those products. So HPV, just I showed you the human papilloma virus, which so it was also here a very effective thing that happened. Here is the typical case that you see on the left, as they are, and after treatment with basant, this is how they become very clear vagina. The glue cells which are there in the originally infected stage were all gone or nearly gone. And the odor, which was not very pleasant, became totally healed, and the pH of the vagina restored to acidic range where it is that of a normal, healthy vagina. But this is, I have already told you how I went into this type of research. And you see, as they had told me, India has the largest number of lepers in the world. So I knew not much about leprosy, in fact, nothing. So then I learned why it is caused, how it is caused, and I tried to develop an immunotherapeutic vaccine, which is also now we know is an immunoprophylactic vaccine against leprosy. So the path that we adopted was to select a mycobacteria which should be immunologically cross-reactive with M. leprae, and it should be able to elicit immune response from not only the tuberculoid leprosy but also lepromatous leprosy patients. Tuberculoid leprosy is one end. The other end is the LL patients where they are the worst that could be. So in vitro and in vivo investigations, eventually this is the mycobacteria that we have chosen. And it's a nice looking bacteria, as you see. It's my favorite now. And the Baris did the sort of analysis of this bacteria, and we know the sequence and the analysis of this bacteria, and it named it as Mycobacterium indicus pranis. Pran is the name which my mother gave me. So Gursaran Prasad is because my father was a Radha Swami, so he took me there and they gave my name Gursaran Prasad. And
That is why I'm GP Talwar. Luckily, many people call me Gursaran Pran Talwar. Anyway, that's a minor issue. What is interesting is that it is not only so marvelous against leprosy, but it is also usable as an autoclave vaccine. And it is effective in tuberculosis also. And you see, there are a category of tuberculosis which are difficult to treat by the available drugs. By the way, I gave this MIP to Cadila on a non-exclusive basis to make it available to public. This vaccine clears. If we had to give the drugs, that is the rule, but this was put extra. In addition to the drugs used, we gave this, they combined this vaccine, and the patients were healed faster and in shorter recovery time. And otherwise, it's a long time that takes to treat leprosy. And the people who were given the vaccine also had no granulomas, and their reactions were reduced. And interestingly, they all became lepromin negative to lepromin positivity status, so that they were less likely to get reinfected. And slow responders could respond faster. This is some of the cases that you see who received not only the drugs, but also the MIP, our vaccine. And as you see, from first to the last patient, the ugly sort of signs that leprosy has on the human beings were healed. No drug available anywhere in the world can achieve this. So you see the first case, second, and I've just chosen 4, 5 cases to show you what happens to these leprosy cases. Unbelievable, they become normal. This fellow from Nepal, the patient CHB, touched my feet and said, God bless you. That is why I'm still 99. So, and still going. And look how these ugly signs are also removed, which the drugs alone have never been successful anywhere in the world to achieve. So it's a very unique vaccine for that. What is also interesting is I told you that there is a category of tuberculosis which is difficult to treat. And this is a clinical trial which the slide gives the results. Those who receive MIP plus MDT, the usual drugs for tuberculosis, almost all get healed. Whereas in this category of tuberculosis, but multi-drug alone only achieves about 77% healing. So this one case that did not respond so well to MIP inclusion was a defaulter, or he didn't come for regular treatment, and so like that. Another very interesting property is that the people who receive MIP, in addition to drugs, had very little response. You see, those who receive MIP are shown in blue, and those who receive only drugs, 50% of them can have relapse of this type of cases. So therefore, it's not only good for leprosy, but also tuberculosis. So leprosy has come down in the country to really very, very low levels. And, we could eliminate it completely, but the people who are dealing with leprosy are government employees, and those who treat them are also employees. They said, "Gee, then what will happen to our salary? Our jobs will be gone." So you see, it's one of those situations where they don't give it regularly or so that regularly. But in any case, in tuberculosis, see, it's effective, plus the relapse rate is much lower than with drugs alone. Now you see, this is something which we never expected. In one of the professors at AIMS said, "ये आपका सब चीजें ठीक कर रहा है, तो मेरे cases हैं of anogenital warts. Let me use it to see whether they can be helped." So they did do, and I'll show you the results. Also, I will not tell you about my vaccine against HCD for birth control that I mean, the time allotted, even this is difficult to all communicate. But here is the patients with the anogenital warts above, and on treatment, would you believe MIP, the vaccine which was made for leprosy? Around the venous, you see those ugly warts are all gone, completely gone. See how, the patients don't believe that they've become so cured. So imagine what this can achieve. To sum up then, we have developed a marvelous vaccine, I would say, with the non-pathogenic cultivable mycobacteria, so that it could be cheap eventually. And its gene sequence is totally determined. It is named as Mycobacterium indicus prani, my first name, NII, National Institute of Immunology, from where the field trials were conducted. And it clears bacteria, clears granulomas, and upgrades CMI response of the BLLL patients so that they don't get infected and leprosy patients again, which used to happen in those which only were treated with drugs. And you see, we can eliminate completely. It has come very low, but I told you that there is a reluctance because the jobs of all those employed for leprosy would go, and they are all in government service. So otherwise, our vaccine is not only good for leprosy, but also tuberculosis, and for anogenital warts, for myelomas and some other cancers. And it's a potent invigorator of immune response. My current research is on cancers, and cancers of an advanced nature which are drugs resistant. Every drug available in the world, they are resistant to. Such cancer patients die very early. I mean, they are short lived cases, and it's a worldwide problem, not only in India. So we have made an Immunovac is a monoclonal antibody single chain variable linked to sarcine. That this monoclonal antibody piece can recognize such cancers because, interestingly, these cancers which are resistant to all drugs express ectopically HCG. Would you believe that? And so we have a single chain from a monoclonal antibody against HCG, which is then linked to a toxin. So the antibody leads the drug through the cancer tissue, and sarcine kills it. And we have 13 cell lines of such cancers, and I can tell you, I didn't think that I had enough time to include this area, but you see, it's a promising entity which we are on at current stage, and let me hope that someday we reach the end during my lifetime and help the whole world, not only India, for a cancer which is at present non-treatable. Thank you very much. Next slide. Oh, sorry, next slide. Oh, yeah. This is how this Immunovac acts in the nude mouse. So the cancer is reduced quite substantially by giving this. And the credit should go to all those who have collaborated with me in during my that I have presented to you, and they are either have been my own students or even those which are involved in other institutions on doing clinical trials and so on. So they should all be thanked also. Thank you very much.
Thank you, sir. Thank you so much for your excellent overview of your excellent contributions.
I will be happy to respond.
Can I repeat?
Thank Thank you very, very much.
Now, sir, for the affection that you give to all of us. Thank you so much.
I am available for any questions or clarification.
Yeah.
यार ये बंद आंखें में पटा इधर खड़ा हो जाता हूँ
Lights.
इधर की तो लाइट बंद कर दो
Light बंद
हाँ जी बोलिए
That's okay.
सॉरी यस
MIT teach now with so moving forward, when COVID first came to India, we had a hypothesis that, for a particular type of natural killer cells might be effective in controlling it. But we did not know how to increase the amount of these natural killer cells in human beings.
Which cells?
Sorry?
Okay, sir.
Natural killer cells, NK cells.
Thank you.
So, if the Dr. Population Mark carried out a trial in our institution using MW or MIT in healthcare professionals. So we took a control arm, took a cohort of 50, this published now in several journals. Cohort of 50 healthcare individuals who were given MIT 2 doses and versus the rest of the professionals who did not receive the sequencing of that. Found a dramatic effect on decreasing the amount of adaptive. So we have the subsequent used it in, as you showed, in myeloma as well. We have used clinically in refractory myeloma. Yes, demonstrable effect. And I just don't know how it works, but it's a wonder vaccine, wonder drug, wonder immune network. Thank you so much. Another question?
So all of you are happy?
One more question.
Good morning, sir. It's very nice talk, and I'm blessed to hear this talk. I'm working in the Leprosy Trust India. This organization is mainly working on the leprosy disease. So, as we want to work on MIT, and but we are not able to procure MIT for the use of leprosy disease. So, can I get the information? Is like from where I can get this MIT vaccine?
This is the bad thing of Cadillac. The owners of Cadillac became very friends to me, and that is how I thought it was a well-established company. I listened to them, but I don't know they are selling it this same MIP under different names for psoriasis, for many other diseases where it is effective, but they are charging it.
So very high cost here.
Yeah, they charge, and they are so confused that many people who are working in leprosy don't get this flag. So I'm prepared to give it to any other company because now they don't have any exclusive rights to make it. So we can give it to other companies who would be not making profit but supplying for a cause. I'll be very happy to give and license MIP to any other company if you can find me one.
Thank you, sir. Actually, this is the real problem we are facing in
You are not the only one. I've heard from many others. And every time I tell to Cadillac, क्या हो गया भाई? ये करेंगे आपको कभी शिकायत नहीं आएगी, but it still continues. And reason I tell you all, I've already told you they sell it for other diseases at a high price.
because many benefits in the leprosy disease we also offer, but we are not able to use for the benefit of leprosy patients for what it was initially made of.
Right.
That's why.
Right. But please find me an alternate company which should work for a cause rather than for only profits. Find me that and I'll license it to any other company. I'll be very happy to do so because what you have told me has been said by many. And ultimately, I found that they may be making so much money on psoriasis and other things for which they are selling the same myth under different names. Any other issue? Well, thank you very much for your little and for appreciating whatever I did. Thank you.
So, we thank you again. Thank you all for being patient in here. Thank you so much. Cheese with that. Just check with
Thank you, Chairpersons. Your presence has truly enriched the proceedings and ensuring the session flowed smoothly and meaningfully. Now, I would like to request our respected chairpersons to kindly present our respected Professor G.P. Talwar with mementos as a gesture of our heartfelt appreciation for enlightening and inspiring talk today. Now, I would like to invite Dr. Ambak Rai and Professor Amit Avasthi, President of IIS, to kindly come forward and present token of appreciation to our respected chairpersons. As we now move forward, we now arrive at very another proud and special moment, the presentation of IIS Mid-Career Scientist Award. May I have the honor of inviting our respected chairperson, Dr. Santosh Kumar Kar, who is former professor at JNU and vice president of IIS. Dr. Uma Kanga, who is additional professor at AIMS New Delhi and vice president of IIS. Dr. Sarat K. Dalai, who is director at Institute of Science, Nirma University and vice president of IIS, and Dr. Arvindan, who is associate professor in the Department of Genetics, University of Madras, and vice president of IIS. All the dignitaries are requested to kindly come forward and lead the felicitation ceremony and present the IIS Mid-Career Scientist Award to our distinguished awardees.
ज़्यादा वोट मिलोगे तो फिर निकल जाएगा all again to the prestigious award ceremony of the society, Nuclear Sciences Award. And as per the society protocol, applications are received and screened, and we have selected Professor Gibraltar Award. Much better. And I will request you to introduce feedback. It's a pleasure to introduce Dr. Girdhari Rao from is a senior scientist at the National Center for Cell Science, Pune. His research focuses on understanding the cellular and molecular mechanisms of immune tolerance, particularly chemokine receptor signaling and neuroimmune communications in inflammation and autoimmunity. His laboratory is investigating the chemokine receptor signaling and autoimmune communication in inflammation and autoimmunity. Dr. Girdhari Lal, your floor is yours now, and your presentation is for 25 minutes, and then there will be a felicitation.
All right. Thank you very much, sir, and thank you, Indian Immunology Society, for this honor, and it's really wonderful. I can tell that from this society, I'm almost like 20, more than 20-year associated and learning from society or learning immunology from there, teaching and training. So it's wonderful to forum to learn, and it's more even important today because we have centenary member. Who actually founded the society. The members are here also in the dais, those who actually started the society and presenting the information what we learned actually in the laboratory today. So, like sir says, we work on autoimmunity and tolerance. And when I say tolerance, tolerance you can imagine tolerance in many facets. Let's say tolerance in autoimmunity, tolerance in transplantation, tolerance in cancer as well. So now the biology, how tolerance is created, that's what scientists do. That's what the laboratory in NCCS we do. Working from last 15 years at NCCS, we had several important discoveries in chemokine receptor signaling, how it works in autoimmunity. I'm not going to discuss that part today. I think that there's something new and exciting going on in the laboratory. So I thought let's discuss something is neuroimmune communication in autoimmunity and tolerance. So neuroimmune communication, when we say we got excited almost like 7 8 years back, that why immunology and why neuroimmunology, where is the problem? So looking back, actually, if you look at different aspect of immune system, what you will find that sometimes your own response, which is psychological or neurological response, alters your immunity. Now if it is so, how it is so, and what are the molecules, what are cell type involved in that, that's what curiosity will us to learn something more about neuroimmune communication. And the publication this came in 2017, that also makes it more interesting why we do this type of research in India. What you see here in the graph, I mean, if you look at the green versus red, there are a lot of neuropsychological disorders or neurological diseases which are predominant. And one of the quotations says that 1 in 7 Indian were affected by mental disorder. Okay, so if you have mental disorder, and if you look at those different types of disorder, either conduct or the anxiety disorder, you will find that each state fares differently. And I would like to point upper two graph. The red bars are more on the bottom state or southern state of the country than the northern part. That makes more interesting, and that's what most of those states are more rich, are most financially stable. Now, accordingly your mental disturbance also goes up. That way, when they actually studied later, they published this study in terms of looking at communicable disease versus communicable neurological disorder versus non-communicable neurological disorder, which makes it more interesting. So now, if you look at this, when I say communicable disorder, that means you are infection-induced neurological disorder, which is there in the bottom. Let's say encephalitis, meningitis, or tetanus. There are several other autoimmune diseases, which is again neurological consequences. And the top graph on the right hand side, what you see, the non-communicable neurological disorders are dominating in the state, which are like just we mentioned. Now it's a concern. It's a concern in say that if autoimmune disease are actually responsible, do we know the biology of it? Okay, moving ahead, what I'm going to discuss is autoimmunity. So let's look at how autoimmunity perceives. If you look at most of autoimmune diseases, if you are more psychologically stressed, or it can be financial with family, social, or whatever ways you have more neurological disturbance in your body, most of these autoimmune diseases flare up. That means it's suggesting us there is something goes on wrong with the neurons, and the immune system just see as a symptom of the disease. Now, if it is so, if you go more closely and you look at it, most of depressed people have more of inflammatory cytokine up. And if you go to clinic, for them, the patients are prescribed anti-inflammatory drug to bring down the inflammatory reaction, and that also brings down the other neurological disorder as well. So there is very strong connection with neurons. There is very strong connection with how these neuroimmune system talk to each other, and that's what our focus was. That's how we got the interested in. Now, when we talk about systems or regulatory systems in our body, there are several of them. Three most important one: the immune system, nervous system, and endocrine system. Most of the time, what we study is either immune system or nervous system or endocrine system. Now, if you actually altering the new immune system, you see the consequences are nervous system. I just gave example how inflammatory, anti-inflammatory drugs to psychology drug, or if you in for example, let's say in anti-cancer drug, right, you try to boost the immune response so high that to kill the cancer, but what you see, these two things are also goes up, and we label them as toxicity, neurotoxicity and endocrine. They are actually naturally responding to them. The one system you disable or disturb, others also get disturbed. Now question is, if we want system to bring back to the normal position, we need to know how these systems are talking and how we can actually balance them. Also, very important example I'll give you. If you look at endocrine system, if you look at early age of girls, you see that when they have hormonal disturbance, they see the pimples on their faces. Now, pimple on the faces is immune reaction, disturbance created by hormones. Now, you see all those changes, the immune response and endocrine system, how they are talking. Another example, the menopause, which when the hormonal disturbance happen, that immune system and all other body function changes. So there are regulatory connection. We need to understand the biology of it. I'll give you one example here. This is very simple. It's very interesting, but very simple experiment done. What is done here? The nerves which drains to the sciatic nerve, which drains to the lymph node, is just surgically disconnected. Now, if you surgically disconnect, you see the size in the middle, the lymph node swells up. And if you look at all those immune cell types, all of them are disturbed. So now, if disturbance of these neuronal connections making the immune system goes haywire. So that's how we need to now revamp in a way that how disease initiates. Is it immune mediated or somebody else induces the immune response to see what we see as disease or symptom? Now, since I'm going to talk about gut, gut is very interesting organ. If you look at them, 70 to 80% of our immune system line across the gut lining, not in circulation, not in other tissues. That makes it more sense. In terms of the gut also drains almost like 100 million neurons all the time. What goes on in gut, and that's why probably people label it as second brain. That's where we're trying to focus on. There are how these neuron transmitters, how neuronal signals transmit signals, to the immune cells. And when it comes to the neurotransmitter, I'm going to take just one example today to discuss. We work on several other as well. Acetylcholine. We know that cholinergic systems, neurons release acetylcholine and then it transmit the signal from one neurons to other or to the one system to other. And there are very important component to it. There are acetylcholine receptors. There are two types, nicotinic, muscarinic, and also the enzymes required for acetylcholine synthesis, that is acetylcholine or choline acetyltransferase, and also the neurotransmitter is degraded quickly, that is acetylcholine esterase. These molecules actually require for synthesis. Now, what we know most about all of these things are neurons producing this neurotransmitter. But if you look closely, there are immune cells do produce these neurotransmitter as well. Now you have two sources of acetylcholine. One which is coming from neuron, other coming from this other source, non-neuronal origin. And now the question what we ask: does this non-neuronal origin of acetylcholine or the acetylcholine, how they actually impact the immune system? So we use a gut inflammation model. So we use IBD patient. I'm not going to discuss. I'm sure most of you will know IBD, inflammatory bowel disease. In that case, what happens if you look at the colon, you will find that there are a lot of inflammation in the gut. And if you look at histology, you'll find a lot of immune infiltration. Like I mentioned, there are 100 million neurons draining. Question is, how does they are altering? What are neurological disturbance you see in the gut, and which is also driving the immune system to have the pathological consequences? Obviously, you can go ahead and do all those the RNA-seq. You will find n number of genes up and down. Now, question is that do we see molecules or molecule responsible for this neurotransmitter receptor signaling or neurotransmitter production? And if we look closely, what you saw that the cholinergic pathway, there are a lot of molecule, either receptor or enzymes or the molecules which can alter or involve in those things. They are clearly different between IBD and the control biopsy. This is RNA-seq data. And if you look at the enzyme from the colonic biopsies of the patient, the chat which is required for synthesis of acetylcholine is actually going down. And if you look at the serum, the choline, which is one of the precursor required for acetylcholine production, that also goes down. So what it suggests that there is actually deficiency of neurotransmitter signal in the IBD patient. And if you look at those with the serum, I think it's hiding the lesion. You will not able to read. So the square is representing the IBD patient. You see the strong correlation between the choline and chat mRNA when actually the disease is onset. So it means there is actually disturbance in choline. In fact, in the clinic, patients are prescribed for choline-rich diet. The IBD patients are prescribed, and it works for some of them. Now, looking at more closely to just only some other things like cytokine or immune response gene, you again start seeing there is total alteration in the system. And one of the cells which we're discussing about FOXP3, which is also you see in IBD patient, actually the enzyme expression actually pattern changes.
Dr. Gidari, you could use the mouse on the system.
Yeah. Okay.
Probably it works. Yeah.
All right. So, is it working? Because I'm using here is it's not displaying it, it's displaying here. So maybe it's not connected in the way the projector in this. Anyway, so what you see actually here with the chart expression, chart is again is enzyme required for synthesis. They are totally altered. But our interest was not just chart in the tissue or in the gut tissue. Is it immune cells also produce the chart or neurotransmitter? If it is, how it impact? That was the question to begin with. So to do that, we actually purified the different types of T cell because our interest was originally in the T cell. So you can actually purify all of these cells, Th1, Th2, Th17 or Treg cells by flow cytometry, and then you can do RT-PCR or protein assay to see that how these enzymes are produced. What you see on the right, on the bottom left hand side, two of the cell type Th2 and Treg cells, they are actually having lower chart expression. Now, if you look at other enzyme which is required for degradation, they are not much different in those cells. What it means that the cells, non-neuronal origin T cells itself is producing these neurotransmitter, and their production of these enzymes are altered between the cell type. What we are discussing here between inflammatory, which is Th1, Th17, and regulatory or Th2 or Th2 type or Treg cells. Now, obviously, if you want to do a lot more study, you can't do in humans. So we have to move on to the mouse model. So in mouse model, you can induce colitis. So first, we just ask question: where are these chart enzyme in the mouse? So what you can see here, the red color, which is representing chart on the screen, different organs. You can see that it is expressed on almost all tissue. And if you look at the cell type, the different types of differentiated cells are also expressed. Now, this data is coming from chart EGFP mice, where chart is actually expressing along with the fluorescence protein. So now you don't need to kill the cells. You can easily GFP can be measured by flow cytometry. So now you see that all of these cells are expressing this chart molecule. Now, to test that, how does it work in inflammatory condition? The expression of this pattern changes. What we did, we took this mice and gave the disease colitis, and now you see that chat expression actually in the colitis induction it is decreased, suggesting that chat expression actually somehow protecting the immune system, protecting from the pathology of the disease. To understand more details, what we did actually, we started looking in these mice, in chat EGFP mice, who all are expressing in what are different types of tissues, including spleen, mesenteric lymph node, or Peyer's patch, which is nothing but gut-associated lymphoid tissue, including spleen outside. Now, what you see in the red arrows, those are like what I showed you, Th1 and Th17 cell, what was in human. You see that humans also, they are actually upregulated with the DSS treatment. And this is the cell what we are seeing, all these are gated on chat is GFP. So that means GFP positive cells, how they respond. We also measure the GFP negative, that I'm not showing just the lack of time, but important cell type which is contributing in pathology, including regulatory or inflammatory cells, they are altered. Now, to understand the biology in more detail, we want to make sure that is it the chart which is CD4 which is contribute in the CD4 which is contributing. So obviously we have to go to back to the mouse again, where we want to deplete or delete chart expression only in CD4 T cell. Rest every other part of the tissue in mouse is having chart expression. Now in that situation, we actually once we delete using Kree-Flock system, now you have mouse where CD4 is the only missing chat. And then ask question, if I induce the disease, you have all neuronal chat, all other tissues chat production or acetylcholine production. Now CD4 is missing. What will happen to the disease pathology? In that situation, what you see here, that the mice, which is here, the red arrow, what you see, the in the bottom is red square. If you actually deplete chat, you actually have much more severe disease. Now, what I showed you earlier, that whole mouse was either defective. Now, in this situation, only CD4 T cells are missing chat. They do not produce acetylcholine, and disease severity is much more. That means CD4 T cells are doing something by producing locally acetylcholine. But mind you that chat or acetylcholine production with neuronal cell versus non-neuronal cells, but physiologically is different. Neurons can accommodate or hold the acetylcholine. Whenever they signal, they can actually throw the acetylcholine or transmit that. Whereas non-neuronal origin cells, they have no way to control the mostly actually as synthesis goes on. So now the in the microenvironment, what it suggests that probably the non CD4 origin of acetylcholine maybe in the microenvironment is modulating the immune response in such a way that the pathology what you see is of more inflammatory in type. Now when I say inflammatory in nature, now come back and then see in these mice what are things going on. So upper panel what you see is actually the cytokines, um, intracellular cytokine staining. Almost all of these regulatory or inflammatory cytokines are altered as the disease pathology goes. And also, you can also see the reflection what goes on in the serum in the bottom rows. So again, it's hiding the clock is hiding the legend, but the first two, the square one is the one which is more effective. That one is treated and untreated, and you can make out that now when you treat when you have CD4 missing, you actually have altered inflammatory cytokine response going up. You can also go back and see that see them that they are actually having lot of these cell type alters in the gut associated lymphoid tissues. For example, I'm showing you the data for Peyer's patch and the laminar propria. All of these cells what we saw in human now just only altering effect through the CD4 T cells, the whole pathology is becoming disturbed. Now, to understand this, how does it work? Because in gut, regulatory T cells or TS17 cells contribute in terms of pathology and pathophysiology, and chemokine receptor do play major role. That's another area of laboratory which I'm not going to discuss how they modulate the function, phenotype and function. But I want to show you something here. Now, those mice, what you're seeing chart deficient, they are actually only CD4 chart deficient mice. Look at the FOXP3 cells, not much difference in terms of percentage. If you look at the amount of protein they express, then you start seeing the difference. If you look at the type of molecule they express, then that's a clearly that most of regulatory molecules are down if if you have deficiency of the chat in CD4 T cells. Now, if we zoom on a little more detail and ask question, what happened to those Tregs, those two types of Treg? You see, they are totally forming a different types of cells. Now, cluster 8 or cluster 6, you see the difference. They form a totally different types of Tregs when they are in vivo differentiating. These are in vivo data. And now if you want to see those, what is cluster 8 and cluster 6, if you look at the row 8, the molecules actually expression pattern of variety of molecules. So all this what you're doing seeing is all multi-parameter flow cytometry from same sample. That means all what you're seeing is on the same cell. So all these things give you variety of Tregs of different lineage. Why do we so why it is so important? Now, you know that Treg has to function it has to go to different organ, different tissue, interact with different cell type to perform its function. Probably nature has created a way to generate this diversity among the Tregs so that these Tregs can mobilize to different location, interact with different cell type using different molecules to adapt for the positive or regulatory function. All right, these cells, when we thought, okay, if they are compromised, let's test in vitro, they are compromised. You can also go ahead and do the for proliferation assay for the effector cell proliferation. They actually do suppress the proliferation. So I'm running low on time. So what I'll do, then they ask question if they are compromised Treg in the chart deficient, CD4 chart code deficient. If I use these Tregs and transfer into the mice, can it protect disease? And if you see in the bar on the right hand side, the square bar, if you actually transfer Treg cells from this defective, they actually not able to control disease. So this mice, this you can see the pathology where you have the knife. This experiment is done by adaptively transferring knife cells, which actually induces colitis within 3 weeks. And then if you transfer Treg cells along with the knife cells, you protect from colitis. And now if you're transferring Treg from which cell type, which origin, that's what actually you will see that how their performance are. So the upper panel, what you are seeing is from the colon. Like I said, these cells has ability to mobilize to other organ. Other organ also get affected with knife. Mice transfer that skin or GBSD if your mice is little heterologous to the system. Skin also protected from these Tregs, which are adaptively transferred. And also the survivability of mice is also protected. Compromise if you have deficiency in the chat deficiency. Now, what I showed to you is that there in the microenvironment, you have neurotransmitters produced by neurons. You have neurotransmitter produced by immune cells. And what I showed to you is specifically only on CD4 T cell. In the tissue microenvironment, their contribution in terms of inflammatory or regulatory function is effective, is altered, and it suggests that the neurotransmitter what we see is not just neurotransmitter, it's the interorgan crosstalk between the neurons and immune cells, as one of the milieu in the microenvironment. But here is the problem. Can we target this as a disease, controlling the disease, as using a neurotransmitter? Can anybody think of it? I cannot. Definitely not. Then what is use of this? That's part of probably story. The next time I'll discuss, but I can give you a glimpse that this neurotransmitter has to, acetylcholine has to bind several of neurotransmitter receptors, and the receptor expression pattern changes with the tissue and the distribution in the disease pathology. So now how they distribute, where they express, where they are to work on, that's actually a wonderful area to work on. That's what we're working on that. And with that, the note, I will thank all my students who participated, especially Amrita and Namrita, who did this acetylcholine work and the funding source, and also our clinical colleague at AFMC and DY Medical College and the facility. Thank you very much. I'll take the question.
Good luck. No questions. No question. So, I don't, I mean, our pattern says maybe one or two questions, why not? So, this is also, yes, please.
Yeah.
Well,
Both. It act on both. That's what I showed you, immunosuppressive data with the in vitro data with the inducibility rate and other one also. So it works on both. Right.
yeah, I had also the same question already you have answered. And then this neurotransmitter expression label also linked with the activation status of T cells.
Yes, it does. That's what I was trying to show in the when you differentiate from different stages, they do have different expression pattern, and that's why different cell type also behave differently.
Going back to your concept slides, as you are showing, endocrine, neuro, and immune. How do you categorize diabetes too? Because mostly people above 30 develop that. Mainly, as we consider, stress is a main factor. So
Right.
do you think such phenomenon would be applicable?
So I was definitely expecting or not expecting, but that's true. We actually thought of this, like I mentioned to you, diabetes. Then what we have other area of work going on neurotransmitter. One of them is called serotonin, the happy hormone versus stress. So when we start looking at the serotonin and serotonin receptor expression on immune cells, not acetylcholine, because you can't blame everything at one place. So when we look at the serotonin receptor expression, they also comes with 15 different types of receptor, and our focus was cancer, but then we found actually how diabetes is actually working through one of the serotonin receptor. That receptor is str2b. What we were trying to do is to protect the cancer, because a lot of these cancers have this. So we were trying to do experiment in mouse by agonizing or antagonizing this receptor, see that how tumor grows. And surprisingly, I asked the student to just do the toxicity. As I said, what toxicity I do in mice. I said, okay, go and measure the glucose level in the blood. That's a super easy, and you just follow up. We'll see the later what we'll do. When you agonize the STR2B, just like streptozotocin, you actually induce disease within 3 to 4 days.
Oh my God.
And alteration of this or action of this particular receptor also works on immune cell. We have a lot of other information, maybe some other time. But see the endocrine system, neuronal signals, what we label as, and the immune system, how they're responding. So there is a strong connection between these disease, how it works, and how to. So when we look at one cell type or one section of the disease as an immune system or endocrine other, probably missing lot of things. In biology is not just a one system. It's a combination of all.
Very interesting finding. There's a question there. on the-
Thank you. So
There's
it
a
was a
lady
nice
there.
talk.
No, she was there first. Yeah, go ahead.
Yeah, thank you. I was just curious. So you are looking for the plasticity of the cells, T cells, right? So have you checked the Th17 cells, which are important for this colitis development?
Yes, I
Yeah.
showed you
So,
the data.
oh, maybe I have
Right.
missed it.
So TS17 cells also upregulated. Tregs are down when you deplete the CD4 chat in the CD4 T cell.
These are the
So,
cells when in the adaptive transfer when you were giving
so
this.
both in
Cha-
adapt, yeah,
so
both,
these cells are changing.
yes, both in vitro as well as in adaptive transfer experiment.
So
They actually are do altered.
okay, and I have another question. Have you
Yeah.
gotten chance to look for the B cells there?
That's what we are looking at. There are a lot more to learn. B cell and also other cell types, we are looking at it. Yeah.
Thank you.
So
Girdhari,
I think we
one.
have to take only one last question because in the interest of time.
So,
Yeah.
Oh. Can somebody pass a mic to him?
so Girdhari, quick question. Congratulations, nice
Thank you.
talk. Is there any published literature of Jiva studies where these autoimmune diseases has been, assigned to some SNPs related to these receptors or?
There are now, once we started looking at, now there are a lot of things coming out. Yes. Yeah.
So do you have receptors for acetylcholine on the lymphocytes?
Yes.
And the how different it is from the neuronal cells in terms of its affinity constant or etc. etc.
Okay, so that's very interesting question. Neurons do have constitutive expression at different level, but immune cells do not have that receptor. So, for example, there are two of them, the nicotinic and muscarinic. We mostly interested on muscarinic. There are five different types of receptor, and when we look at all these five receptor on different, let's say Th1, Th2, or different lineages, their expression pattern changes, and the response to each one of these receptors from inflammatory to regulatory also alters. Now question is, if we want to target this receptor, we need to first understand what each receptor does and how it works. So yes, expression is altered through the differentiation pattern.
And the lymphocytes in the gut
Hmm.
in the inflammatory condition, as opposed to another peripheral lymphoid system, spleen. Do you see the difference in the receptor status?
Yes, there are some differences, especially if you look at gut associated. I do not have the data here, but yes, if you look at gut associated lymphoid tissue, there's a totally different expression pattern what you see in the spleen even or in circulation. Yeah.
Thank you. I think you've been very kind to accept questions. Although in the award ceremonies, we usually have a trend that questions are not asked, but it
It's
was so
science,
interesting.
so
It was
we
so
love
interesting
science.
that
Thank you.
people couldn't stop themselves. So, anyway, thank you so much for the wonderful talk. And we congratulate you for the award. And
Thank you.
may I request you to felicitate?
So now all of you should join. Sure. All of us will take him.
Thank you, esteemed chairpersons, for leading this wonderful ceremony and for your invaluable contributions and continued dedication to the Indian Immunology Society. Now we will be taking a short tea break, but before that, everyone is requested to please assemble outside the MP hall for a group photograph. We kindly request everyone to reach their respective venue within 10 minutes as we have an exciting lineup of sessions up ahead. After the tea break, the parallel sessions will begin at their respective venues MP Hall, Seminar Hall, LHC 3, and LHC 4. Kindly reach your respective venues on time so that sessions can proceed smoothly. Thank you and enjoy your tea break.
priority because our mind is the first.
टेक्निकल टीम को दे दूं
your name.
ओके
इधर आएगा. Okay, let's try. It's one. Okay. And now I'm speaking to you. Sir, SP sir, SP sir, change करें. Okay, great. तो 50 from the page out. Okay, here. नहीं नहीं so timer मतलब कि I just have to judge बस बता दो okay okay बस चलेगा ठीक है I think it should be okay I just need to connect and start the previous okay I'm over there okay yeah all good thanks Hello. Hello, hello, hello. Hello, 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 hello. Come है. Hello, check. आ रही है. Come है. Okay, I'll do that. अजीत सर, बाहर वाले स्पीकर की आवाज बढ़ा दीजिए. Hello, 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 hello. Check, check, check. हेलो. सही है. सही है. पक्का.
Everyone is requested to reach your venue within 5 minutes as we are going to start the session soon. Also, the chairperson are humbly requested to reach their respective venues on time too.
Everyone is requested to reach your venues within 5 minutes as we are going to start the session.
Also, the chairpersons are humbly requested to reach their respective venue on time. I request Dr. Pravesh Gupta and Dr. Karthika Rajiv to please submit their presentation to the technical team. I also request them to please enter the MP hall as soon as possible. May I repeat, Dr. Pravesh Gupta and Dr. Karthika Rajiv, please submit their presentation to the technical team. We are starting the sessions in 2 minutes. Please reach back to your respective hall as soon as possible. Final announcement, we are starting the sessions in MP Hall. I repeat, final announcement, please reach back to MP Hall and the respective halls as soon as possible.
Welcome again, ladies and gentlemen. We hope you all had a refreshing short break and are ready to dive back into another exciting scientific sessions of IMMUNOCON 2025. It gives us immense pleasure to welcome you all to this scientific session currently ongoing in three other different halls. Here in MP Hall, we will proceed with very insightful session on modulation of immune responses. It This session promises to unfold fascinating insights into how our immune system can be turned, regulated, and modulated, a topic that lies at the very heart of modern immunological research and therapy. As we begin, we take this opportunity to extend a very warm welcome to our distinguished chairpersons for this session. We invite Professor Deepankar Nandi, who is from Department of Biochemistry at Indian Institute of Science, Bangalore. He completed his PhD with Nobel Professor James P. Allison at US Berkeley and postdoctoral training with Professor John J. Monaco at University of Cincinnati. Since joined IISc in 1997, his lab has focused on T cell activation, interferon mediated nitric oxide responses, and a microbial cell mechanism. Combining genetics, biochemistry, and immunology, he has mentored 23 PhD students and remaining deeply committed to research and teaching. We scheduled two chairpersons for this session, but due to unavoidable circumstances, Professor Amit Kumar Dinda will not be able to join us today. So this session will only be supervised under Professor Dipankar Nandi. With our esteemed chairperson now on the dais, I would like to request Professor Nandi to kindly come forward and take charge of this session.
We'll start off with this. Got it. Got it. Yeah, fine. Take it. Take it. Okay. Fine. Where is, right. So, a very uh, good day to everyone. Let's start off with this session. So, the first speaker, as I see, is Dr. Arvinda. So, Dr. Arvindam, I have information over here, is an associate professor in Department of Genetics at University of Madras. His research focus is on immunogenetics, diabetes immunology, and infectious diseases. He has published exceedingly well and received several grants and and mentors many students. Dr. Arvindam, please. Yes.
Very good afternoon, one and all. First of all, I would like to thank the organizing committee for giving me this opportunity. My association with the Indian Immunology Society starts right from 2003, where I first presented in Lucknow, Sanjeev Gandhi Institute. And at that time, I presented my PhD work RBCG, and Professor D.N. Rao was the chairperson. So it's almost more than 20 years where I come back here as a principal investigator. And it is an emotional moment for me because I'm presenting again as a principal investigator before the same distinguished immunologists in our country. So the topic today, which I'm going to present to you, is metabolic hygiene hypothesis, a game changer in the field of diabetes. Now, I have been working in the field of diabetes for almost 15 years now. Basically, we concentrate on type 2 diabetes, and there have been very many serendipitous findings which we have identified. So, these are few things which I would like to share with you. Now, let's start with hygiene hypothesis. I am obviously not going to bore you with what hygiene hypothesis is here, because like almost all of you would know what hygiene hypothesis is. And the most important thing I just highlight, hygiene hypothesis was first put forth by in the year 1989 by an epidemiologist, okay, Statram. So this is what is very catchy about it because he's not an immunologist, he's not even a biologist, he's an epidemiologist who got trained in statistics. But he was able to, purely based on statistical analysis, he was able to come up with this monumental hypothesis. So what does the hypothesis say? Childhood exposure to infections is absolutely necessary for training the immune system because only when the immune system gets trained properly during the early stages, it would reduce the risk of autoimmunity and allergic diseases, which means infections, your childhood exposure to infections definitely reduce the incidence of allergies and autoimmunity. So this is what the hygiene hypothesis mainly says. And from that time point, there was no turning back. There have been tremendous research which is happening in the hygiene hypothesis. Now, as I told you, I've been working on type 2 diabetes for quite some time, almost 15 years now. And basically, like diabetes is of three types, type 1, type 2, and type 3. Sorry, not type 3, gestational diabetes. And of course, there are minor forms also.
And immunologist's favorite target is type 1 diabetes because it is an authentic type 1 autoimmune disease. But I decided to work on type 2 diabetes because that is the most prevalent form of diabetes in India. It is a metabolic disease. It has been classified as a genetic disease also. But the current census is that we should call it as a inflammatory, chronic inflammatory disease because chronic inflammation plays a very important role in gestational diabetes. We have done some amount of work on gestational diabetes also. Now, I spoke about the hero or the star player. Let me also talk about the not so famous person, lymphatic filariasis. This is a largely neglected disease, but for a long time, Indians have been suffering from this disease. India has long been identified as an endemic country for lymphatic filariasis. Okay. And among the affected population in the world, 90% of the individuals are subjected to LF. In India, the prevalence of LF is endemic with about 553 million people at the risk of acquiring the infection. The prevalence of LF in Indian individuals is distributed at filarial parasites carriers as well as those with clinical manifestation. But the most important thing which I would like to highlight here is there is tremendous amount of interface between metabolic diseases and infectious diseases, which has not been properly explored till date. And whatever I'm going to show you is basically originated from a serendipitous finding. But since, like in the 15 years we have put everything in place, I'm able to formulate it as a hypothesis and then present it to you. Okay. Now, coming to the scope, so basically these studies were conducted at two parts. The first part is predominantly serology and epidemiology based. Second part is predominantly molecular immunology and cellular immunology based. Meta-inflammation in is chronic inflammation associated with metabolic diseases. It leads to insulin resistance, which is a major etiological factor for type 2 diabetes as well as cardiovascular disease. Finally, infection confirmed immunomodulation against inflammation. The relationship between immunomodulation, meta-inflammation, and insulin resistance is largely unexplored. And that is exactly the point which we wanted to explore. So the working hypothesis which we had is when you look at infections in toto, you can broadly classify them at two types. The bacterial and viral infections are predominantly pro-inflammatory, which means they would induce meta-inflammation. On the other hand, helminths are predominantly immunomodulatory, which means they will inhibit meta-inflammation. So when helminth inflammation is there, then automatically it downmodulates meta-inflammation. And because of this, it reduces the risk of insulin resistance and beta cell loss, and to some extent confer protection against diabetes. So the initial questions which we asked, what is the prevalence of filariasis? That is how the whole study started in type 1 diabetes, in type 2 diabetes, as well as cardiovascular diseases. So this is, I was very, very lucky enough to start my career at Madras Morgan's Diabetic Research Center. So they have conducted a huge epidemiological study in Tamil Nadu, and they had almost 4,500 serum samples which have been very well characterized. These serum samples belong to various grades of diabetes, and Dr. Morgan was kind enough to give me those samples. All the samples which I have used in the study come from phase 3 of the cure study. If you just type it as cure study, you will realize like there are more than 200 publications which have come out of this particular study. Okay. So the first experiment which we did itself actually disproved our hypothesis. Our hypothesis is that chronic diabetes impairs immunity, which would make individuals more susceptible for filariasis. But as you can see in the first figure, the prevalence of filariasis actually drops down when you grow from control individuals to pre-diabetic individuals to newly diagnosed individuals and to chronic diabetic individuals. So definitely, rather than synergistic interaction, there is an antagonistic interaction between diabetes and filariasis. We saw the same thing for type 1 diabetes, and in type 1 diabetes, it is expected because we already have the hygiene hypothesis. But this phenomenon was not seen in CAT patient, coronary artery disease patient, which gives us a clue that the kind of meta-inflammation which we are talking about in metabolic diseases are way different in type 2 diabetes and cardiovascular diseases. You cannot club all the metabolic diseases under one umbrella and call them as meta-inflammation. Okay. So based on these preliminary experiments, okay, we designed The next set of experiments which were targeted to decipher the mechanism behind this immunomodulation. So the questions which we addressed was, what is the effect of immunoendocrine axis in DM, and what is the effect of LF on cellular immune response in DM? So here we followed up the patients, collected the blood samples, then we stimulated them with TLR2, TLR4 ligand as well as filarial antigen, and did a series of experiments which looked in for cytokine chemokines by ELISA, QRT-PCR experiments, FACS experiments, redox plus markers, etc. Okay, what is the effect of lymphatic filariasis on immunoendocrine axis in DM? So this is a complicated side. Obviously, I'm not going to go in for all the details. This is basically a serological side which was carried out. We had 4 groups: control group, LF group, DM group, and DMLF group. But the important markers which showed up here are IL-1 beta and alpha defensive. Okay, this is pertaining to the innate immune markers uh, which were studied. Okay, then we looked in for adaptive immune markers, and none of the adaptive immune markers were very different from filariasis and DM filariasis. And then we went in for chemokines, and in the chemokines, the one which actually showed up was IL-8, which is very important. And this was the third player which plays an important role in this mechanism. Then, because it is immune endocrine, we look in for important hormones as well as growth factors which could play a role. And as you can see, the 3 important hormones, insulin, adiponectin, leptin, didn't show much effect. But VEGF is one thing which was significantly augmented in DMLF subjects. Okay. Then we looked in for T cell exhaustion as well as Tregs, and there was no much difference here. And then we looked in for redox markers. And as you can see, filarial infection on an overlay of diabetic background reduces lipid peroxidation. There were no obvious protein peroxidation here. Nitrate metabolism was not much affected. So this is a kind of working model which we have. As I told you, the important players here is IL-1 beta, IL-8, VEGF, and reactive oxygen intermediates. And these somehow coordinate together and then try to reduce the inflammatory burden, thereby conferring protection against diabetes. Then we look in for innate immune responses, where we studied toll-like receptor responses. We chose TLR2 and TLR4, and IL-27 is an important player which showed up. And adaptive immune cytokines, IL-12 is an important marker which showed up. IL-33 is another important marker which showed up. IL-9 is another important marker which showed up. Chemokine MCP1 is a marker which showed up. STF1 showed up. COX2 was differentially expressed. IDO was expressed. And then we looked at MSC and co-stimulatory molecules, no much difference. We looked in for adaptive immune response using LF antigens. IL-12 showed up, IL-9 showed up, IL-33. The same 3 cytokines showed up. STF1 showed up. Okay. And then we looked in for T cell activation as well as TS polarization. TH9 cells were significantly higher. Okay. So I just rushed through the whole thing because obviously you can imagine the amount of work that has been done. But coming to the summary, childhood familial infection can significantly reduce the risk of metabolic diseases like type 2 diabetes, which we are calling as metabolic hygiene hypothesis. Initially, it was called as extended hygiene hypothesis. We are calling it as metabolic hygiene hypothesis. Filarial infections, like bacterial and viral infections, can dampen meta-inflammation by means of immunomodulation, which is the root cause of insulin resistance, which leads to metabolic derangement. Filarious-mediated immunomodulation takes place by modulating the immunoendocrine network. Identifying and characterizing filarial products which could confer protection against diabetes could lead to novel therapeutic avenues in conferring protection against diabetes. Finally, decreasing incidence of LF, this is the most important thing, by means of mass drug administration could have a serious impact on the increasing incidence of metabolic disorders, which we are currently witnessing. Thank you so much.
Thank you so much, Dr. Arvindan, for seeking to time. Can we have 1 or 2 questions? Yeah. Priyanka?
Hi, Arvindan. Great presentation here.
So I'm just wondering that you mentioned about meta-inflammation, right?
Yeah.
And in case of meta-inflammation, we also call it as a low-grade systemic inflammation,
Yeah.
right? So in that term, you have showed data for type 2 diabetes. And do you have any insights about adipose-related inflammation? Because as you know, meta-inflammation adipose plays an important role. So do you have any insights on that?
Yeah.
Thank you.
So the thing is, because these are all clinical studies, we couldn't have access to the adipose tissue. But we also plan to do some animal experiments where taking adipose tissue and then look at local inflammation. Right now, we haven't.
Yeah, I think, yeah, there's.
So,
Initially, are the diabetic people, those are infected lymphatic malaria?
yes, sir. So what we did was we took a cohort of diabetic people, screened all of them for LF, and stratified them as LF positive and
Did you monitor the glucose levels?
yes, sir,
See
we
if they
monitor. What happened is almost we did a follow-up study after 10 years. At that time, many of the LF positive individuals have actually become LF negative individuals. Once the LF goes down, the protection goes down. It is not permanent.
Yeah.
Yes, yes.
whether HB1C was also looked into.
Yes, sir. We looked at HbA1c also. It was significantly lower in LF positive diabetic patients compared to LF negative diabetic patients. Yes, sir.
It was fantastic that you
Yes, sir.
see why I'm telling this. The presentation I mentioned, since it's coming back. Since, as you just mentioned, is a very
Yes.
the immunosuppressors and the human is very viral
Exactly.
and bacterial immune.
Yeah.
Why I'm asking you filariasis is because I also did. It's not the sense that now filariasis. Say I will shorten it, but they will start using the ivermectin albendazole is used by government of India to for these
Yeah.
which I'm resisting because albendazole it is effective. You have any thought? Do something. Social platform because we can provide this. Can we work together?
Definitely, sir. That would be my pleasure. I will
I
definitely.
think to you or to reflect in discriminately given in all of
Yeah.
for
Infections in the stomach.
I'm very this is a wonderful opportunity.
Thank you, sir. My pleasure.
One last question by a student over there. There. So I encourage the students to ask questions. This is a good platform for you all.
Thank you, sir. It was a really great lecture. The diabetes is causing the compromising the immune. Check the any comorbidities as sir was there, some other virus.
So the thing is like there is one more comorbidity we are looking at, that is tuberculosis. But as you know, filariasis and tuberculosis are way different. Tuberculosis is pro-inflammatory, filariasis is anti-inflammatory. So the picture which we are getting for tuberculosis is much more complicated and multifaceted. Of course, I do not have time to present these things here. Maybe some other time I will present on diabetes tuberculosis.
Thank you.
So let us all thank for a very nice talk.
Thank you.
Thank. Okay, we'll move to the next one. The next one is by Jyoti Prasanna, who's a professor and dean at the Manipal Institute of Regenerative Medicine, where she leads a research at the intersection of stem cell biology, immunology, and hepatobiology. So I welcome Jyoti, and please go ahead.
Good afternoon, everybody. It's not starting. Yeah. So today I'll be talking about an enzyme called as 3' 5' ectonucleotidase. Why is it interesting from an immunological perspective? is that on the left-hand corner, you see ATP, and the right-hand corner, you see adenosine. ATP is pro-inflammatory in nature, and adenosine is anti-inflammatory in nature. Although there are several ectoenzymes which convert ATP to ADP, there is only one unique ectoenzyme, which is CD73, which converts AMP to adenosine. So this enzyme is not specifically expressed in particular cells. It is expressed in many barrier protection cells like the macrophages, mesenchymal stem cells, and endothelial cells. And we'll be talking about how this enzyme would help in protecting tissues uh, with respect to inflammation. So why this is important is like there are several purinergic receptors which bind to ADP, ATP. So in case there is over-exaggeration of these purinergic receptor signaling, when CD73 is inactive, there can be exaggeration of sterile inflammation by activation of the inflammasome. Uh, Apart from that, this enzyme is also activated by inflammation and also by HIF1, which is a mediator of hypoxia signaling. So why we got into studying this enzyme was because of the fact that we were working on mesenchymal stem cells. These stem cells differentiate into stem cell lineages like the adipogenic differentiation and the osteogenic differentiation. And these cells express a huge amount of CD73, which is active on its cell surface. When you co-culture these mesenchymal stem cells with T cell blast, as you can see that there is a suppression. However, the suppression mediated by mesenchymal stem cells is dependent upon inflammation sensing, because IFN gamma receptor knockout mesenchymal stem cells fail to immunomodulate. So the sensing of inflammation is very critical for the immune modulatory properties of mesenchymal stem cells. So the first question we asked was, if you generate NT5E knockdown mesenchymal stem cells, would they differentiate? So they show skewed differentiation pattern towards osteogenic differentiation. So the osteogenic differentiation is enhanced, whereas the adipogenic differentiation is reduced. This is important because osteogenesis is, there is a role of interferon gamma signaling with respect to osteogenesis. I'll come back to it in a short time. So what happens to the immunomodulatory properties of CD73 knockdown MSCs? As you can see that the blast suppression is not mediated by your CD73 knockdown MSCs. In consensus to this, you can also see a decrease in a plethora of immunomodulatory molecules which are secreted by mesenchymal stem cells. Sorry. So one of the obvious molecule which we wanted to look was IDO, because as I showed you that interferon gamma knockout mesenchymal stem cells do not immunomodulate. We wanted to see whether CD73 knockdown MSCs do not modulate because they are not sensing IFN gamma. And as you can see here, the prototypic enzyme, which is regulated by interferon gamma, IDO, is downregulated in IFN gamma treated CD73 knockdown MSCs. What does that mean? It could also mean that either the interferon gamma signaling is not going through or the receptors have been downmodulated. And to our surprise, we saw that in CD73 knockdown MSCs, IFN gamma receptors are indeed downmodulated. Now, when we talk about mesenchymal stem cells as therapeutically relevant cells, the therapeutic efficiency of mesenchymal stem cells is dependent upon inflammatory sensing, and they are more effective in cases of inflammatory disorders. So, when we talk about controlling inflammation, the key cell types which come into our mind are macrophages, because macrophages are the central cells which could actually regulate neutrophils, T cells, B cells, and other cells of the immune system. So we were looking at literature that how is MSCs actually mediating their therapeutic effects, and we came across a paper which says that if you deplete macrophages in the cells which have been in contests where MSCs have been transplanted, then MSCs are not able to mediate the immunomodulatory effect, suggesting that it is in fact the MSC educated macrophages which are in fact doing the job. So in a published data, we had in fact shown that mesenchymal stem cells tone down macrophage inflammation, and this is very contextual. When there are M1 macrophages, they downregulate it, but if it is an M2 macrophage, it could upregulate it. So the usage of MSCs for transplantation could be a double-edged sword, and it depends upon the inflammatory context. And when we see CD73 knockout MSCs, when they are co-cultured with your primed macrophages or M1 macrophages, they do not immunomodulate like your control cells. As you can see here that there is a reduction in TNF alpha production with control MSCs, whereas CD73 knockdown MSCs do not do the same job. So why is it not doing? We tried to look at a plethora of immune modulatory molecules which are produced by your mesenchymal stem cells. And as you can see that CD73 knockdown cells do not secrete immunomodulatory molecules like TGF-beta, but overexpression of CD73 rescues this effect. So is the case with respect to your IDO. So the similar effects were also seen in context of primary macrophages, where either overexpressing IDO or overexpressing CD73 could rescue the effects which were abrogated in CD73 knockdown MHCs. So we published this paper which says that mesenchymal stem cells could immunomodulate only when CD73 is active on its cell surface. So next we were working on endothelial cells, the other barrier protection cells. And as you can see that in human aortic endothelial cells, CD73 knockout results in an EMT-like transition. It also, there is an abrogation of angiogenesis, as you can see in the capillary tube formation. And this is a fibrin bead gel assay. As you can see here, this mimics the sprouting angiogenesis which happens in vivo. When in response to inflammation and hypoxia, one of the cell in the endothelial system is annotated as a sprouting cell, and this sprouting cell leads the angiogenic program. As you can see in mutant cells, the sprouting angiogenesis is compromised. So apart from that, in your CD73 knockout HAAC, specifically in response to inflammatory factors like TNF alpha, the reduction in capillary-based angiogenesis is further exaggerated. Apart from that, there is ectopic expression of RUNX2 and OXTREX, which are ossifying factors. There is a decrease in anti-mineralizing factors in mesenchymal stem cells, suggesting that they play a very, very important role in endothelial functions and angiogenesis. So what is this relevance? So since CD73 plays a very, very important role in macrophage polarization in MSCs as well as in endothelial cells, we thought we could ask this question in a tumorogenic context, because the tumorogenic context also has all these cell types. As you can see here, the tumor microenvironment is a very complex environment. And of course, it has different cell types, and the immune landscape in the tumor microenvironment is continuously being sculpted. And we wanted to ask, what are the key molecules which could be determining the sculpting of this tumor environment, and can CD73 be a predictive marker? So during that time, we looked at the literature, and we saw that CD73 deficient mice have, in fact, increased anti-tumor immunity and are resistant to experimental metastasis. So it became an interesting question for us. During that time, interesting student, a pathology student, joined our lab, and she was good at histopathology, and we started looking at oral cancer and the different pre-malignant lesion conditions. And we wanted to ask the question whether there is a change in CD73 and TGF-beta signaling. As you can see here, TGF-beta signaling can shift between a tumor suppressor role or tumor promoter role, depending on whether it's the early stage of cancerogenesis or late stage of cancerogenesis. So you can see here that in the different pre-malignant conditions, ONM is your normal mucosa, OSMF is your fibrotic condition, OLKP is leukoplegia, and OSCC is the really notorious carcinoma situation. You can see a differential expression of TGF-beta in this context. So we wanted to ask similarly, we see differential immune infiltration in all these OMPTs as compared to OSCCs. So we wanted to ask, is CD73 affecting the TGF-beta signaling axis? And as you can see here, in CD73 knockout gingival fibroblast, there is a attenuation of TGF-beta signaling. There is an attenuation. TGF-beta also in turn regulates NT5A activity, which is rescued by using a TGF-beta receptor inhibitor. So now we wanted to look at the expression of CD73 in all these cohorts. And as you can see here, OSCC has very high levels of CD73. OSMF has least, whereas OLKP has no expression in the epithelium, but in the stroma. So now we wanted to check what happens in the fibrotic condition where there is an enhancement of TGF-beta signaling. And as you can see here, in the fibrotic condition, CD73 is reduced. You don't have adenosine being produced. So it clearly plays the TGF-beta CD73 signaling axis plays a very, very important role in tumorigenesis or tumor prediction. So we further wanted to subcategorize the OSCCs into well-differentiated, moderate differentiated, and poorly differentiated states. And as you can see here, in the well-differentiated state, you have less expression. In the moderately differentiated oral cells carcinomas, it is very high. But in the poorly differentiated carcinomas, you don't have it. So clearly, the nutshell, which we feel is that in an inflammatory milieu, CD73 is upregulated. And this influences TGF-beta. CD73 tones down inflammation. TGF-beta in turn influences CD73 activity and influences the immune activation threshold. So the unanswered question, I have more unanswered questions than answers. So the question which we would be further asking in the lab is, does CD73 and TGF-beta form an excess in a tumorigenic context which sculpts the immune landscape of tumors? Can this be relevant in terms of immune therapy? So I think I'm on time. I would like to acknowledge my funding agencies who have been gracious enough to fund us, our clinical collaborators from the Kelly Dental Hospital, and all our PhD and master students who keep the passion in research alive, and of course my mentors, Dr. Kar and Dr. Nandi, who have sustained my interest in immunology despite being in a stem cell institute where I'm supposed to do more of stem cells. And this is my group. Of course, most of the time, sometimes they work and most of the time we enjoy. And of course, this is our beautiful institute. And we are one of the few institutes in India who have a master's course in translational immunology. And I'm looking for good faculty. I'd be open to consider their applications. Thank you.
Great. Thank you, Jyoti, for being absolutely on time. Can we take a few questions?
It's coming from.
Beta would be present in the tumorigenic
Very next week, cell is responsible
so
producing so much load.
So many cells express TGF-beta. For example, mesenchymal stem cells do express TGF-beta. Myeloid cells express TGF-beta. In a tumorigenic context, the TGF-beta levels change depending upon the stage of tumorigenesis.
The suspicion is on T regulatory.
Yeah, so the next step which we are going to do is to basically plex all these molecules together, look at CD73, Treg cells, FOXP3 positive staining, TGF-beta, phosphospat3, and see whether there are micro-niches within the tumorigenic environment which can help in prognosis and tell the stage of the
I have one
Yeah,
small
go ahead.
question. CD73 knockout, when you knock the CD73, so if gamma receptor 1 and 2 both are down, so how CD73 is doing down regulation of the receptor expression?
We think it could be via NRFs and also to aryl hydrocarbon receptors, and which bind to alpha gamma receptor and does it. But I didn't show that part because it's an unpublished part.
The next question is that related to this only. When you are doing the CD73 knockdown, knockout model, so this is specific to MSC and CD73 or is it global? Because
No, the
CD73
culture experiments which we did, there we did the knockout in MSCs. In endothelial cells, which I showed, it was biotic endothelial cells. So we did it with separate cell types and then try to understand it in biopsies, basically.
Because CD73 is present on the memory base cells.
Yeah, CD73 is very
Huh.
pleiotropic with
Yeah.
respect. It's expressed mostly in all barrier protection cells,
Exactly.
including it was first discovered in endothelial cells, actually. The activity was discovered in
Nice.
endothelial
Thank you.
cells.
Yeah. Jyoti, what a pleasure it is to see you presenting your work. Technology in JNU, then PhD in Indian subs. You are now come up to this stage. The reason that I'm talking to you, some of our work done time 5 years ago has shown TGF beta controlling the tumor microbiome and taking the environment caused by the cells actually are involved. Have you got any TGF beta cancer and hitting the tumor inside the microbiome, alleviating the suppression utilized by the cancer cells?
Sir, I think it is more about staging and the regulation of TGF beta signaling rather than TGF beta signaling per se, because you have the SMAD3 pathway and the SMAD5 pathway. Both of them can determine whether it will work in a suppressing or a activating form. And tumor microenvironments are quite complex. Right now, I don't have data to answer your question. But
would you be interested in developing
yes,
at least a
yeah,
dialogue?
yeah, yeah.
Thank you. Thank you. She is one of the very who have an MSc Clinical Immunology program. They had 7 students there, and this is the only where they have micro MSc
In translational immunology.
immunology.
So there are the reason for doing it is either we have people who love immunology or hate immunology. I don't know whether I can say that. So I thought there should be more youngsters who should start loving immunology and consider it as a field. So that was the
Young people are loving it. They have no opportunity to express.
thank you, sir.
Questions from students? Okay. If not, okay. Thank you so much, Jyoti. Let's give her a round of applause for. The next speaker is Dr. Kartika Rajiv. Okay, Kartika is a scientist E1 at RGCB Trivandrum. She's an infection biologist whose research focuses on microbial pathogenesis, host pathogen interactions, and cellular immunology with regard to chlamydial infections. Kartika, stage is yours.
Thank you, Dr. Nandi, for your kind introduction. This is my first time in IMMUNOCON. Okay, so today I will be introducing you to a new or a very recent discovery in our laboratory where we have discovered a new population of immune cells, that is neutrophils, that is supposed to actually kill the bacteria, is supporting the growth of the bacteria, and the bacteria is using this for host immune or suppressing the host immune response. So to give you an overview of the bacterium, this is actually a sexually transmitted bug. If you look into sexually transmitted infections caused by bacteria, chlamydia stands first and causing nearly 129 million infections every year, causing like 70% of this is asymptomatic in nature. That means 1 in 20 women are positive for it. As you can see, it can cause pelvic inflammatory diseases, sterility, and whatnot. So if you look into the bacteria, what you see here is then cells from the uterus, human uterus, and we have infected with bacteria. And actually, they cannot grow outside the cells. They can only grow inside the cell. So you can see the inclusion being formed, and they simply replicate inside. And they exist in 2 different forms. They go inside the cell, replicate, and come out, which takes nearly 48 hours. So when we were discussing or finding out why is this remaining asymptomatic was when we first introduced the bacteria to neutrophils and checked how that reacts. So normally you all know, me being immunologists, that neutrophils really act by netosis, right? So what we see is that when we introduce the bacteria to the neutrophils, they completely paralyze the neutrophils. They have different ways of doing it. One of this is basically using its own proteases to cleave away the receptors, and this was published in 2018. From there, I caught up and then investigated: these paralyzed neutrophils, do they persist or do they die? For this, first of all, I isolated, or the student basically isolated neutrophils from healthy individuals infected with bacteria for different time points. Very simple experiment. So what we see here is that soon after isolation, the bacteria, the neutrophils start dying. So this is quite normal with neutrophils. But upon infection, you can clearly see that they can block this effect. And it's evident in this graph here too. In red, you can always see the infected population. They can block the cell death. So now, are these bacteria viable is a very important question. So what we did, we just isolated these neutrophils and infected them, and then looked for the progeny formation. So what you see in these green spots are the infected bacteria in HeLa cells. So absolutely brilliant that 24 hours to 48 hours, they can actually stay inside these neutrophils, that they're potent killers, and just grow there. Okay. So now came the important question: what percentage of these cells are getting infected? So now we used an MOI of 10. That means one cell gets 10 bacteria. So irrespective of the MOI, I can say going from 10 or going till 100, not all cells gets infected. Only a 15 to 20% of the cells gets infected. The rest, we call it bystanders. So if you look closely, you can see not all cells are infected. Only few of them gets this infection. So what happens to this large population of the bystanders who are nearby? Are they all also able to block the cell death? So very nice. We just sorted them out into infected population, uninfected population, and looked for cell death. Interestingly, we can see that irrespective of being infected in that milieu, the bacteria can induce or block cell death in the neighboring bystander cells too. So we knew now that something that is secreted during the infection might be doing the role. So now we just took the supernatant of the infected cells and put it on fresh neutrophils. And exactly what we thought was right. This in red, you can see the supernatant from the infected cells. They can simply block or prevent cell death also in the bystander cells. So now came the important question: what is the identity of these cells? Why are some infected, some not? Okay, so we employed single cell sequencing right away to find this question, and we had really super good data in this. And what we discovered was that the neutrophils what we have seen in the textbook is not just one kind of cells. They exist in 7 different populations. And the infections, basically, if you look, you can see the UMAP here, which is a bit complicated. Let's get into the system. So here you can see upon infection, there's a new population of cells that is being driven into a long living phenotype, or LL neutrophils. So if you look into transcriptome, you can see the transcriptome in 7 different populations differ widely, and that what this N7 population, which is actually increased upon infection, is actually producing a protein called PI3. So initially, when I looked into PI3, I thought it was PI3 kinase, which is actually important for the cell survival. Actually, it isn't. So if you look more closer into in the lab, you can see that the infected cells are very less in number, but they are very well positive for this PI3. So what is PI3? In immunology, the PI3 is basically a protein called elafin. Elafin is a very small protein which is just 6 kilodalton in size, and its function is basically to block in neutrophil elastase. This is the reason why I saw such a good protection. So this is all data in silico. So now let's go to the lab and get things working right there. And here we can clearly see upon infection, the human neutrophils, there's an increased amount of mRNA, which is expressing elafin, as well as the protein level is really high upon infection. And now we just checked if this protein is secreted, because it's known to be a secreted protein. And very well, upon infection, you can see all these infected samples upon time, there is a good secretion of this protein. At the same time, if it is secreted, it should also be blocking the neutrophil elastase activity. It's absolutely doing doing the job, it can block the neutrophil elastase. So you mean to say that recombinant elafin, if I put into the cells, can it increase the lifespan of neutrophils? The answer is yes. So you incubate the cells with recombinant elafin, it can actually increase the lifespan of your immune cells. So now we also block this the surface or secreted elafin using antibodies against it, and we could see that the neutrophils is no more able to support the infection. Rather, they go towards cell death and clears the infection. So now, how critical is it to do it? So you know that you cannot do any genetic manipulation on these immune cells. So we used HL60 cells and depleted the elafin level using lentivirus, and then we looked for infection right away. So without elafin, the bacteria cannot grow. And in mice, if you express elafin, you can see the infection is much better. So it very well depends upon the presence of elafin for its survival. So now came the important question, how is elafin regulated? So it's already known in the literature how it's regulated. So it was nothing out of the box. So it is regulated by CBP and CREB pathway. That's exactly what we see in these infected sample. We can see that the CBP is upregulated and CREB remains phosphorylated or activated. So we use the inhibitors of this to clearly show that the infection can be actually curtailed when you use any of these inhibitors, as you can see here too. The HSP60 level goes down and the elafin level also goes down. And if you use a second inhibitor, which inhibits CREB, you can also see the same effect. In the progeny, you can see that the infection population is completely depleted if you inhibit this pathway and deplete the elafin level. So how is CREB regulated? Everybody knows CREB is a cyclic AMP regulated protein. So it's very easy to think that way, right? But then there's a big twist in the story. We did not see any cyclic AMP getting regulated. So we tested in every different types of cells possible, but apparently chlamydia do not activate cyclic AMP. So then how exactly is this happening? So we looked a little bit into the literature, and we could see that beta arrestin, the G protein coupled receptors, are regulating protein, are when it is phosphorylated and regulated, they can basically activate this downstream pathway. And upon infection, what you see is that exactly we see that beta arrestin is highly upregulated upon infection. But then there was again a twist. We did not see the mRNA level. The mRNA level was kind of stable. So we knew that there is some kind of a post-translational modification that is happening to this protein. And exactly that is the case. So you pull down beta arrestin from the neutrophils, infected and uninfected, and you can see that the protein is completely deubiquitinated. So we were excited to find what is really deubiquitinating this beta arrestin. So deubiquitin, when it comes to chlamydia, they have two deubiquitinases, CLADAP1 and CLADAP2. These deubiquitinases are actually secreted and decorated onto the inclusion membrane. That's how they communicate with the cell. So what my student did is she just expressed DAP1 and DAP2 onto HeLa cells. And she could clearly show that beta arrestin is upregulated and deubiquitinated exactly, only very specific for DAP1. And now she created a mutant of this with just taking away the catalytic domain of CLADAP1, and she could see that the beta arrestin is no more regulated in this scenario. And now we also created we got as a gift this are cell bacteria from Duke University. So these are mutant bacteria which are deficient to CLADAPs, and you can clearly see they do not express DAPs. They do not regulate beta arrestin 2 too. So this is a very specific effect. So now we really challenged the neutrophils with all these bacteria. So mutant, the wild type bacteria, the mutant bacteria, we produced recombinant CLADAPs. And at the same time, we actually gave the mutant bacteria recombinant bacteria, so recomplemented it. So what you clearly see is that the mutant bacteria is absolutely not able to activate this whole signaling pathway. At the same time, if you see here, they are pushing the cell towards cell death and for the better clearance. Okay, we did not stop there. Since we need to go for a translational, potential for this project, we actually designed novel peptides which can basically inhibit the elafin. So how we tested is that neutrophil elastase has a known target that is HGF. So very much unrelated. So HGF is normally like that. When you treat with neutrophil elastase, it gets cleaved. That's very well known. And now what we did is we used elafin to protect it, and you can see a good protection. And now we use the peptide against elafin. You can see the the target is getting depleted. So that means it is blocking the elafin. And this peptide can also actually, here you can see, if you use the peptide together with the infection, you can see that the infection can actually, the cells will not block anymore cell death, and there is much more clearance of the bacteria. Okay, so what I wanted to show you so far is that the bacteria is able to hijack some specific cell types in the neutrophils, and what they use their a virulence factor called DUPS to actually stabilize beta arrestin 1, thereby activating TREP and CBP pathway to transcriptionally activate elafin. And elafin, once it is produced, it is also secreted very well. That can actually block the neutrophil elastase activity, not only of the cells which are infected, but also the bystander cells, creating a paralyzed situation in the infection scenario. So to the most important slide, I would say, to acknowledge, first of all, I would like to acknowledge our director, who's now not there, but he was really instrumental in all these research activities. And then again, the important collaborators, particularly Lynn and Fee and Valdivia and Robert, who actually supported us in very many capacities, and also Surya, who was a postdoc and designed this novel peptide, and Rajiv and Santosha, who to really support us in very different capacities. And particularly the lab members, all my PhD students and the group, and particularly Paridhi, because she did almost all the work, 90 to 95% of the work. She's now in the 4th year of her PhD, and we are soon submitting this paper. Thanks a lot to the blood donors as well as my facility, and thanks for all the fundings that I could gather with to get this paper working. Thanks a lot.
Thank you so much, Kartika. Maybe I'll start. Can you put together in a sort of, the inhibitor, that expression of the inhibitor, is that right?
Yes, sir.
Right. So will that, I mean, in terms of translation, will that affect, normal individuals, other neutrophils, because only some cells will be infected, right?
Yes, actually, only a very small population normally gets infected. And this infection is basically regulating the bystander cells. So when you put the inhibitor, it inhibits elafin as such and will lead to the death of the neutrophils. That's what we will see.
Yeah. But I was thinking in vivo context, I think it might have some dysregulation of neutrophils, this sort of strategy.
So we have actually could not check it in vivo. To study that, we need to go for that. But the unfortunate thing with this is that in mice do not have elafin. This ortholog is not present in mice. So what we have done is we have used lentiviral system to express the elafin in the uterine horns of the mice, and then we infected it. So only then we could see. But I believe that elafin is highly upregulated in the in when there is a cytokine storm in the lungs. At the same time, it is also upregulated in psoriasis and other skin diseases. So there, this peptide could be very efficient. Yeah.
Any questions? Yeah.
Yeah, thanks for the wonderful talk. So have you tested against other bacteria, some other gram-negative or positive
Yes,
bacteria?
sir. Very, very interesting question. Actually, we want to do this now, but I think because see, the transcription factor that is regulated is CBP beta, and it's highly upregulated by many bacteria. So they will regulate elaf, and this strategy might be one, for example, in MTB2. We are testing that. The sad part is this protein is very difficult to work with. It's only 6 kilodalton. So only very students, not all students can get this in bulks. So we have tough time with the training the students and getting them there.
So have you started to test the level of elastin samples? I mean,
Uh,
there is some correlation, high level of this elastin in the disease progression.
I'm sorry, I did not get
So
you.
you mentioned that elastin is secreted.
It's
So have you tested in the real samples when the
no,
patient have the infection,
no,
whether a higher level correlate to more severity and more
no.
level?
We could never get with human samples. Yeah.
Thank you.
Thank you, ma'am. Said mice is not express, mice doesn't express elapin.
Exactly.
So any other laboratory animal like a rat?
We have, I don't think any of this low forms of animals are expressing an ethanol of it, but we have not tested in guinea pigs or in yeah, yeah, we have not tested it. We do not know it. We need to test it because we were more focused to honestly to get this paper published. So we are now communicating.
Thank you.
There is there a yeah, go ahead.
Hello, ma'am. I'm a. I saw your quite wonderful talk.
Thank you.
So I just where you did to the single like where
There are a lot of companies which can do
in
it.
India only or
And MedGenome can do it. For example, here I did it with the collaborator. I went to Denmark to get this done, and they did the sequencing for me. But the analysis was done by both parties, right? By us and by them. Rajiv was basically from our side, and Fee from the other side. So now he is a professor in China. Yeah, but here you can do it, and very many companies are there who are coming forward.
because they have low RNA content. And we have
Yes, yes. Actually, they are. So when we were doing it, I did this in 2022 to 2021, 22 at the end of that. So that time, actually, the kits were not set for only poly-A-Ts. We did not have the multiplexing for the for the random exams, but you have that now. You also have a technology called Flex, where you can actually fix yourself and then do the sequencing, because then it is easy if you want to outsource it completely, but otherwise it's not really a tedious task to do it. So we have done it. It's not, if you have the machine in your system, in your facility, then you can do it, because we do have it now. We have the next genomics in RGCB, so you're welcome.
Okay, all right. So let us all thank full talk.
Thank you, sir.
Okay, we'll move to the next one. The next speaker is Dr. Gupta, who's an assistant professor of biochemistry, AIMS. Dr. Gupta's research focuses on immune dysregulation and sepsis and infectious diseases, emphasizing DC functions by RNA-mediated immune regulation. Dr. I'm really happy that all the speakers are keeping to time. I just will encourage all the students also to ask questions because it motivates the speakers.
Thank you for the introduction, sir. So first of all, I would like to thank you, organizer, especially Dr. Ambak, for giving me opportunity to present my ongoing research work that is funded by the SERV DST, where basically we look the expression of microRNA, specifically in the dendritic cell, due dendritic cell during the sepsis, and and their association with disease severity and outcome. So in 2017, WHO declared that sepsis is a global threat and about 11 million people died because of the sepsis, as well as that death include about 20% of the total hospital mortality is because of the sepsis, 20%. Now, the sepsis is caused by the various, there are so many causes which leads to the sepsis. So in our study that we are doing from the last 1.5, about 15 years, where we selected the trauma injury model, because as you know, trauma and injury represent the sterile inflammation, and they are more susceptible to develop the sepsis complication in the ICU in the hospital. So what kind of the immune response is there if there is shift in the sterile inflammation to the pathogen-induced inflammatory condition? So, in the trauma, basically multiple organ failure is the major cause of the death, and it is really challenging to differentiate the patients who having the sepsis, means pathogen, and other is sterile inflammation. Here you can see if there is an injury, road traffic injury, if about 20 to 30% patients died immediately. And nowadays you can see government even in the Uttar Pradesh, no helmet, no petrol. Why it is so? To prevent the early injury, death because of the early trauma, road traffic injury. And after that, if patients survive, later they develop the sepsis complication and die. So our focus is to look about the why patients are developing the sepsis complication. As you know, nowadays we have good tertiary care hospitals, and despite of that, there is a huge number of the mortalities in the hospitals. So inflammatory response, as you know, if there is any injury, either trauma, either burn, that leads to the inflammatory response. Pro-inflammatory and anti-inflammatory, as we know. So there is a should be balance in these two: early inflammatory response and as well as late inflammatory response. If there is a disbalance in these two, that leads to the multiple organ failure and death. And in this, as I told you, we are working from the many years. We have observed in the AIMS New Delhi, in the trauma patients, patients who having the same level of the injury, same body part injury, few patients are developing the sepsis complication and others are not. So we perform the cytokine gene polymorphism, measure the multiple cytokines in the patient sample, traumatic patient at different time point, and correlate with their outcome. Also, we measure the T helper cell subsets, Th1, Th2, T regulatory T cells, and Th17. And this work has been published in the good journal. And we observe if there is a shift in the Th1 to Th2, means pro to anti-inflammatory, inflammatory, there is a more if anti-inflammatory response, patients are primarily developing the sepsis complication, and also we have observed the mortality among those patients. And recent study that we have measured the monocyte subsets again in these patients, those patients who developed the sepsis and other who did not. And observe monocytes, there is a three subset: classical, patrolling, as well as inflammatory. Patrolling monocytes is more during the sepsis condition. And then we find out monocyte derived DC who are basically having the nature of the classical, either the patrolling. So in this study, we have proposed to measure the function of dendritic cell. One, find out the expression of specific microRNA, selected microRNA based on the RNA-seq data. I will show you the sequence of the RNA. And then we have measured their what is their changes and association with the disease severity and outcome. These are the basically inclusion criteria: traumatic patient within 8 hours of the injury, and those patients who have already traumatic brain injury as well as other kind of inflammatory condition, cancer and disease, we exclude them. Study design include: we recruited the patient within 8 hours of the injury, those who have been severe injury, collected the blood sample day 1, day 7, and day 14, and outcome up to the measure 28 days, means longitudinal study design. And we are measured the whether they develop the sepsis complication or not. One, whether they survived or not. If they have developed the sepsis complication, what kind of the changes in the dendritic cell level, their function, microRNA level, and whatever we have observed so far. So we want to correlate all of them in a study. So here total 21 patients we took for the total 62% we recruited, and out of them only 46 patients who finally we took in the uh, analysis, because it is really difficult to get the all three sample and follow up patient for the 28 days, because sepsis patient they succumb, they died in between. So 21 patients we post-traumatic and non-sepsis, and 25 we got the sepsis patient. And you can see SOFA score. SOFA is basically sequential organ failure. It's used to measure the extent of organ failure. As I told you, so multiple organ failure is the major cause of death in the sepsis. So those patients who develop this sepsis, you can see here SOFA score is more, and that is the clinician is used to diagnose the patients basically by the SOFA score and CRP and PCT. We don't have any specific marker for the sepsis, despite of having the huge mortality, as I told you about the 20%. So this one is the flow representative data. We took the blood, 2 ml blood sample, lyse the RBC, gated on the CD45, are leukocytes, and lineages. Lineages means B cell, T cell, NK cell, we exclude them and took just HLA-DR positive. That is called CD45 positive, HLA-DR positive, and lineages negative cells are called CD and dendritic cells that we measure. And we also measure the activation marker on the dendritic cell, that is CD80 and CD86. Here you can see the during the sepsis at different time point, you can see the level of dendritic cells are going to be reduced, whether sepsis condition or non-sepsis condition, but significantly reduced in the sepsis. Then we measure the activation marker, as I told you, CD80 and CD86, that is also reduced during the sepsis condition. So number of the disease is reduced, also their activation is going to reduce during the sepsis condition. Next, as I told you, we other blood sample we isolated the PBMC and sorted the dendritic cell by the magnetic sorting, and confirm their sorting means dendritic cell, as you know, is less than 1%. You need a huge amount of, means a minimum 10 ml of the blood sample. Then only you either you have to pull the sample. And then dendritic cells, what we did, just we confirm by the flow cyte their purity by the fluorescent microscope, isolated the RNA and measure their expression by the qPCR. So these are the simple protocol you can find out in the stem cell. This is how to enrich that. So as I told you, total 12 microRNA we have selected. 12 microRNA, those who are basically based on the RNA-seq data, those who are involved in the dendritic cell maturation, their function and activation. Two housekeeping genes as well as one spiking control. So what we did, first we took the PBMC as in the healthy sample as well as trauma patient, means inflammation and healthy individual, and measure the whole change of the whole change expression of this microRNA. We have observed only 3 microRNA is more expressed in the inflammatory condition, means trauma. Again in the PBMC, we measure the trauma versus sepsis. Those who develop the sepsis complication, what kind of the fold change in microRNA? We have observed total 6 microRNA are going to be changed during the sepsis condition. Next, what we did, as I told you, so among the in the sorted dendritic cell, we have measured the expression of the microRNA and compared with the in both healthy and trauma condition as well as in sepsis condition. We have observed a total. You can see here as same microRNA, whatever their expression is less in the PBMC, their expression is many-fold high in the dendritic cells. So these microRNA, whatever we have selected, they having the more important role in the dendritic cell function and also in the may have in the cytokine secretion, that we have to be check in the next level. So here you can see as compared to PBMC, the expression of microRNA is more in the dendritic cells, and you see microRNA 146 to 21 were significantly down regulated, whereas there are 6 microRNA here, 42, 55, 21 and 222, and let's 7, they are upregulated. Now whether these are change, what is meaning of these upregulated, downregulated, whether they have any correlation with disease severity or not. So we have observed those patients who died because of the sepsis, means they have sepsis, and also those who died, they having the high, you can see about 4-fold elevated level of 34A, 140, 42 as well as 155. They having the more level in the death, those patients who died. So conclusion is the patients who developed the sepsis and died because of the sepsis, they having the high expression of these 3 microRNA, 34, 142 and 55, and they have also association with the SOFI score. I have not shown that data. SOFI score means, as I told, sequential organ failure assessments, means they have developed the sepsis, also developed the multiple organ failure, that is called septic shock condition, and they died. And here activation marker CD80 and CD86 is also reduced. I have not shown the data of the uh, cytokines, that is still we are measuring. Whatever we did this isolated the PBMC as well as also dendritic cells, did the co-culture experiment and measure their cytokines. So as I told you, these are the preliminary data and this is the ongoing study. So once we will compile, might be having the good outcome about the which microRNA, if there is down regulated, either up regulated, which cytokine is going to be changed, because this regulation in the inflammatory condition and sepsis of the cytokines is very important, and that is the deciding whether patient is going for the septic shock or not. And and other thing is, as I told you, there as present we don't have any specific markers, so molecular markers is very important to diagnose this sepsis in the ICU setting, because clinically only clinicians are looking the clinical features, and clinical features appears once patients are in the critical condition, either they have severity. So early marker is very important, and nowadays, as you know, we have a lot of the advancement in the technology, so molecular markers are very important. So I would like to acknowledge, of course, funding agency, science, serve DST, ICMR, that they have given fund to complete our previous work. And students, yes, we cannot do any work without the students. So I would like to acknowledge the students and faculty member. And my mentor is here, Dr. Dina Rao sir. He is used to still guiding me to write this kind of the project, do that work. And always he is actively, now at the age of even 60, sir still is guiding. Okay, thank you, sir. And I would like to take the questions from the students.
Okay. Thank you, Doug. Yeah. Any questions? Can-
Yes.
Just wait. Wait.
Not a question, just as you see that this is a deduction of CD80 and 86 are there. So you can see the target seed sequence is there or not of this microRNA. They are directly targeting this CDs or not there.
Okay.
So microRNAs, how this microRNAs linked with this CD80 and 86 down regulation. So whether they can be directly targeting to the CD theta UTR region of the CD80 and 86, and they can downregulate this one. So you can get the direct target or not. So if they can direct target, if they have the region, then you can see the direct target of this CD80. If not, then indirectly might be.
Yes.
So then there's a connection that how this microRNA is upregulated, regulating the downregulation of the genes. Thank you.
Yes. Yes. Basically, what happened at the corner, and as they are easily produced, and their production is entirely dependent on signaling. Okay. So link signaling with the function, that is the until unless we don't exactly how they are changed cytokine. First thing, until unless we know the cytokine first, then we have to go for the marker, activation marker, and for that we go for the knockout expression. Otherwise, we can't confirm it.
Any other? So if there are no other queries, let's thank you very much, W, for a very nice talk.
Thank
The next speaker, Monisha Dhim, is a professor in Department of Microbiology, Basic Sciences, Central University of Punjab. She specializes in oxidative stress biology, immunology, and cancer research. Dr. Dhima.
So, good afternoon, everyone. Just a session just before lunch is bad, but yes, I would like to share a few of our work which we are doing in our lab.
No, after lunch is even worse.
That's worse.
So people are looking forward to your talk. After lunch is even worse. So
Okay, thank you. Thank you, organizers, for inviting me so that I can share my work, which we are doing. I am basically working on H. pylori and how it's a notorious bacteria. Most of us know about it. And this is, it's a gram, and most of the time it is a gram-negative bacteria, which grows in microaerophilic conditions, and even growing it in lab conditions is also very difficult. It was most of us we know that it was Nobel laureates Warren and Marshall. They introduced it. They actually they were talking about, people were talking about gastric ulcers. Nobody believed that bacteria like this can cause such gastric ulcers. So their work has contributed very significantly to propose that yes, H. pylori is the one notorious bacteria which causes those gastric ulcers, and that may
lead to gastric cancers. And the important thing about this bacteria is that most of 1/3 of the population has it, but we don't know it unless until it goes to a gastric cancer stage and then gets identified. So that is a bad thing, where researchers are working on it, that most of the population has it, but some develops the gastric ulcers, and that goes to a second level of cancer, gastric cancer. So identifying the gap between how that goes is very crucial. Some people develop it, some no don't. So that is something very interesting to understand, and that is what some contribution our lab is doing on it. Epidemiology of H. pylori is such that a lot of countries where it is more than 80 to 90% also, and so there is for some in Asia and Russia, there we have a good number of H. pylori population having a good amount of H. pylori infection been reported. So, considering this, it was when H. pylori was associated with gastric ulcer. Later on, it was seen that a lot of other diseases are also there which are associated with H. pylori. Chronic gastritis, peptic ulcers, gastric cancer, and gastric cancer is fifth most common cancer, and that is also some of people who are working in gastric cancer. There is a predisposition of H. pylori infection has been reported there, and then malt lymphoma. An international agency for research on cancer has classified H. pylori as class group 1 carcinogen in 1994, and still we are struggling at what the pathogenesis of this bacteria is, how those gastric ulcers, why that is, because and in 2021 it was identified as a human carcinogen by because it is one of the most crucially reported carcinogen in gastric cancer patients. So why this bacteria has so much of leverage, and how it can survive, and that too in gastrum, where we say that pH is so low and it has so much of good thing. We say that most of our stomach is the first place where if we have any infection that goes away. So this bacteria is very smart. It attaches to the host cell, moves towards the gastric epithelium through its flagellar motility, its structural structure, as well as the virulence factors it produces. A lot of things are there which helps this bacteria to grow in the gastric epithelium, which is otherwise very critical, where the pH is 1 to 2. This has survival is possible because it produces ureases, and that neutralizes the acidic pH of the epithelium, and produces ammonia and survives very happily there. And in addition to that, it has a lot of virulence factors which it keeps on producing. I'll just not go to this figure, I'll just show you this. So why this H. pylori pathogen is important, because it has helical shape, flagella is there. There are many adherent factors which help the bacteria to enter. In addition to that, the toxins which are produced, VAC is one, my lab is working on that also. And there are some effector toxins which are very important, which help this bacteria to grow better. And that leads to that pathogenesis, what we see in severe gastric or peptic ulcers. So we are working on this a NAP protein. Today I'm talking about this NAP protein, which is neutrophil activating protein. It, as the name suggests, is activates the neutrophils. But in addition to that, there are many things which NAP protein doesn't taken much of focus when we talk about its virulence factors. Most of the VAC-A and CAG-A are the two virulence factors which have been worked. A lot of work has been reported on these two virulence factors, but its NAP is a little unreported. So our lab, we are trying to focus on this NAP. This NAP is a very interesting protein. It is a ferritin-like protein, which is about 150 kilodalton, and this is something very important because it is very similar to bacterioferritin. So very similar to what we see, because once we see that, why we thought of working on this, because when we see this is a protein which has ferroxin as a center, and it also has a tendency to binds up to 500 iron. So imagine if a H. pylori infection, when we say that there are peptic ulcers, and then later on it is reported that people go to, they get anemic. So there are some pathogenesis has to be very important, where role of iron would be very crucial. So that's why we thought that let us see how this iron taking capacity of this nap versus later on, because that also leads to ferroptosis, which is in presence of iron. If iron is available, how the cell damage happens. So that is what we thought, that this is a good protein. We started working on this protein, and this is some of the, because when we say that it is a neutrophil activating protein, definitely we have to work on how reactive oxygen species work there, because neutrophil, ultimately that is what the purpose of neutrophil. Whenever there is any infection, and when the inflammation increases, then neutrophil plays, and our reactive oxygen species and respiratory burst plays very important role there. So my lab also is also working on various in addition to nap, where ROS is important. That part also we are working on. So this is just a presentation of. So what is the role of NAP in programmed cell death? Because it is reported that H. pylori is so smart that it inhibits the programmed cell death. It inhibits apoptosis. So it was very important to understand that if iron is present, apoptosis is not happening, maybe ferroptosis is happening. So we wanted to see that is there any mechanism other than apoptosis which is happening in that milieu when infection is there. So we wanted to see HPNAP, low role of HPNAP in program cell death, which otherwise in general is said that is not there. So, and second, we wanted to know that how the HPNAP has some role. If there's any correlation between apoptosis and ferroptosis, which is one important thing to understand, because iron plays very important role in HPNAP. And so that's why we worked on two cell lines. One is, why we took two cell lines, because these are A549, which is a lung epithelial cell. We wanted to see one gastric cell line and one epithelial, which is not part of gastrum, rather than it's a lung cancer cell. So we used these two cell lines, and we started working on this. We had this recombinant protein of HPNAP, which we purified, and then we saw how this HPNAP induces, how what is the effect of HPNAP. First, we worked on A549 cell lines, which are lung epithelial cell lines. So if you see that with HPNAP, it was actually killing, cells were getting killed, and ROS is also getting increased in the second image. If you see, so ROS dependent killing was happening in A549 cells. Similarly, we also confirmed that what is happening to apoptosis, and we could see that in that A549 cells, the HPNAP was introducing apoptosis in A549 cells. So this is important to understand that this is not a gastric epithelium, where the purpose of H. pylori is different. So it's a lung cancer cell line where the apoptosis was happening. Similarly, we did some transcription level of BAX and BCL, and there also it was apoptosis was getting induced. The ratio of BAX and BCL was seen there, and then we did a caspase. So this all pro-apoptotic potential of HPNAP could be seen in A549 cells. So this was first part, but then we thought that what is happening, and then we also did some inflammation marker in A549 cells, where NOS and TNF alpha was significantly getting increased when their cells were treated with HPNAP. So this was the part where A549 cells, this is a small project which my master student did. So then I thought that, okay, actual gastric cells, what is happening? So we started working on AGS cells, which are gastric cell line, adenocarcinoma gastric. So in HPNAP, it is very interesting. I would like to share that when HPNAP was alone, just alone HPNAP was treated, the cells were not getting any, there was no change in the cell lines. They were not getting, but as soon as we started giving them H2O2 or CONA, which are reactive oxygen species source of H2O2, means we are providing ROS and CONA. In presence of H2O2 and CONA, the NAP was behaving very differently, and this experiment was done very, I asked my student to repeat it so many times, that is very interesting. So that means that in a pre-deposition condition where ROS is there or inflammation is already there, those virulence factors of HP will work more. They will induce more inflammation, or they will be more damaging than when alone. Some conditions are when ROS is not there, or when there is no inflammation there. And similarly, we did and it was confirmed. We confirmed that with NAP and then Kone. This is the same experiment. Even in the same case, the ROS level was getting increased, not when an HP NAP was alone, NAP was given alone, rather when it was given in uh, uh, before the NAP, H2O2 was given, and before NAP, Kone was given, and then NAP treatment was given. That time the ROS level was getting increased. So this was something very important that finally we thought that let us see what is happening to ferroptosis and apoptosis, because role of ROS and um, iron dependent ROS is very important. So we tried to explore these two pathways, which is ferroptosis and apoptosis. We have used various inhibitors and inducers, apoptosis inhibitor, inducer, and free radical scavenger, which is NAC, and then general ROS, which is NADPH oxidase inhibitor, which is DPI, myeloperoxidase inhibitor, which is SHS, salicyl hydroxamide. So we thought that let us see what happens to cell when we are giving these inhibitors. And in presence of ferroptosis inhibitors and inducers, it was very important to understand that when NAP was given to the cells, there was no significant change was happening in ferroptosis inhibitor. Whereas when inducer was given, ferroptosis in presence of ferroptosis inducer, the cell death was even more higher. Whereas in case of apoptosis, in presence of apoptosis also, the apoptosis inducer was causing a cell death. In this case also, we thought that what is happening to ROS. So now the overall the ROS was also exactly the same what we saw in cell toxicity or MTT based assay or cell death. Similarly, in presence of ferroptosis inducer, the ROS levels were very, very high. So if you see all this data, there is a correlation which we want to try. We want to explore that how these ferroptosis and apoptosis, which generally goes is ferroptosis is not reported in H. pylori as such. So we really want to explore that how this HP NAP, which is a ferritin-like protein or bacterioferritin, how this is playing a role. This is just one part of work which my student was doing. So I have just, and similarly we did some work with iron chelator, and even in iron, in presence of iron chelator, the NAP induced ROS was decreased significantly. So role of ROS and role of iron in NAP induced toxicity was significantly, I mean, it is almost known now, and now we are trying to see how the ferroptosis, we are trying to this part of the study talks about the apoptosis being induced in A5 which is liver, lung cancer cell line, but the role of NAP in gastric cancer cell line is, or gastrum, is very important. And role of iron chelators, they were seen it is definitely the HP NAP, which is a one protein, a bacterioferritin like protein, has a very important potential which can induce a apoptotic like condition, which we say H. pylori doesn't induce that. This is what our work is, we are going on, and the ongoing work is there, which we are continuing. So this work is supported by DBT Bionet project, and my MSc student and a student here has done this work. So
Thank you. Thank you. Are there any questions for Monisha? Yeah.
Sir, I use
I have used H2DCFTA.
DCF for specific.
No, not if we go for mitochondria, then we use socks and everything. This is a general ROS where I have used H2DCFTA.
There's one more student.
Question. Do we already know the mechanism how we activating the is this benefiting the bacteria? Because when neutrophils can act clear the infection. So how
So the part is, as the name says, is a neutrophil activating protein. But how uh, its role in that when infection happens, that is not been explored. You know, its role when infection is there, but how what will make it worst? That is what the problem is. Answers are there, but what is the thing which takes it to a next level when it becomes a cancer? You know, so that is what that thing. If we can understand that, that is what we are trying to see. That iron dependent component.
Yeah, go ahead. Go ahead.
Actually, as we know, neutrophils are both pro-tumorigenic and anti-tumorigenic. Yes. And in context of this H. pylori, when it is leading to gastric cancer, how neutrophils play that can be used as a therapy.
Yes, this is one of the project. Just PhD scholar has joined, and this is one of the continuation of what NAP we are doing. We're trying. He has just started his work. He has just started. He's doing some in silico modeling. He's trying to see that how these factors and neutrophils they play a role. So it is just we are just thinking that okay, because even neutrophil plasticity is important. You know, various types of that would be important there too. So we are trying to see that. That is our next project. Where we
Okay. So let us thank Dr. Monisha for a very nice talk.
thank you, everyone.
It turns out that her talk is not the last one. We may have is Dr. Gupta here? Yeah. So Dr. Pravesh Gupta is an assistant professor at Mayo Clinic, where he leads pioneering research on developing next generation tumor agnostic universal immunotherapies by optimizing phagocytosis circuits. His work integrates single cell, spatial, and functional proteomics to uncover brain specific anti tumor mechanisms. Dr. Gupta. So yours is the last talk. So I think you need to do something. There's a little bit of lag, but it comes. Ah, good. Go ahead. Go ahead, doctor.
Justice. Okay. Uh, just a little anecdote. This is my second Immunocon. In the first one, I bunked my internal university exams to enjoy immunology at Immunocon 10 years back. So today I've been invited as a speaker. That's great. And I'm going to narrate a journey of last 6 to 7 years where I've been pursuing multi-omics immune microenvironment interrogation of human brain malignancies. And I'm going to try to convince you that we found novel anti-tumor phagocytic modulators. Uh, this is the unmet clinical need that we are trying to address. So about 200K patients are affected by glioma, adult glioma, pediatric, and brain metastasis in the United States. And this is the standard of care where surgery, radiation, and chemotherapy, forms the standard of care. And immunotherapy is the emerging complementary modality which is being investigated thoroughly to disrupt the brain immune microenvironment. um, just a little background. Uh, Lymphatics is the highway for immune cell trafficking. It was in 18th century that Paolo Mascagni made the drawings for lymphatics in human brain. But it took almost 2 centuries, to confirm these findings by Jonathan Kipnis' lab, where structure and functional features of CNS lymphatics was defined in this Nature paper. Uh, Brain, as we know, is immune privileged, but I guess this dogma has been shifting because this is now considered as selectively permissive for a lot of immune cells, which I'm going to discuss in the results that we have. Um, and this is dominated by microglia and tumor-associated macrophages, which are largely characterized to support tumor growth. Uh, Without even understanding what are the cellular complexity in human brain, uh, T cell checkpoint blockade therapies in GBM was generalized just by the sheer motivation that it worked in other malignancies. And the results have been really dismal. It was not until 2019 and 2020 the first fragmented cellular complexity of myeloid cells was presented in these two Nature papers, Nature Neuroscience papers, but this gave a pretty good idea of what the complexity looks like. Uh, Brain tumors are generally characterized by low tumor mutation burden and neoantigens, and T cells are scarce. Yet, CAR T cells are being prioritized by different studies, as I show here, targeting uh, many antigens which are either tumor specific or tumor associated. And most of these studies, they have been partial responses, um, and that's what we are trying to understand. How do we uh, you know move the needle in the field? So imagine, brain is a very highly regulated organ, with respect to immunity. What we are trying to do here is put fuel to the fire which is already inflamed. So brain tumor is highly inflamed, and what we are trying to do is put CAR T, which gets shuts off because of the unique regulation which is existing in the microenvironment. So we had this, simple question: what is the cellular complexity which comprises the, immune cells in the brain tumor microenvironment? And we chose, certain glioma subtypes, which I'm going to describe in the next slide. And we had a simple hypothesis: do we have immune cells which can fight the gliomas? So it appears that, this is a published paper where we show that brain immune microenvironment is myeloid cell rich. So is the case not only in adult tumors, but also in pediatric tumors. And the microenvironment is myeloid heavy. Okay. So we are trying to think if we can leverage these dominant cells and create circuits that could be used as next generation immunotherapies. So to understand this, we developed a reverse translational brain tumor immunology program at MD Anderson Cancer Center, which was funded by the Moonshots program. We assembled a suite of technology pipelines, both single cell, genomics, proteomics, and spatial, just to understand the cellular and molecular complexity in different brain malignancies, and reverse translate those findings in mouse models which mimic glioma pathology. Uh, what we did is we used primary and relapse IDH mutant and wild type gliomas. Mutants are the long-term survivors, and wild type is the short-term survivor patients. We isolated the CD45 immune population, which appears is, we have both CD45 high and low, which until this study, people were not able to figure out. And what we did is, using the discovery cohort, we did a single cell RNA-seq and performed a validation of those findings through spectral cytometry. So, we could confirm whether those cells exist or not. I think this is the methodology I'll skip. So we saw, I already showed this slide, the myeloid cells are enriched in human gliomas, regardless of whatever is the mutation or whether they are long-term survivors or not. This is the complete atlas. We have 22 different immune cell types in human brains. So I don't know whether it's immune privileged or it's immune reactive. This is for the field to decide. But it appears the dominant population is myeloid cells, which comprises microglia and the monocytic derivatives of both macrophages and DC origins. Uh, um, and what we notice in particular is microglia, they reduce in numbers during relapse, regardless of the IDH mutation status, as you can see in the data here. Here. And what we also noticed is T cells, they increased in numbers. Okay. So I'm not sure what T cell is doing. And also when we look at the ISC slides, T cells are very limited in numbers and they hardly interact with the antigen presenting cells. This has been our experience. Um, okay, why this is not moving. I'm sorry. Okay, yeah. So using this data, what we wanted to clarify few dogmas in the field, this obsolete characterization of M1, M1 and M2 macrophages. Rather, we found that there's a spectral phenotype that we observe. M1 can coexist with metabolic modules. M1 can exist alone, or M2 can exist with metabolic phenotypes, and several other modules. So we provide a guideline that is just not the M1 and M2, rather it's a spectral polarization model of macrophages which the field should adopt in order to better understand the functions. And it was heartening to see that a follow-up study from this guidelines was published, in a xenoline model where they validated some of these phenotypes presented here. And then there's another, guideline we wanted to give to the community is LM22 is the bulk genomics signatures that people use for deconvoluting oncology data sets at the bulk transcriptomics level. But there was no brain-specific reference. We created this brain-specific reference of gliotime where uh, different flavors of immune cells, it just saturated at 36 different functional flavors across myeloid and lymphoid cell types. And we applied this data set on a glass cohort which mimicked our discovery cohort of primary and relapse IDH mutation and wild type. And what we saw, if you apply LM22, if you use a wrong reference, you get the wrong picture. Whereas I would say rather incomplete picture, rather, so if you use the right reference, you will get a rather complete picture. Uh, and this was really interesting for us. Amongst the inflammatory microglia, which is quite heterogeneous, we identified granulysin expressing inflammatory microglia, which is really canonical only for T cells. This molecule is a peptide which is expressed mostly by the T cells and the NK cells. Rather, we were surprised why granulysin is being expressed under inflamed conditions in microglia in malignancies. This was just the genomics data. We wanted to confirm whether this really holds true at protein level. So we did a multispectral codex assay, and we found that TREM2 expressing microglia, they co-express granulysin. As you can see, also in another technology which we deployed is spectral cytometry. So all these orthogonal technologies just confirm the existence of this inflammatory microglia which co-express granulysin and TREM2. And we were just curious to know what does it do. So here we saw that granulysin expression correlates with phagocytosis. We did a 40-plex spectral cytometry investigation of different types of microglia with the TREM2 and GNLY co-expression. And it appears that GNLY seems to be a sole driver for phagocytosis score, as you can see here. So, moving to the next story, which is STEM2, what we also looked at is STEM2. What does STEM2 do? It appears that TREM2 also regulates phagocytosis in glioblastoma. And we confirmed this finding through an in vivo study where we labeled the CT2A, which is a glioma model, with GS screen a reporter. And when we sorted the cells, what we saw is TREM2 sorted cells. They phagocytose the glioma cells, regardless of where it is expressed, whether if it's expressed on DC, microglia, macrophages, TREM2 expression governs phagocytosis in glioblastoma. Okay. Yeah. So yeah, so that's the TREM2, as another immunomodulator that we identified. So we pursued, unbiased screen of ligand receptor interactome analysis to understand, are there any other, immunomodulators that would, suggest to any alternative functions. We identified galectin-9 as a signaling hub in gliomas, across, IDH wild type and mutant, and we confirmed this with protein level expression that is present in microglia and macrophages and not in tumor cells. And yet again, we found another molecule that galectin-9 expression correlates with enrichment of, cell addition and phagocytosis pathway. So what we have been constantly discovering is molecular modulators that can modulate phagocytosis. Um, we did a patient-derived co-culture assay just to understand if this was true from the single cell interactome study that we observed. Uh, and here again, what we found is galectin-9, whether downregulated at gene or protein level, impairs microglial tumor cell addition, and which is quantified here. So, I hope I have convinced you that we have found multiple immunomodulators as opposed to what people think is tumor-associated macrophages just promote tumor. and we have provided evidence that this can, engulf glioma cells. And here we provide a case for myeloid cell therapy. What we did is we look at the patient samples and we found a unique population of myeloid cell derivatives, which can be used as therapeutics. And this is in the brain sample from the surgery room. And what we did is we can't extract these cells from the brain, so we were looking for blood correlates of these myeloid cell derivatives. And when we transplanted these myeloid cell derivatives from glioma patient bloods in an unengineered format, uh, and adoptive cell transfer was done, and we found there was a potential of these cells to cure mice at 120 days. So what are we trying to aim here? We are trying to create next generation immunotherapies based on myeloid cells by, optimizing phagocytic circuits, which we have identified, and also TME modifiers. Hopefully, in next 2 years, we would be able to, provide the proof of concept that MCD-like cells, when engineered with these phagocytic circuits that we have identified and, TME modifiers, will provide the first-in-class, off-the-shelf allogenic cellular vaccine for not only brain tumors, but also other solid tumors where this mechanisms exist. So this has been a story for past 5 years where we have developed this, brain tumor discovery platform, and we have identified the cellular and molecular information. We have decoded this, and we provide this cell atlas for brain tumors, which can be now used as guideline. We have optimized the phagocytic modulators, which we are engineering right now as allogenic cell therapies. And hopefully in next 2 years, we'll have some data. I would like to thank, Dr. Bhatt, who is my mentor, and the members who contributed to this study, Shivangi and Mackenzie, and also Silvana from UTMB, and the clinical collaborators, and the most importantly, the patient donors, without which this study would have not been possible. Thank you for your time and attention, and I would like to take any questions if you have.
Thank you, Dr. Gupta. Are there any questions? Yeah, please go. Just 1 minute. Wait for the mic.
So, very nice presentation. Question is about the myeloid derived cells. As you know, nowadays, basically immune suppressive environment is created by the myeloid derived suppressor cell in the tumor microenvironment. How this MDSC derived cells is differ from the other tumor MDSC? Is
Yeah.
there, have you characterized this one?
So yeah, we have deeply characterized what these MCD-like cells are. And for the MDSC, which seems to be the paradigm in tumor biology, we don't believe there are any MDSCs in situ. This is just based on 1 or 2 markers. People have been characterizing it. But let's say if you took those cells directly from the patients and you co-cultured it with the stem cells, tumor stem cells, or let's say glioma stem cells, I don't know if there is any evidence like that. So most of the times people do this in cell culture models or mouse models. You know, by large, this study is more patient driven, where we are trying to discover um, new concepts and use patient samples to confirm those. So in vitro and in vivo does not recapitulate, I would say, even in the case of granulysin, this was missed largely because this molecule is not expressed in mouse. And if I didn't do this study from patient samples, I wouldn't have discovered this. So is the case for MDSCs. I think there needs to be systematic interrogation on MDSCs from different tumor, models from patients, and see it for ourselves whether it does suppress the tumor.
Okay. We will discuss later.
Yeah.
Yeah. So, if there are no other questions, I'd like to thank Dr. Gupta for his very elegant presentation. Thank you so much for making it on time so that you could give the talk. Thank you so much.
Thanks.
Let us thank all the speakers for their talks. I really appreciate the fact that all the speakers really kept to time, and I didn't have to berate them. And thanks to the audience also for asking questions. Overall, I think it was very useful. We should now be ready for lunch. I'll hand it over to the organizers.
We extend our heartfelt thanks to our respected chairperson, Professor Deepankar Nandi, for their valuable time, insightful comments, and for so gracefully conducting this session. Your guidance and encouragement have truly enriched our discussions today. Now I request our chairperson to kindly come forward and present memento to our distinguished speakers. All the speakers of this sessions are requested to please come upon the dais. I warmly invite Dr. Pravesh Gupta, Dr. V. Arvindan, Dr. S. Jyoti Prasanna, Dr. Kartika Rajiv, Dr. W. Lal Gupta, and Professor Manisha Dhiman. And before you all depart, please stay with us on the stage as we will proceed with the group photograph. Thank you, professors. Now I request Dr. Samir Srivastav to kindly present token of appreciation to our respected chairperson as a gesture of gratitude and respect. With that, we formally conclude session 3 of Parallel Talks, and we sincerely thank all our speakers, chairpersons, and delegates for their active participations and contributions. We will now take a break for lunch, which will be served at the designated dining area, and we will resume the discussions at 3 p.m. And the next round of parallel sessions will begin at 3 p.m. So we request all participants to res- kindly return to their respective venues. Thank you, everyone. Enjoy your lunch. I request all the chairpersons, speakers, and the participants to please reach the MP hall as we are going to start in 2 minutes. Good afternoon, everyone. We hope you all had a refreshing lunch and an engaging time at the poster session. Let us now move forward with our afternoon panel session titled Immunotherapy. This session will explore one of the most transformative and rapidly advancing areas of modern immunology, the science of immunotherapy, its mechanism, and its growing impact on the diagnosis and the treatment of immune related diseases and cancers. As we now commence our session, it gives me a great pleasure to invite our distinguished chairperson to kindly come forward and take their seats on the dais to lead this session. We are honored to welcome Dr. Shrikant Sadhu, research scientist at the Translational Health Science and Technology, Faridabad. Dr. Sadhu holds MSc in Animal Biotechnology from the University of Hyderabad and a PhD in Infection Biology and Immunology. His research span host pathogen interactions, immune responses in viral bacterial infection such as salmonella, and the role of Th17 and IL-9 producing T helper cells in inflammation. He also investigates how diet and gut microbiota shape immune regulation. Dr. Sadhu has published extensively in high impact journals, holds patent in immune modulatory therapeutics, and is a life member of the Indian Immunology Society. He has also been honored with the prestigious recognition, including the G.P. Talwar Young Scientist Award and the IUIS Rising Star Award. With our esteemed chairperson on the dais, I would like to request them to kindly take the charge of the session and invite the speakers with their respective talks.
So I think my co-chair, Professor Javed Agarwala, is not here. So we will start with the session. I kindly invite Dr. Joginder Tushreshi. He is an associate professor at US Davis and investigator at the Comprehensive Cancer Center. And he has former at University of Virginia and pharmaceutical industry. He was awarded the U.S. Department of Defense Breakthrough Award and the U.S. Department of Defense Ovarian Cancer Early Career Investigation Award. So, Dr. Joginder Tushreshi, please.
Thank you all for coming and present some of the work that we have been doing. Today, also how you can learn from our ancestors. And if you think about it, our ancestors was our parents or grandparents, but I think in terms of evolution, our ancestors are primates. Oh, oh, sorry. So should I start again? So thanks all for coming. So today I'll present some of the work that we have been doing at UC Davis Comprehensive Cancer Center. And I'm really thankful to the chairperson and organizers for giving me this opportunity to present some of our work. So today I'll talk about learning from our ancestors. So most of the time, like as a kid, they say learn from your parents, learn from your grandparents. But in terms of evolution, I think we need to learn from primates. Those were our ancestors. And I'll give you an example that they have developed the ways to fight cancer and infectious diseases. And I'll present one specific example since it's a 15-minute talk. So every time anybody is diagnosed with cancer, they go to clinic. What are the treatment options? The doctors will cut them open, and we have a good name for it. It's called surgery. And they'll poison them with a lot of drugs. They lose hair, they become sick, and there is a good name for it. It's chemotherapy. And if they still don't work, they'll burn the patient, and which is again a great name, radiotherapy. Believe it or not, it's been happening since last 15 years, 50 years. This is still the standard treatment of care for a lot of cancer. Thankfully, we have new treatment around the corner, and these are basically cancer immunotherapy, cellular therapies, and oncolytic viruses. So my lab work on two of these aspects, and I will give you a very unique story today, which is just accepted just 2 days ago, and I'll show you the data. So for the students, few of them whoever here is, what is immunotherapy? Instead of relying on toxic drugs, harmful radiation, you basically empower patient own immune system to fight the cancer. And for that, you have immune effector cells. So I'm sure this slide everybody knows. The field has revolutionized as soon as there was a discovery of immune checkpoint inhibitors. And you can see there are activating receptor, inhibitory receptors, and there is a lot of success of immune checkpoint, PD-1, PD-L1, CTLA, and there is a growing list of it. So in today's talk, I will focus on this TIGIT molecule. All right, so TIGIT it's an inhibitory activating pair between CD26 and TIGIT, and it's a big target for last decade. And I'll tell you, everybody was studying it very wrong. And we'll give you example today. And these are two lead antibodies from Genentech and Merck, like tiragolumab and vibostolimab. They have clinically completely failed.
So how does TIGIT signaling work? In just 1 minute, basically what happens is TIGIT bind to this tumor expressed receptor called CD155 with high affinity. The activating receptor is CD26, but the affinity is 100-fold lower. You can see 100 millimolar versus 1 millimolar. So every time there is a chance, this is basically the inhibitory receptor is engaged. And this is an evolutionary thing. We want to keep car under the brake so the immune system is not overactivated. So TIGIT became a hot target for last 10 years. Ira Melman was at Yale School of Medicine. Genentech brought him, and this was his pet molecule for last 10 years. And in every single tumors, you'll see a trend that normal versus tumor, there is a high TIGIT expression in colon cancer, breast cancer, ovarian cancer, you name the cancer. TIGIT expression on the CD3 positive T cell is pretty high. So that obviously give a target like PD1, PDL1, or CTLA that, okay, this is a great target for immunotherapy. So obviously, every time there is a target, a lot of big companies go make antibodies and then take it to the clinical trial. That's what happened for 10 years. What happened now in last year? Here is the data. TIGIT has completely failed. There is a complete disaster with the TIGIT clinical trial, phase 2 trial, phase 3 trials. And there you can see that Roche, which is Genentech, they have pulled out their antibody, tiralogumab, even in combination of PD-1 have failed. You can see everything, cancer trials have failed. And now you can see they are firing like whole division. So Genentech has completely shut down their division of the cancer immunology. The Ira Melman has gone as a vice president. He's now a Parker Institute. Merck is laying down people. So TIGIT is the failure of the decade. And I'll tell you a story today, which was started by two undergrads, and we will explain you that something went wrong. So, for example, this failure is not new. There was a failure one time for the CD26 and CD28 antibody. Again, CD28 activation domain, a lot of you people put in your CAR T cells. So there was an idea that we can activate CD28. And what happened was there was a trial given to 8 patients, 6 patients died within 3 days. So that was another disaster for the CD28 agonist antibody for immune checkpoint. And the difference was what they found is that when it was tested in monkey, the CD4 effector memory cell did not express CD28. And when they were tested in monkey, it was very safe. So just to let you know, everybody, every time there is a clinical trial, before that you test into the mouse model, you get a good data. Then finally you study in the monkey. You want to make sure it's a safe. You do the dose estimation studies, and then you take to the human clinical trial. When CD28 antibody failed, it was found and discovered that it is because in monkey and even in mouse, the expression of the CD28 on effector memory cell, as you shown here in the D, is significantly less compared to the human. And that is why there was a cytokine storm and the death of the patient. So, with that hypothesis, we decided to test like maybe similar thing might be going on in the TIGIT because it was a good molecule. All the data showed it should be successful, but every clinical trial from Genentech, Roche, Merck, it has completely failed. So, and again, another argument for us was because we know that our ancestors, as I said, are significantly less susceptible to cancer. You know, human and primates, we share 98.5% of the genome, but but the cancer incidence rate, for example, ovarian cancer, monkey doesn't even get aggressive ovarian cancer, and you can see the human. So huge difference in cancer disparity. So then I gave this work to two undergrad students. These are two undergraduate in my lab, Eric and Francis. And I was like, we had a good study in FAST, which was published decently. So I said, let's test this hypothesis if that might be going on with the TIGIT. And the idea was, we are a protein engineer. What we did was, we generated a lot of these pairs like CD25 TIGIT, ICOS, PDPDL1 as a Ig conjugated proteins. And our goal was in all these protein in middle, as you can see, the red star. I don't know if there is a pointer, the red star you see, we engineered a protease plasmin site. So every time there will be a cleavage, the 25 kD band will be released. But if there is any additional band released, because on the top you see the gray, that's the extracellular domain of these immune checkpoint pairs. You should see a two bands rather than the one band. That's the idea here. And so what they did was they purified, they expressed clone purified human PDL1, rhesus monkey PDL1, CTLA, sorry, ICOS, CD20, CD155, TIGIT, all the pairs, human. And we did just incubated with the plasmin. And as you can see, these are full Western blot. There is a 25 kD band release in all of them, which is because we engineered plasmin site in all of these receptors. Except what you see in the rhesus TIGIT in the F, we see that there is a 28 kD band released only in the primates but not into the human. So that was interesting observation that in monkey, the TIGIT is cleaved by the protease plasmin. We also wanted to make sure that let's test this with the monkey plasmin. And so that's what we did. We generated monkey plasmin, and we also tested with the monkey plasmin. And again, what we see on the left-hand side is a human TIGIT panel, right-hand side, the rhesus TIGIT panel. Rhesus TIGIT produced extra band, and the extra band is coming because it is being cleaved in between the ectodomain of the receptor, which is the TIGIT receptor. So that's very exciting. Then finally, I'm not going to show, we did a comprehensive mutagenesis to find out where the band is being cleaved. And we made a lot of mutations and did the screening. Ultimately, it led to that it's being cleaved at the position R119. As you can see in human evolutionary, we have TGR, but the monkey have RGR. So there is evolutionary substitution, and I'll explain why there is evolutionary trade-off resulting to the cancer. And we saw, oh, wow, this is the site we confirmed. And biology is very interesting. If you look at the structure level, the CD155 and TIGIT, they interact within lock and key like interactions. As you can clearly see in the E, almost like lock, the key is getting into the lock shown here with the black CC loop, key, and lock I have written. So what we did was we made a mutation in the Y133, which is basically a mutation into the key. And you can see in the G, the binding is completely gone. And in another case, then we took the TIGIT full length and saw its interaction with the CD155. Obviously, it binds at the nanomolar range. Then what we did was CD155 TIGIT, we took the soluble cleaved fragment, which is released by the plasmin in macaque. And you can see it's also similarly binds to the CD155. That suggests that the cleavage product in monkey, not in human, can be released, and that soluble cleaved product can bind to the CD155. Then what we did was, because we have a national primate research center, we got the PBMCs from the monkey, and we tested the same thing. We enriched the CD3 positive T cells in human and the rhesus, and then we look for the TIGIT expression in minus and presence of the plasmin. And you can see the human, there is no significant change, as you see in the B, N of 3, but in case of rhesus monkey, you can see that there is significant loss of the TIGIT from the cell membrane, from the lymphocyte T cell membrane. And when we use the inhibitor of the plasmin aprotinin in the right hand panel, you can see that it clearly rescues that lot of loss. And similar in the western, in the bottom, in the C, you see that there is a significant rescue at the 40 kD band when we use the inhibitor. Again, confirming that it is being cleaved by the plasmin, even on the surface of the lymphocytes, which are the T cells. And then we did IP. In fact, what we took was we grow these PBMCs, we took the soup and we pulled down with the tiralogumab Genentech antibody. And what we found here is, you can see in the lane 4, you see, I mean, it's not the best looking blot, and I'm not proud of it, but I'll show you that there is a pull down only in case of when there was rhesus PBMCs, which is monkey PBMCs, treated with the plasmin. And in the bottom side, what we did was we incubated these cleaved fragment, human TIGIT, which is cleaved by the rhesus TIGIT cleaved by the plasmin, the full length, all these guys, and incubated with the tiralogumab, which is Genentech antibody, which has clinically failed. And you can see all of them form complex on a non-reducing gel and run at higher band, suggesting that tiralogumab, the Genentech antibody, complete effectively bind not only to the membrane TIGIT, but the TIGIT which could be which is cleaved by the plasmin. We also did FPLC because we're protein people, and you see that in this case on the G, the TIGIT, the tiralogumab completely shifts towards the higher aggregates in presence of the soluble TIGIT. At the same time, in the bottom, abelumab, which is anti-PD-L1, which is obviously PD-L1 being unspecific, there is no change into that. Again, confirming that the Genentech molecule, which has been in human trial, before going to human trial, it went to monkey, the data is completely wrong. Because what they wrote is this is a pretty safe study. Instead of working on the T cells, the released soluble TIGIT is saturating this tiralogumab and potentially taking away from the lymphocytes. The next question we asked is if these antibodies, probably if they occupy TIGIT, tiralogumab, ipstolimab, Genentech, and Merck's antibody, can the plasmin still cleave? And, sorry. So what we did was in the B, you can see these are the potential on the A with the stars. We are showing their epitopes on the extracellular domain. And in this case, you can see the epitope, most of the epitope region shown in the gold and yellow falls into this lock region. And this area where it is T19 is R119 is a little bit far away from that area structurally. And this is the vibstolimab epitope in the right hand side. In the middle, it's a tiralogumab epitope. So what we did was we pre-incubated these receptors, TIGIT Fc receptors, with these antibodies. On the left, you can see antibody alone. In antibody, we also engineered plasmin site. In the right hand side, antibody alone. In the left hand side, they were pre-incubated for 1 hour before adding the plasmin. And you can see, despite presence of these antibody, plasmin is still able to release and produce this 28 kD band, again suggesting that even if these antibody in primates were able to bind to the lymphocytes, the target cell, the plasmin protease, which is, obviously in blood, it is involved in the blood clot, but in tumors, it's highly elevated, especially in ovarian tumors. The whole SIT formation is directed and driven by the plasmin mediated cleavage of fibrin. So the plasmin protease in tumors and in monkey, in general, can release the soluble TIGIT, which is a negative regulatory molecule. So this is our working model. What happens in human lymphocytes is that's the working mechanism that the antibody which we have designed, tiralogumab, vibstolimab, can bind to the TIGIT and can allow CD155 to engage CD26 and let the T cell activating. But what happens when these studies were done in monkey? That this TIGIT is cleaved, soluble TIGIT is released. We showed you the data that soluble TIGIT binds as good as the full length TIGIT to the full length CD155. And in the same way, I showed you the data that the soluble TIGIT can be engaged by these clinical antibodies. Showed you example of tiralogumab and can be taken away from the lymphocytes, which is not the intended working mechanism. So then we asked, because as I said earlier, that primates generally don't get cancer. So we asked, maybe this evolutionary substitution is present in all the primates, and maybe that might be going on. So we did some evolutionary tracking of this mutation, and what we found is, as you can see in the right hand side, the 119, you can see it goes from prosimians, old world monkey, new world monkey, great apes. What happens is that this mutation starts happening into the new world monkeys as well as in all the great apes, which is human, gorilla, but not into the, again, not into our very old ancestor. They still maintain this guy R, which will release this negative regulatory TIGIT, and is one of the mechanism by which, we know that it's an immune inhibitory and can really advance the cancer. But those our ancestors found a way to chop it off and leave that negative thing out of it using the proteases, but not the human. Obviously, there are a lot of work that cancer is a price for our big brain. There is a lot of work has been done that the apoptosis, the brain size, we have like threefold more neuronal cortex in human compared to the primates. And there's a whole lot of work. We also have published some good papers in that direction, that the cell death is another mechanism which is undergoing a lot of evolutionary trade-off. A lot of proteins which are involved in the spindle checkpoint assembly, they are evolutionary mutating significantly. For example, I'll show you this simple thing, that why there is a mutation, why this mutation is in human, like great apes versus the old world monkeys or simians, because they have evolutionary mutation rate in these old world monkeys,
Sir,
as you can see, 10
please
to 15.
conclude. We are out of time.
Out of time? Okay,
Yeah.
this is my last slide. So 10 to 15 mutation per generation, but in human, we get almost 70 to 100. So we get a lot more mutation every time we give birth to the kid compared to the primates. And that's why evolutionary these things happen. So ultimately, our goal is that we can apply these findings from our ancestor, learn this. So for example, soluble TIGIT is a great therapeutics, which our primates, our ancestor has already been applying to them, and they don't get cancer. So although there is, with human brain, all these advancement, evolution, we have cognitive abilities, all this improvement in another layer of immune system, but there is evolutionary trade-off which result in the cancer. So I explain all this. So in the end, I would like to acknowledge the people which do the work. This is my lab. They're undergrads, graduate student, collaborator. This is one of the pictures from the Christmas. These are our funders, and we have postdoc position available if anybody interested. And I'll be happy to take a question.
Thank you. Any questions? Anyone?
So nice talk. So, I mean, I don't know if it's a question or it's just a comment. We were told that mice is not a good model. Now you tell us that you are telling me that even now these models are sometimes can be deceiving. So, do you suggest to sequence these regions for any safety studies that are going to be executed with respect to testing these antibodies?
Sure. Yeah, it's a great point. And yeah, obviously the last studies we can do in monkey is because they are close to us. We have similar genome, 98% similarity. Most of the pathways work very similarly. It's most of the proteins are pretty conserved, but for some of them, like as I told you, for CD28, now for the TIGIT, we have found certain. So What we are saying is it's still a great model, but Genentech need to go back to their safety data and dose estimation data and maybe do the clinical trial different ways than what they have done, because a lot of those are decided based on, okay, we have 50 mg per kg, 100 mg per kg. Based on that, those are decided. And in fact, TIGIT did not even work with the combination of PD and one and that.
And the next comment was, why would we mutate
Sir, can
a,
you?
a residue which is not which probably is evolutionarily causing cancer? I mean,
Yeah.
in apes they don't have cancer, as you said.
Yes.
So
Yeah.
what was the reason of this? There's no selection there.
Yeah,
The
the brain.
negatives.
Yeah, the most of the so for example, spindle formation gene, for every 5,000 years we are getting 10 mutation in it, and every mutation enhances our cell division process. We have published a work like fast ligand, which is in wire for cell death. We have acquired a mutation that there is less death to develop our brain. So some of them are random evolutionary trade-offs which are happening. And as I said, the evolutionary rate, the mutation rate in human great apes is much higher. So the chances of some random mutation. But again, yeah, we need to be careful about some of the data.
Thank you. Thank you, Dr. Joginder. That's a nice
Thank you
talk.
very much.
Yeah. I invite next speaker. Dr. Mayank Singh is an associate professor in the Department of Medical Oncology Lab from AIMS, New Delhi. His research focuses on immunotherapy, ubiquitin, deubiquitin signaling, and epigenetic regulation in cancer, emphasizing cancer stem cell biology and liquid biopsy diagnostics. So, Dr. Mayank, please.
Is it there? Yeah. Thank you. Good afternoon, everyone. So after the evolutionary biology, I think let's just shift our and go to the immunotherapy. So my work basically, we at AIMS work on my lab work on different aspects in the cancer biology. Predominantly, like we used to work on a lot of intercellular signaling, like your ubiquitin signaling and basically S-track inhibitors. But when we shifted to AIMS, so we focused our areas to the cancer immunotherapy, and that is what we are. So basically, if you see that we work on deubiquitin signaling cancers, we work on the dub inhibitors, specifically USP37 and USP14. The second area which we work upon is the development of the CAR T cell based therapies. Basically, these are the adoptive cellular therapies which we work upon. The third area of our work is the liquid biopsy in different cancer. Predominantly, we work on head and neck cancers and the cervical cancers. But today I'm going to talk about one thing which has come up really in the recent time, and it is basically what we call as a CAR T cell therapy. If you see, this is a typical flowchart of a CAR T cell therapy. This is how it works. And if you see here, it's basically the way it is done is that you extract the blood from the patient, then you do apheresis and or leukapheresis, and isolate the T cells from the patient. Then what you do is that you take out a CAR, which is called a chimeric antigen receptor, and then you actually transfect it to the T cells. And what you get out of it is basically what we call as a CAR T cell. And these CAR T cells then are expanded ex vivo. And once they are expanded, and after that, they are infused into the patient after some days. And generally, the timeline is 20 to 25 days. And once it is done after that, these CAR T cell will go and bind to your target cell, which is basically your tumor, and then it's going to kill it. So this is how the CAR T cell therapy works, and this is the typical CAR T cell therapy workflow. So if we talk about like the different like the generation of the CAR, when we talk about CAR T cell, we talk about different generation of CAR T cells. See, CAR T cell is not something which is new. It has been here for a long time. If you see in the 1970s, the Weizmann, at the Weizmann Institute, the Ishar et al., they showed their first generation of the CAR T cell. If you still look at that, that is the first generation of CAR T cell. But what happened in that case is that these T cells were unable to give optimum amount of output. The reason was that the optimum co-stimulation was absent in those. So that is why when the Carl June group took over, they actually what they did was that they added an additional co-stimulatory domain, which is called a second co-stimulatory domain to that. So what it did is that after that you get a T cell persistence, the proper killing, and majority of the CAR T cell which you see in the market today are basically and approved by the FDA also are the second generation CAR T cell therapies. Then you have got third generation, fourth generation, fifth generation. Fourth and the fifth generation are collectively called armored CARs because they are generally used for the solid tumors, and they have additional domains which basically allow these T cells to survive in the harsh tumor microenvironment, which is the solid cancers. So when we talk about the CAR T cell, it is very important to have a proper marker, because you see the CAR T cell, when you develop a CAR T cell therapy, it is not against a particular cancer. It is against a marker, right? It is against CD19 in the case of AML. Then you have got the BCMA, which is called B cell maturation antigen. So that is where the marker is very important in this case, because the CAR T cell is going to kill anything which is going to express that marker, right? So that is why it's very important. If you see the CD19, it's a good marker of BLL. If it's a good marker for non-Hodgkin lymphoma, though that is why it is approved to that, because it has got a restricted expression on these cancer subtypes. If you see BCMA, or a B cell maturation antigen, it has got a good expression on the multiple myeloma. So that is why you have the multiple myeloma, you have the anti-BCMA CAR T cell therapies, and in non-Hodgkin lymphoma, you have CD19 CAR T cell therapies. So, but these are the other markers which are also in that. But generally, if you see the CAR T cell therapy, this is how it looks like. But this is an older slide. So what you have right now is that you got four CD19 specific products in the market. Recently, one more was approved. Then you have got two BCMA specific product in the market, also approved by the FDA. So this is how it looks like as of now. If I talk about the BCMA, like a B cell maturation antigen, this is how it is. You have got the ligand. The BCMA is expressed on the surface of the plasma cell, which is the multiple myeloma cells, and after that it binds with this ligand, which is upwell, and and does the NF kappa base signaling. But if you have the inhibitor of a gamma secretase, then gamma secretase is going to cleave these BCMA, and after that, most probably the CAR T cell therapy is not working in those patients. So that is why you got high gamma secretase inhibitors are combined with the CAR T cell therapies in this case. And there are many clinical trials, whether you talk about ADCs, antibody drug conjugates, your CAR T cell, and there are multiple trials are going on which are targeting BCMA. And these were the result of the Karma trial. It was a 72% overall response rate. So it is a pretty safe target, that is that. So if we come to this is how the multiple myeloma looks like. It started from MGUS, just proceed to all the way to plasma cell leukemia. And by the plasma cell leukemia, you have got very much high expression of the BCMA there. And this is generally what happens in multiple myeloma. You have got this treatment naive tumors. You give them the first line of therapy, then the mutations occurs in these patients. You give them second line of therapy, then as further mutations occurs, and these occupy and accumulate and accumulate. So finally, what you have is you have a relapsed and a refractory patient, and for those lines, the CAR T cell therapy is approved in this setting. So when we started this work at AIM, so we thought, okay, so we the first thing what we need to do is that we need to generate our own antibodies, like because the generally when you do go for a CAR T cell therapy, it's a chimera, it's a basically chimeric antigen receptor. So you need to have an SCFE which has come from an antibody, which is going to be a BCMA ECD antibody. So let's start by like first generating our antibodies first. So we thought we will use three different approaches, but finally we stick to the hybridome approach for production of our own antibodies. And this is how our cassette looked like. Basically, what you are seeing here is that we have got that this is an SCFE which was derived finally from an antibody. Then you have got a hinge domain, then you got a transmembrane domain, and after that you got two co-stimulatory domains. So the car which we were working upon was a second generation car. And this was the workflow which we started. First, we isolated the BCMA from the patient who were coming at IUCH. We scored their serum BCMA levels also, and after that, we generated the whole BCMA protein. We generated the BCMA ACD protein as well, and then subsequently we transfected these cells and did all the molecular biology assays to assess how it is. So this is how it looked like. These were the patient who came at ISCH. We looked at their serum BCMA level was up. We isolated the PBMC from this patient, did the PCR, and got the product out of it. And this is what we did that. So this was the cloning and the expression of the ECD domain. So this was just an extracellular domain of the BCMA, which we cloned out. And after that, we tagged with a histide. It was a histide BCMA. So we did the expression. So we got a pretty good expression of this histide BCMA. After that, once we had that BCMA, after that we went ahead, we used the XP293 system to generate, and we got a pretty good yield out of it. It was 250 mg per liter. And after that, we did the further, we basically, what we did that we purified this BCMA protein in association with Dr. Kalpana Luthra lab at the biochemistry. We used the ACTA system, and we got a pretty good, the BCMA ECD protein out of this. Further, this protein was characterized, and we did the HPLC-based purification. Basically, we went ahead and did all the purification steps, did the mass spec analysis, and further, after that, what we did was we got these hybridomas. These are the 20 hybridoma clones which we got out of this immunization studies, and out of that, the top 5 clones were these clones. These were the 5 clones, And after out of this 9C4 clone, which you are seeing, has got the highest affinity. We got the best affinity out of these clones. So once we have this 9C4 clone, we went ahead and then obviously we thought, okay, we now you have the pretty good antibody binder. So let's just sequence this and see how it goes. So then we went for the sequencing of SCFE, which we basically outsourced, and we did the generation of lentiviral plasmid encoding these BCMA and TCDs, and then we did the viral particle generation. So this was the methodology which we used for that. So once we did the BL analysis of this antibody, also the 9C4, we got a pretty good curves out of it. So we understood that our antibody was working pretty well. It was binding to target and that. So this is what you are seeing here is that this is our own protein, the BCMA ACD protein, and we have used our own antibody to actually clear. So that was pretty clear. The band was pretty clear. So after that, we thought, okay, let's then we had our collaborating department of biophysics, Dr. Hidayat. So we worked with them, and then we went ahead and started sequencing the domain of this antibody. And what we found out was that we found 4 unique epitopes in these antibodies, which we generated. So the binding was pretty good in that case. So we further did the molecule studies here, and after that, so once we were like happy, okay, look, everything looks good here. So we took the SCFP out of this antibody and we cloned into the second generation car. And this is was our car, which we basically it was basically a BCM ECD car. And after that, we first did the transcription of the 293 cells. We got a pretty good expression. Then we isolated the T cells from the patient and the lentiviruses. We were finally able to generate the CAR T cells out of it. So this is how our actually the whole workflow looked like. We got these antibodies, we characterize them. In fact, what we are working right now, we have cloned this SCF into 3 different vector backbones, the CAR backbones, as well as multiple SCFEs have been generated. The top 3 SCFEs have already been characterized at our end. And we have did that cytotoxicity assays. So I'm not showing that data here. But right now, what we are trying to do is that we are trying to like push for really like the murine models studies, especially the nude mice studies, and see like where it goes. The other thing what we are doing, what we are seeing here is that this is how it looks like if we have developed a BCMA CAR, but we also understand that obviously we have done that. Then if you see the failure of the anti-BCMA CAR T-cell therapy, what happens is that the BCMA loss that happens at many times in the multiple myeloma patients. So we think that that it can be addressed by developing a most value bispecific CAR. So this is one specific aspect we are working upon. We are working on development of a anti-BCMA SLAM F7 CAR as well. As well as what we are working upon, we have taken the N94 antibody and we have conjugated it with the orostatin F, which is like a drug. So we have trying to develop this ADC-based approach to tackle those things as well. This is one. And then this is how it looks like. This is how the CAR T cell therapy looks like. It is like obviously CAR T cell therapy have got very good efficacy, but the point is that they are very expensive. We all know that. But then you are also trying to work upon on an ADC approach, like you can develop off the shelf product also simultaneously. So this is also what we are trying to look upon. So generally when you work in this area you keep hearing that okay you've got many CAR T cell therapies in the hematological malignancies. What about the solid malignancies? What is happening in that? So the reason we don't have a good product in the solid malignancies is that because we don't have a good target as of now, and because we don't have a good target as of now, that is why you get a on target off target effect in the solid cancers. But recently I think there's a lot of progress has been made in the glioma. That thing is being done. Secondly, the third way is to develop what we call the armor cards. So by armor card, you can develop car which have NTPD combination or HIF1 alpha inducible car or catalase car. So generally what happens is that when the T cells go into the hypoxic tumor course, is that you got that way the T cell actually do not work. The T cell energy occurs in those cases. So those are the approaches which are being used. You can work on the design of the car, something which we are also been trying to do here. So this is what we are looking at right now. So we have generated a preclinical second generation car. We have filed for the Indian patent last year. So now this is being published also. And what we are trying to do is that we are trying to develop a pipeline for future CAR T cell development, protein scale up, expression, antibody engineering, in vivo studies, and capacity building for development of lifestyle therapy at Ames Delhi. But if you see in the Indian scenario, it's not easy to work on these kind of things. That is what we have understood. Like the development of the academia and the industry consortia is important here, with clear demarcation of the shared responsibility, how we are going to do that. If we do something in the academia level, then how would we take it to the patient? That is one of the important things. The focal point should not only development of CAR T cells, Also the development of the vector, the delivery systems should be researched, and that is where the academy industry needs to come in. The clear-cut cost sharing system need to be there. The focus should be on the in-house production of the regions, so that the reliance on any particular source cannot be there as such. So obviously, let me give you a perspective. Right now, the ID cell it cost in US like $120,000, the single infusion of the CAR T cell therapy. So if you want to do something in India, you have to significantly bring the cost down. It has to come at different levels at different places. So that is how it has to happen. So this is our which we work upon. I'm there, Dr. Ranjit Saur, Dr. Raja Parmanik, they are the medical oncologists with me. Then there's Dr. Mohanty, I think Dr. Kalpana Luthala, I'm a supported is helping all the like all the protein production and the protein purification assays. So that is how it is. Thank you so much. We will take the questions.
Thank you, Dr. Mayank. Any questions?
So you get your sequence human patients. Then you do the testing in preclinical model. Does that work?
Get the sequence from that. We preclinical model, yes, we are doing that.
No, so same, or you have to change the antibody sequence.
So the thing is that what we are getting is we are generating an antibody again an ECD. That's the extracellular domain of the BCL. So
That is conserved.
yes, that's conserved.
What is the time from you expect? I'm not saying you are starting, but eventually, what do you think? That is also very important, right? From beginning till you get your CAR T cells. What do you what do you estimate
So
how long it's going?
that timeline is something which you run against always. Either that is supposed to be 24 days. So generally, what you do here is that you put the patients on the immunodepletion therapy and basically reprogramming the whole system. Then after them, you before that you actually take out the cells and you engineer them. So generally, what happens, what we have also seen is that we face multiple issues. We take out the T cells from the patient, we culture them, they go into senescence. There's a lot of T cell energy which is present in multiple myeloma patient. First, second thing is that even we have tried taking the patient from healthy donors also, they also suffer the energy sometimes because we have to, that is why we have to do a lot of stratification. Secondly, after that, once we do the transfection, with these, right now what we are doing, we are using a conventional system, which is not actually lot like a system which you use for the large scale production of the CAR T cells. We are in talk with some companies which are already working in Indian space regarding the indigenous lentiviral system, which we can use in our clinical trial once we go ahead. But yeah, those are the things. So the whole thing, the immunodepletion, and after that production, expansion, and infusion, the whole thing should happen within 25 days. That is the standard timeline that is going around, and that is what the people. But for that, you need a really smooth system to go through and.
My ignorance. Can you preserve them
Sir,
and
can
then
you
inject, or do you have time?
can we
Yeah,
later?
they
Can you
discuss
freeze
those
and then
things.
inject? Is it does that work?
Yeah.
Can you freeze your RT and then
No,
revive?
generally
That
not this time. Not this
Thank
that then that will be the off the shelf product, right?
Thank you, Dr. Mayank, for such a nice talk.
Thank you, thank you.
Our next speaker, Dr. Prakriti Tayari, is a professor at the Department of Bioscience and Bioengineering at IIT Bombay. Her research involves developing material platforms to recruit and program host cell in vivo regeneration of tissue or manipulating immune system for cancer therapy and various immune-related pathological conditions. Dr. Prakriti Tayala, please.
First of all, let me thank the organizers for giving me this opportunity to present my work here at this forum. And I also want to thank Dr. Mayank Singh for setting the right context for my talk and how he's actually saved me some time from a limited time that we've been given. So I'm going to talk about the CAR macrophages, which can be programmed in vivo for triple negative breast cancer immunotherapy. So just to give you a brief overview of what my lab does is we are a biomaterials focused lab and we work on developing 3D scaffolds as well as nanoparticles for various applications, including tissue engineering specifically for wound healing purposes, and developing in vitro 3D platforms for developing tumor and disease models. But what I'm going to talk today about using these platforms for cancer immunotherapy purpose and for specifically for CAR macrophages in particular. So just to give a sort of a motivation for why the solid tumor treatment has not worked very well for with CAR-T. So we know the solid tumor, there are about 85% human cancers are solid tumors, and the typical modalities are surgery, which removes the tumor but still left leaves a significant number of tumor cells in the area. Then we have chemotherapy and radiation, which causes injury to the adjacent tissues. Then we have hormone therapy, but which not all cancers are responsive to. And then the recently very common immunotherapy, of which which we can use in a targeted manner. And some of the various types of immunotherapy that we have are the cancer vaccine based or the checkpoint inhibitors based. But what I'm going to talk about is specifically the adoptive cell therapy, which we have heard a little bit about the CAR-T, and today I'm going to talk about the in vivo approach for CAR macrophage therapy. So, not going into the detail of the adoptive cell therapy, specifically the CAR-T, where the process involves removal of cells, modifying them ex vivo, expanding them, and reinfusing them in the patient. The challenges associated with those are specifically, apart from the fact that it's expensive, it is very time consuming. As you've heard, it can take from 3 to 4 weeks to expand them ex vivo, and also the manufacturing and the logistic costs and the transportation cost adds to the overall cost of the therapy. So, and also the CAR-T, as has been told, has been very successful for the hematological malignancies. And but however, for CAR-T in solid tumor, apart from the fact that we haven't found a good target, there are other reasons that there is the T cell exhaustion, and also there is an immunosuppressive environment in the tumor microenvironment, and which makes it a little bit more difficult to penetrate the solid tumor. So, how the CAR macrophage therapy helps is that in tumor, we know that about 40% of cells are macrophages. And the therapy or the approach that we are proposing is that if we can use this motivation for CAR macrophage, where the circulating macrophages can infiltrate the tumors, and which are typically called as tumor associated macrophages, then we can avoid this the typical steps of I mean, in the CAR macrophage therapy, there is also you have to remove the cells and the isolate the monocytes, modify them ex vivo, and sort of reinfuse them. But here we are reinfusing the CAR macrophages instead of the CAR Ts. And the mechanism of action of the CAR macrophage therapy is that just like the CAR T cells, this interacts with the tumor cell based on the antigen receptor interaction, phagocytoses the tumor cells, and then presents the antigen on its surface, leading to the subsequent T cell activation. So, using this mechanism of the CAR macrophage therapy, we know that we want to use this for an in situ modification of CAR-M, which can potentially bypass the logistical challenges of the adoptive cell therapies, where the cells are modified ex vivo, be it CAR-T or CAR-M, where you have to go through the apheresis process and the modification of the cells ex vivo and the reinfusion of the cells, which causes a lot of it is a complex manufacturing process. Plus, there is also a lot of patient variability, which can lead to differences in the time it takes for the expansion and the subsequent reinfusion. So, in our approach, what we are proposing here is that if we can make this CAR plasmid for CAR macrophage therapy and complex with a polymer, which is again targeted to macrophages,
we make this polyplex system, which is targeted to macrophages, and then we do an intraperitoneal injection of these macrophages because there is a lot of macrophages in the mice and in the peritoneal cavity. What we also did is an intratumoral injection of a sting agonist, which I'll talk about later. And post this injection, the sequence of events that happens is that the macrophages get modified with with the help of this polyplex. They get modified with this receptor, and this receptor then helps interaction with the cancer cells. So we have used a PDL1 based antibody as a receptor, and the PDL1 expressing cancer cells interact with these CAR modified macrophages, interact and then phagocytose these cells and express the antigen on its surface, leading to subsequent T cell activation and cancer lysis. So this helps in the subsequent reduction of tumor burden, and that is what I'm going to show in my results today. So as part of the development of this CAR macrophage approach, the development of building blocks involve the development of the delivery vehicle. So we used mannose as a targeting agent because the mannose receptor is expressed on the macrophages. It's a CD206 or the mannose receptor. So we used a chitosan-based system, and we modified it with mannose, which helps in the targeting. So we did the characterization of the mannose-related chitosan. We designed the CAR plasmid and used the anti-PD-L1 SCFE, which recognizes both the human as well as the mice PD-L1. And we also used a fluorescent reporter EGFP as a marker. Subsequent to sort of designing the plasmid and the polyplexes, we made these polyplexes and did a physical and biological characterization of the polyplexes, which involved the size analysis, the charge analysis, and the cytotoxicity of these polyplexes to the macrophage cell lines. And what we see that at a ratio of the DNA to the polymer ratio of about 1:60, we get a reasonable size of about 200 to 300 nanometers of these polyplexes, and the zeta potential is about 30 plus 30, which is good for the interaction with the cells, and the viability is greater than 80%. We further did a characterization of its ability to protect the plasmid or the DNA. And when we incubate these polyplexes with DNAs at different concentrations, we see that it protects the plasmid, which we do not see when the plasmid is incubated with the DNAs, which we see in this gel results. So we see that the polyplex protects the degradation of the plasmid by the nucleus. Further, we also see that for the transfection of cell lines in vitro system, we see that the 1:60 concentration or 1:60 ratio performs well for the transfection of the cells in an in vitro system. Further, we wanted to see if these transfected macrophages can actually uptake the tumor cells. So what we did was we used these various conditions where we used the untransfected cells, untransfected macrophages, and co-cultured them with the 41 cells. As I have not shown the data here, but the 41 cells express about 40 to 50% PDL1. And when we co-cultured these cells, we do not find much of phagocytosis. When we co-cultured these 41 cells with GFP expressing macrophages, we see a little bit of uptake of these cells, which is about 12%, likely because of the adjuvant effect of the mannose-related chitosan. However, when we incubate them with the CAR-T modified macrophages, we see a significant increase in the uptake of these cells, which is about 25%. Then we also looked at the in vivo expression or the transfection of these cells in vivo and the distribution of these transfected cells in tumor-bearing mice. So we looked at both the dorsal and the ventral side of the mice, wherein we injected the instead of the CAR plasmid, we had the luciferase expressing plasmid, and when we inject these plasmids, these polyplexes intraperitoneally into mice, we see that the distribution of these transfected macrophages is a good amount in the lung and liver areas. And when we isolate these lung and liver macrophages and also the tumor macrophages, we see that there is a significant expression of these of the CAR in these macrophages, which are isolated from these various tissues, which tells us a little bit about the cellular distribution of these macrophages in these tumor-bearing mice. So further, we went ahead and did some in vivo studies wherein we we created tumor with these 41 cells which express PDL1, and we removed the tumor and looked at the PDL1 expression of the cells in these tumors, and we see that not only the tumor cells but also the immune cells that infiltrate the tumors express this PDL1. So there is a lot of PDL1 expression in these tumor cells. So this is our schematic for actually performing the mice studies, wherein we create a subcutaneous tumor with the 41 cells, and after about 15 days, we do an intraperitoneal injection along with an intratumoral injection of STING agonist, and we do these injections at a frequency. We do these 4 injections and collect the tumor, spleen, and blood at the end of 25 days. And when we look at the tumor volume, we see that when we compare it with the control, we see that while the CAR polyplex itself performs well, the combination of the CAR polyplex along with the STING agonist perform the best as compared to the other conditions. We also did another condition, another experiment where apart from the STING agonist, we had a condition where we had a GFP polyplex to see the effect of the GFP plasmid along with the STING agonist. And we see that as compared to all of these conditions, combination therapy where the CAR polyplex along with the STING agonist perform the best in terms of the reduction of tumor size and tumor mass, which we see from both of these graphs. Further, we also looked at the macrophage polarization in the tumor microenvironment, and we see that this CAR plus STING treatment condition increases the M1 phenotype of the macrophages, which is good for anti-tumor effect. It also decreases the M2 phenotype, which is an immunosuppressive phenotype of the macrophages. It also increases the CD8 plus the cytotoxic T cell infiltration into the tumor, into the tumor, and there is not much of a change to the regulatory FOXP3 phenotype in the tumor microenvironment, which tells that all of these conditions together help in the tumor reduction and have a reasonable anti-tumor effect. Finally, we also did one ex vivo studies where we collected the spleen from all of these groups of tumor studies and looked at the co-culture of these cells isolated from spleen with the 41 cells. And again, we see that the combination system of the CAR-T, of the CAR-M and the STING agonist had the maximum tumor killing of these 41 cells and also had high cytokine levels, specifically IFN gamma. So together it says that these the combination system actually performs very well in terms of the anti-tumor treatment, and the CAR macrophages actually have the potential for overcoming various challenges that are seen with the CAR-T. The macrophages infiltrate the tumors and can be genetically modified with CARs to enhance the targeting. And the big plus is that we have used a non-viral system, which is considered to be safe for in vivo application, and the combination therapy has shown reasonable effect as compared to some of the other conditions. Finally, I would like to thank my group has helped do various studies in along the immunotherapy area, and not to mention my various collaborations and the funding agencies and our department and institute for providing all the infrastructure support. Thank you, and I can take any questions.
Thank you. Any questions, please?
Thank you. Thank you. Very interesting presentation. So I had a couple of questions. And the first one was PDL1 is expressed normally in gut and other tissues, right? Reactivity down. So I would think that targeting PDL1 might be quite a toxic thing to do. So I'm wondering, first of all, and especially when you combine plasmid that has a CD3 signaling in a macrophage, have you looked at cytokine production? Have you looked at gut damage when you do these treatments?
So we have done the histology of the tissues isolated from the different organs, and we have seen that there isn't much difference. I mean, there isn't any inflammation that we see in these groups. And so,
But did you look longer? Because I saw your tumor model. You harvested mice 3 days after the last injection. So that's a very short period. So if you stop giving your plasmid or your liposomes, and then you let the mice go 1 week, what happens? Is there still protection?
that's a very good point. Yeah. So the reason why we were not able to continue for much longer is because of the ethical, considerations of the control mice, where we have to, we are sacrificing. What we could do in a subsequent study is keep the treated mice for much longer and see what happens. So because we wanted to, sort of sacrifice them altogether, and the limitation with the size of the tumor in the vitro, we had to stop it at day 26, but we were planning to do a subsequent study for long.
Might be worth measuring cytokines in this.
Yes.
In the serum.
Thank you. Thank you, Dr. Prakriti. It was a great talk. Thank you. I invite next speaker, Dr. Kulwant Singh. Is an associate professor in the Stem Cell Research Center, Department of Hematology at SGPGII, Lucknow, India. Dr. Singh studies how cell type specific transcription factors, epigenetic regulators, work together to regulate stem cell lineage progression and how their dysfunction lead to development of disease. Dr. Singh research group is also working on CAR T cell therapy. Please, Dr.
So I would like to thank you, Dr. Ambak Rai, for invitation, and the chair for the kind introduction. So we are placed in the Department of Hematology, SGPGI, Lucknow, and basically I'm not a immunologist by training, but I started working in the CAR T cell area for like 2-3 years ago. And what we are looking for is right now with the CD19 CAR T cell therapy is like 4 of them are uh, approved by FDA, and 2 of them are in India also. But the issue is the relapses. Okay, and one of the relapse is the lymphoid to myeloid cyst that is very much overlooked. And 3 years ago, we started working on this in this area, and I will share some preliminary data that we have. So this is for basically for students. So CAR, the concept of CAR come from this Greek mythological monster, and his this monster head is made from the comes from the lion. Half of the body comes from the goat, and the tail comes from the um, snake. Similarly, the CAR also comes from the 3 different components. So the intercellular part, co-stimulatory domain and signaling domain, comes from the TCR. The transmembrane domain comes from the CD-alpha CD28 and hinge domain, and the antigen binding domain comes from the antibody. So if you change the antigen binding domain, you can target a specific antigen on any other tumor or cell type. So why CD19 is a favorite target? Because it is expressed from the pro B cell to the plasma cell completely, and basically majority of the leukemia and lymphoma are derived from these cells stages, and that's why the CD19 is a great target. And similarly, the BCMA is also expressed in the multiple myeloma, like Dr. Mayank said, and uh, and this is the favorite target for the multiple myeloma. So, uh, so the CD19 CAR T cell therapy has given a excellent response in uh, relapsed and refractory kind of the tumor cells. For example, complete remission rate in BLL was 81 to 86%. Multiple mantle cell lymphoma is like 67%, so on and so forth. And this therapy is also associated with the toxicity, so like CRS and ICANS. So Indian CAR T cell therapy has also similar kind of the response. So uh, this is a great hope for uh, the patient who are relapsed or refractory to the existing therapy. But despite initial response, eventually the patient relapses. And the mechanism of the relapse could be two types. One is antigen positive relapse that can be shown in the right side, and another one is the antigen negative relapse, where that is shown in the middle. In antigen positive relapse, the CD19 receptor is maintained on the surface, but CAR T cell is non-functional. So the CAR T cell is exhausted. And in the antigen negative relapse, you can see the pre-existing CD19 tumor cells can start growing, or the CD19 is down regulated on the surface of the uh, tumor cells, and this is how it escapes from the tumor cells. And the third kind of the mechanism is the tumor cell switches its lineage from lymphoid to myeloid. In this condition, the target antigen CD19 is automatically downregulated because of the lineage switch. So, and it occurs in this initially it was thought it's like a rare types of the relapse, but recently, and sorry, and this lymphomyeloid switch, it doesn't occur only in case of the CAR T cell therapy, but it also occur with the bites. Bites are the bispecific T cell engager. So what they do, they have two SCFB domain. So one goes to bind to the T cell, and it brings the T cell close to the tumor cells, and it also works in a antigen dependent manner. So it was initially thought that it's a rare event, and I think that's why it is not. People were not working in this area very much, but it attracted me, so I started working on it. So and region was so recently a study was published in the blood from Nirali Shah group at um, NIH, and she basically collected the data from the multiple institute, and she identified that the around 7.4% of the patients re, of total relapse patient were account for the lymphomyeloid switch. So then we started working in this direction, and sorry, I started working in this direction earlier, but this is a 2025 paper. And to study this, we made a model. So what I did is we took the lymphoid cells. This is a CD19 positive lymphoid cells, and inducibly express the CBP alpha. This is a marker for the myeloid lineage. So it's a master transcription factor that reprograms the lymphoid, and it in a cytokine cocktail of the IL-3 and MCSF, and the cells change towards the myeloid lineage. And the way we measure is we measure the CD19 or CD19 and as a marker for the lymphoid, and MAC1 as a marker for the myeloid lineage. So this you can see here, it's uh, there are two phases. So at day 0, 99% cells were CD19 positive, but as the differentiation proceed, you can see the in quarter 2, right? CD19 MAC1 positive cells comes, and those CD19 MAC1 positive cells again converted into single MAC1 positive cells. So the differentiation transdifferentiation process is occurring in a two phase. So in one phase, the CD19 is downregulated, shown in the blue color, and the double positive cells shown in green color is so the CD19 positive cells are getting down and the double positive cells are going up. So and after 3 days, the CD19 the double positive cells come down and the MAC1 positive cells goes up. Okay, so there are two phases, and so in this model we also characterize the gene expression. So the transcript level of the CD19 was reduced, and very interestingly, the upstream regulator of the CD19, that is PAX5 and EBF1, was also down regulated. Okay? During myeloid differentiation, during this differentiation process, and and the markers for the myeloid lineage like CD11, and several cytokines were also increased during the transdifferentiation process. We also, when because the leukemia cell is non-adherent, and when it differentiate, it becomes adherent, and it accumulate lots of actin at the one end in the form of the stress fiber, and that can be seen in the right. So the actin fiber is present all around the cells in the right top, and it is localized towards the particular site at the in the bottom images. Finally, we characterize these cells with the help of the electron microscopy, and you can see at least three different types of the changes during transdifferentiation process. The one change is chromatin is very much open in undifferentiated state. When we differentiate it, the chromatin get compact. And second thing, you can see the lots of processes has come on the top of all around the cells. And third and fourth thing is the phagosomes has started appearing. So this confirms that the our system is working. When you express the CBP alpha in lymphoid cell, it is changing the lineage towards the myeloid sites. So using this protocol. So in this model, so you can see so I told you the like two phases. In the first phases, CD19 positive cells converted into double positive cells, and then double positive cells converted into myeloid cells. So in the first phase, we screened around like a small scale inhibitor screening, and what we found is in the what we found, so the blue color bars, the bottom one is the percentage of the CD19 positive cell, and the top one is the double positive cells. And what we found is last two drug 10 and 11, these were significantly inhibiting the lymphomyeloid switch process. Okay, so and of particular interest, why we are interested in the drug 11 only, the reason because the safety profile of this drug was already established. Okay, so one thing you can see here is in case of the drug 11, the number of these cells were like relatively lesser than this. So this inhibition in transdifferentiation is also possible because of the maybe toxicity. Okay, so we uh, performed a gradient of this inhibitors, and now you can see in this slide that the even at 50 nanomolar uh, or 100 nanomolar, the CD19 positive cells are going up, and the CD19 MAC1 positive cells are going down. And the bottom green color line shows the PI. So based on this data, we decided to go for the concentration of like 200, 300 something. Eh, so it seems like that at this concentration, possibly it is not a toxic. So um, to confirm this data, we use another analog of this drug. Okay. So we call it as a drug 12, and we observed the same kind of the phenomena in this. Okay. And so finally, what we did, we just checked the expression of this uh, lymphoid and myeloid genes in the presence of the inhibitor, and these genes were all the lymphoid genes were found upregulated. So the system is back, and the myeloid was downregulated. So with this, I would just we have a long way to go, but I will just convince that the if the CD19 uh, is present, CAR T cell can target it. But if the lymphomolyte switch happens, is drug 11 and 12 can inhibit it. So in the next phase of the our study, we are going to go work with the in the mouse system to develop the lymphomyeloid switch. Uh, but we have. So with this, I would like to thank you for my team. I have a very small team, and the Pran Chris has done all kind of all work that I present um, today, except the electron microscopy work that was done by Dr. Subhash Chandra Yadav at AMS New Delhi. And I would like to thank ICMR and DST for the funding. And uh, thank you for your presence, and I'll happy to have a question.
Thank you. Any questions?
Yes.
Have been able to identify the mechanism of these drugs? How does this block the switch?
Yes, I know the mechanism, but because we have the preliminary data, I know where it works, but we have the preliminary data and we haven't published it. So I'm sorry at this
We can
time.
wait
I
to
can,
understand.
I can tell you exactly.
Thank you. Okay.
So it used to be like the people used to think that not think that it is a rare event, but in last the reason why it is not been able to detect it, because post therapy relapse, after post therapy, so median time is just 4.8 months. So possibly it is not. But
There are other reasons as well.
there could be other reason. So I'll tell you two things. So the two reason people are hypothesizing right now is the one is the immune pressure. So the immune pressure put by the car can also induce the lymphadenopathy. And the second thing is the CRS. So in CRS, the interleukin 6 level goes very high, like 1,000 times. Okay. And there is a very old paper, maybe 20, 30 years ago. Someone has shown that the interleukin 6 can induce the differentiation, can switch the lymphoid to myeloid lineage in very selected type of the leukemic cells. So for the time being, only two mechanism is being considered.
Thank you. Thank you, Dr. Gurun Singh. Such a nice talk. So I thank all the speakers for their excellent talk and insightful discussion. My gratitude to the organizers. It was an honor to chair the session. And thank you. Thank you, everyone.
On the behalf of the organizing committee of Immunocon 2025, I extend our sincere gratitude to our respected chairperson, Dr. Srikant Sadhu, for the valuable time, guidance, and for conducting this session with such grace and depth. Your insights and leadership have truly enriched today's discussions. As a token of appreciation, we would now like to present memento to our distinguished speaker. I request our chairperson, Dr. Shrikant Sadhu, to kindly come forward and present memento to our esteemed speaker, Dr. Kulwant Singh. Can we have a huge round of applause? Dr. Jogendra Tushir Singh, Dr. Mayank Singh, and Dr. Prakriti Tayalia. Can we have a huge round of applause? I request all the speakers to be there on the stage for the group photography session. Also, a very kind request you to please stay here on the stage for the photography session. Now, I request Dr. Samish Srivastav to kindly present memento to our respected chairperson, Dr. Shrikant Sadhu, as a token of our heartfelt gratitude. With that, we come to the formal conclusion of our session, Immunotherapy. We thank all our chairpersons, speakers, and delegates for their active participation and valuable contribution. We will now break for a tea, which will be served in the refreshment area. Kindly reassemble here in the MP hall for the next round of talks following the tea break. Thank you for all your participation. Please enjoy your tea break. मैं अपना सिस्टम डायरेक्ट कनेक्ट कर सकती हूँ तो वो हो जाएगा ना कनेक्शन
कब है?
कल है जीपीटी वाई की सेशन में 10 से 11 क्योंकि टाइम वो है ना ये लैग कर रहा है सिस्टम एनिमेशन लास्ट वाला है लास्ट वन जीपीटी लास्ट मिनट का ये तो है
कल है आपका?
ये कल है
कल आ जाइए, कल हो जाएगा, हो जाएगा।
ये मैं इसलिए अच्छा कल नहीं काम कर
नहीं मैम, आज भी होता, आपका आज भी हो जाता, कोई प्रॉब्लम नहीं है इसमें। इसको बस निकालना, आपका लैपटॉप आ जाएगा।
ये किसी बिना फोन में चल नहीं पाता है बहुत खराब एक्सपीरियंस रहा है वहाँ बहुत ऑकवर्ड होता है कि चल
हो जाएगा,
हो जाएगा ना
हाँ। तो मैं आपकी पीपीटी, चलो आप अपने से कर लें। चलो
ट्राई कर
थैंक यू।
थैंक यू
जरा अपना स्लाइड अपने प्रेजेंट करेंगे कब?
अब ये जरा इसमें
कल
कल वाली में अभी चेक करना है बहुत देर से ये जरा चेंज कर दीजिए
काट दो ना, सब काट दो, सब काटो काटो काटो।
मैं ये कह रहा हूँ वॉल्यूम उनकी वाली पहले वाली डिलीट कर देना ये वाली डाल देना इसमें कर
यार वो बहन का लोड़ा उसी पेन ड्राइव पे नहीं, उस पेन ड्राइव में इस सिम हाँ हाँ
कर्सर बनेंगे कर्सर बनेंगे
कौन सा है, कौन सा रिवाइज्ड?
ये
किसका था ये?
तो वाला तो बोला
कौन से में साली तुम्हारे में कितने ड्राइव्स हैं बस? दुर्गा पाल
तो हो चुका ना तो वो गलत था
अब ये बताओ इसमें से किसका किसका हो गया? डिलीट कर दो और पहले इसको रीनेम कर दो। ये क्या होता है यार, कैसे ये हो जाता है? वो कुछ नहीं है, शिवम को फोन करके कह दो हमारा चाय यहीं पहुंचवा दे। क्या करवा दे? हमारा चाय यहीं पहुंचवा दे।
मैम का दे दूं हो गया ना
अरे
I request our chairperson, speakers, and the participants to please return to the MP hall as we are starting the session soon. I request everyone to please reach the MP hall as we are starting soon. I again I again request all our dignitaries to please come back to the MP hall because we are lacking of time. Welcome you all. We hope you all had a refreshing short break and are ready to dive back into another exciting session of Immunocon 2025. We now begin our session with a special talk marking the start of today's scientific discussion. It gives me a great pleasure to invite our distinguished chairperson to kindly come forward and take the seat on the dais to lead the session. We are honored to welcome Professor Nivriti Das, former professor at AIMS, New Delhi. Professor Das is a highly respected figure in the field of immunology and biomedical sciences, renowned for her outstanding contributions to autoimmune disease research and immunopathology. Her research has focused on autoimmune diseases, immune regulation, and translational immunology, contributing significantly to the understanding of immune mediated disorders and their therapeutic interventions. A dedicated academic and mentor, Professor Das has trained numerous young scientists and clinicians, leaving a lasting impact on India's immunology research landscape. With our esteemed chairperson on the dais, I would like to request them to kindly take charge of the session and invite the speakers for their respective talks.
Uh, Thank you for your kind introduction of mine. And I'm extremely thankful to the organizers for giving me this honor to chair a session by none other but Professor N.K. Mahra, a very well-known immunologist, histopathologist, and many other things that he is contributing to the field of immunology and Indian science. So it is my very pleasant privilege to invite Professor Mehra to come onto the stage and give his lecture, which is, I'm sure, going to be very interesting and very engaging. So, Professor Mehra, we always love to keep on listening to you, but there is this time restraint, so you have got 20 minutes. We will give you the signal
Thank
for
you
5
very
minutes.
much. May I request those who are in the tea room to please come inside the hall? I'm not used to speaking in a hall like this, where there are more seats that are empty than occupied. So again, a request to all those who are in the tea room, please come, including the volunteers. Thank you very much. I thought I will speak to. आ रहे हैं कुछ कुछ फर्क पड़ा कि नहीं? Anyway, I'm going to speak to you. You have heard so much about CAR T cells. So the CAR engineered T cells are precision therapy for antibody mediated rejection following solid organ transplantation. You know, in solid organ transplantation, our problem is the antibody formation there. So what we have over there is, if you look at the alloimmunity driving rejection, there are two arms to that. One is the T cell mediated rejection, which is largely under control, but we do have very effective immunosuppressive drugs, and also no routine test is available. The antibody mediated, which we call ABMR, is a big problem. Out of this, again, there is an acute phase, not very common because we do match the donor recipient very well through the NGS based typing, and we also look for the antibodies at the pre transplant stage. Major problem is chronic rejection due to the occurrence of donor-specific antibodies or the development of what we call de novo development antibodies following transplant. Hence, we keep doing the monitoring of these antibodies. If you look at the alloreactive T cells, their fate following. There is a direct pathway and indirect pathway. And many of you who work in the field of transplantation know that the CD4 T cell recognizes antigens on the APC both in the indirect pathway, the self antigen, or from the direct pathway, which is the from the donor self antigens over there. And once they recognize the antigen, they move into either the memory phase or the effector phase. They help the B cells to make antibodies or the CTLs and all that, or they ultimately terminate either through apoptosis or energy. Today, with all those drugs that we have, with all those tools of matching that we have, we still have unacceptable rates of long-term kidney graft failure, which continues to be a challenge for us. And the pool data shows that graft survival rates have significantly improved over time. However, by 10 years, half the grafts are lost. Whether you take the period between 1970s to the 83 when calcineurin inhibitors came, or after that, even after that, this is true of both if you use the living donors or the deceased donor for transplantation. So the questions that we have is ABMR through generation of donor-specific antibodies is the main barrier limiting long-term outcome. And if that is the case, how do we keep the B cell quiet? And our current strategy is specific and enough to reduce all antibodies and B cells. And we have seen over the years there's a changing trend in graft monitoring there. We have moved from invasive tests to non invasive tests. However, matching for HLA genes between the donor and the recipient continues to be on the top of the story and antibody monitoring. So keep monitoring antibodies and make sure that you can have a well-matched donor recipient pair. This will avoid production of donor-specific antibodies that are detrimental to the graft. However, there have been a lot of advances in the molecular monitoring and imaging, particularly the liquid biopsies, what we call the donor-derived cell-free DNA over there. This can be used as a test to find out if somebody is going towards rejection or not. And this provides dynamic insights into the graft status. Molecular imaging has also come up. It gives you information on what we call transparent pathology. But there are still a lot of challenges. The key challenges are the most biomarkers have low concentration in the blood and urine, complicating accurate detection. Establishing appropriate threshold values is difficult actually, and it varies from one organ to the other. For example, in heart, 0.5 or 0.75 in liver, and about 1% over the kidney, and anything beyond that, you say, okay, this guy is going towards graft loss. So not a very sensitive test as of now. So you need a lot of validation there. We need to develop standardized protocols as well. We have twin challenges when we are dealing with organ transplant, and I will basically talk about kidney, but this is also true of liver and heart. One challenge is our high degree of HLA polymorphism, and as of January 2025, as you can see, more than 20,000 um, alleles have become known, more than 41,000 genes have grown, which means if we are not taking a donor from the family, not the mother, father, or the sibling over there, we're taking from the unrelated pool, many patients will get transplanted with no HLA, with an HLA mismatched donor over there. And the more the matches, more are the chances of development of donor-specific antibodies. The good thing, however, is not every HLA mismatch leads to an antibody response over there. But our question is, which patients will do well and which not? So that still remains a query for us. Our second challenge is what we call antibody challenge. The antibody challenge is identification of non-immunogenic HLA mismatches. Some mismatches are good enough and we can do the transplant. It is doing extremely well, but we don't know which one. Alternatively, based on the success of the CAR T cells, and you know, we have been talking about CAR T cell therapies in cancer, can we use the similar system to overcome the ABMR? At least 3 independent studies have been done around the world, which have used genetically engineered chimeric HLA antibody receptor. We don't call it a CAR here, we call it CHAR, because it is a chimeric HLA antibody receptor, and this can be used, can be transduced to into human T cells. We also have an important contribution by the non-HLA antibodies. I just brought these slides to make you understand that besides HLA antibodies, we have very important role of non-HLA antibodies as well, out of which one very important is the MICA. Now, the term non-HLA antibody is used in transplantation to describe autoreactive and alloreactive antibodies specific for targets other than HLA there. And out of these, as I said, there are many of them: vimentin, collagen, tubulin, endothelial cell proteins. The MICA, the MSC class 1 chain related antibodies are most important there. And we've done a lot of studies. I don't want to go into details of that. Right from 2007, our lab has been doing these studies, and we found patients who have both antibodies, both HLA as well as a non-HLA antibody, they will have a worst graft outcome compared to if there were none. And even those who have only one antibody, whether HLA or non-HLA, the graft survival is compromised. Let's look at first the mechanism of ABMR. How does these antibodies develop? You can see the anti-HLA class 1 antibodies are formed by either the B cells, the B cells which are turned into plasma cells, and one variety of plasma cells are called the long-lived variety of plasma cells. They make antibodies to HLA specific B memory cells, and long-lived plasma cells generate anti-HLA class 1 antibodies that bind to the HLA molecules on the renal graft endothelium. Activation of the classical complement pathway occurs when the C1q binds to the Fc domain of the antibody in immune complexes, leading to the establishment of MAC on the endothelial cells. And that's how the endothelium gets slightly damaged. And the immune cells, such as NK cell, neutrophil, macrophages, also have a very important role as well. When we look at tolerance versus rejection, the question that comes up is B cell your friend or a foe? Could B cell depletion, because antibodies are anyway being formed due to the B cells, the B cells were turning into the plasma cells. Could B cell depletion be a good strategy to achieve tolerance? There are three pathways, as you can see. Number one, allo antibody, as I said, is a major barrier to transplantation tolerance. Once the donor-specific antibody develops, half of these guys will lose their graft. So we have to be very careful that we don't let these guys make antibodies. Second is antibodies adhered to the endothelium. As you saw, damage results in endothelial proliferation, intimal thickening, rapid peripheral circulation, incompetence, and finally necrosis there. But the question is, B cells are functionally heterogeneous. We only talk of B cells, but B cells are also heterogeneous. And this is based on their location, ability to migrate and contribute to T cell dependence and response. And third, of course, is regulatory B cells. Very important function of what we call B-Regs. People had forgotten the B-Regs. People only talk about the T-Regs. B-Regs have a regulatory effect via cytokines, IL-10, 35, and it can be better there. We have used in organ transplantation a number of drugs. This is a commonly used pharmacological agents that we have which target the B cells during the different development stages. And the most common among them is rituximab. The B cell targeting therapies lead to reduced incidence of ABMR, creating sensitizations and induced tolerance in them. Rituximab in association with IVIG. You give somebody develop antibodies pre-transplant, and I only advise my colleagues, okay, go ahead and give them rituximab along with at least 6 plasma pheresis and IVIG, and that would help. And it has been helping so far. But a randomized clinical trial in humans have shown that the use of rituximab as a sole inducing therapy led to high incidence of ACR. ACR had not been seen in transplantation. The more rituximab you use, you're seeing ACR. It is possible that B cell depletion lead to the loss of the B regulatory cells. Once the B regulatory cells are lost, there's a cytokine storm, and that activates the T cells, and therefore you also develop the ACR. So there are more troubles than the solution. These are the current treatment strategies. that we advise our transplant team once the antibodies are there, PPI, VIG, with or without rituximab, monoclonal antibodies against CD20, but mind it, CD20 is only on the B cells and not on the plasma cells. PPI, VIG, as I said, protease inhibitors, there's several of them, complement inhibitors that are already known, or use of monoclonal antibodies to several of these agents over there. But the question is, these strategies lack specificity and reduce all antibodies and B cells indiscriminately. That's our negative point. We don't want all the B cells to go off. Patients become vulnerable to viral and bacterial infection. We have seen patients who have been given a lot of immunosuppression or these drugs that they die of infections. Occurrence of de novo malignant tumors in cells in them, as I said, the depletion of B-regs may lead to the ACR. So the need is to have more effective and specific therapies tailored to the individual pathophysiology and clinical manifestations. Here I come to the role of the CAR T cells. In 2013, this was a paper which came up in 2013 by Humphreys, honing that killer instinct in the T cells. You sort of do the CAR antigen, the chimeric antigen receptor there, harvest the T cells from the biopsy or from the blood. You use the T cell infiltrate, the tumor infiltrated lymphocytes one side, or you take the CAR T cells and also use them ex vivo and then infuse them back. So you've seen this slides actually, and this seems to work. So the question is, can we use these also for the transplant aspect? Engineered T cells as precision therapy, how they're being used for the cancer, and I've seen that there are a lot of people during the morning have shown you these slides. They take the cells from the blood. The CAR T cells are autologous, and Rahul gave the lecture on the day one. CAR T cells have to be therapies autologous. It is mostly used for leukemias there. I have a company, my company, I'm mentoring a company where we're trying to manufacture car NK cells and car gamma delta cells. NK also because my name is also NK, and NK is a natural killer cell. And you can take that off the shelf therapy. It's not an autologous, heterologous. It's taking us time. We've already worked very hard for about 4 or 5 years. We're very hopeful by the end of 26 or early to 27, that is 4 cancers, 4 solid cancers will be treated by CAR NK cells. Engineered T cells and precision therapy is also being used in transplantation, both in HSCT, the hemato stem cell transplantation, as well for the treatment of GVHD as well. I don't want to go into details of this slide. And this 4 or 5 papers have come up. They're still in a trial stage, phase 2, phase 3. Some of them are approved, at least in Japan, and these are being used. And I'll show you how does it work. This is the company that I'm talking about, the company that I'm mentoring. It's called the East Ocean Bio, an emerging T cell therapy company. Our curative technology for multiple cancers and all existing options. The curative for multiple solid cancers off the shelf. Cost is very less. If you import this from outside, it will about 20 to 25 CR. Our effort is to make it available to Indian patients within less than 5 lakhs over there. And there are superior side effects of that. And our focus is on this is the product portfolio there. We are at the moment looking at the solid tumors, 4 of them. I'm not going to details. Maybe next year I'll come and talk how far have we gone there. Now, this made me feel if we can make this car NK cells as a treatment of solid cancer or the other diseases over there, can we also use them for transplantation? So, the engineered T cells as precision therapy in solid organ transplantation. You know that the chimeric, the CAR T cell therapy is emerging as innovative immunotherapy. Hematological cancer, tumors, CAR T cells. Hematological cancers only, not any other. CAR T cells will not be useful for any other at the moment. They are like living drugs that potentially persist lifelong. Our hypothesis is could be engineered T cells be used to eliminate donor specific B cells and thus overcome the ABM, the antibody-mediated rejection. So engineered T cells, we call them as CORA T cells, chimeric alloantigen specific T cells overcoming rejection by antibodies. And CHAR T cells, not CAR, CHAR, because the H is for HLA there, chimeric HLA antibody receptor are being developed to specifically target and eliminate the B cells. These engineered T cells equipped with novel antigen receptors designated to look into to recognize the alloreactive B cell. These are studies, very important studies that have been done. I will briefly talk about some of these, in the next 2 minutes or so. This is a study. This is done in Spain. Chimeric anti-HLA antibody receptor engineered T regulatory T cells suppress allo antigen specific B cells from pre-sensitized transplant recipients. So what they've done is this is the allo cell. You see the char over there, the HLA-A2 positive and positive. The slide is not very clear. Schematic representation of char receptor featuring a CD28 CD3 GDOS signaling domain expressed on the surface of a human regulatory T cell, binding an HLA-A2 antibody in the surface of an allogenic B cell from an A2 pre-sensitized. If that can be done, we can block the B cell. The B cell will not be making the antibody. We are only blocking the B cells and not other varieties of the B cells. This is where the mechanism works. See the B cell here, and the CHAR T cell is made. So the extracellular region of CHAR construct consists of like of the HLA, the alpha 1 domain, alpha 2, and the alpha 3 domain over there, S specific on the HLA1 molecule. And epitopes on this HLA class 1 molecule can be recognized by the B cell. This binding of the BCR, B cell receptor, will activate the CHAR T cells, which will release cytokines and cytotoxic granules such as interferon gamma, enzyme B, and perforin, and therefore block the B cells. So a very simple but a very effective system of looking. People have also tried to use the CD19 CAR T cell therapy to reduce B cells and anti-HLA antibody, but this strategy has not worked. There was a lecture just prior to T. They were talking about the CD19 CAR T cell, but they are not very effective because they're unable to eliminate the long-lived variety of plasma cells. That's the issue with that. The other study, CAR T cell therapy, paving the way for sensitized kidney transplant patients. Those who of us who are involved in kidney transplantation, once the patient gets sensitized, it's a problem for us. It's unfortunately a big problem to give him a transplant. And this is a study that there is a novel strategy of using CAR T cells to target and eliminate B cells in sensitized recipients. And I don't want to take too much of your time. This study shows that CAR T cell therapy is a viable option targeting B cells in solid organ transplantation. I have only 2 more slides. Now, as of now, this is a schematic structure of different chimeric receptors under study. Not just one, but 5 of them. It is not yet clear as to which would be ideal for desensitization or for treatment of APM receptor. I talked to you about the CD19 CAR. I said CD19 CAR may not be helpful because it's not blocking the long-lived variety of plasma cells there. And we've seen that patients who are given rituximab are still unable to remove those antibody graft. Or you can make A2 CHAR or the CORA cells and all. These are various um, possibilities of chimeric receptors. So what I've shown you in this very little time, the occurrence of de novo anti-HLA class 2 antibodies associated with even higher incidence of rejection and worse outcome in SOT. I showed you that HLA class 1 antibodies are as bad. Nevertheless, CHAR T cell therapy has emerged as a revolutionary treatment strategy to successfully target the HLA class 1 specific B cells and thus prevent antibody formation. The limited experience that we have so far, it shows that the technology has a promise. It's getting standardized. It is possible that greater number of CHAR constructs may be required. If somebody is HLA-A2, the antibodies are formed to HLA-A2, not a single construct, and that is where the more work will actually go into what should be the quantum of the con- constructs you need. The technology could be very helpful to desensitize patients with high immunologic risk. Patients with high immunologic risk have a big problem that they don't find a donor, or those with limited chances of finding a compatible graft. But there is a caution here. While the CHART T cell therapy is effective against the B cell, it alone may not be able to eliminate IgG forming LL, LLPCs, which is an obvious limited technology so far. This has to be addressed. We need a multidisciplinary, multicentric approach to address the unmet needs in making personalized treatment for ABMR. I'm sure many of the youngsters, please look into this emerging technology. It will definitely be used. I'm sure that in the next 5 years or so, you're not going to use this rituximab anymore. You're going to target the B cell, the LLPs, the long-lived plasma cells directly, not for those expensive drugs and the graft. Madam, thank you so much. Thanks for giving me this opportunity.
Thank you, Professor Mehra. I really wonder, continuously you trying to minimize the failure of transplantation. Every time you are coming with something new for us. It is wonderful, Professor Mehra.
That's
Not
a
only you are a
that's a,
pioneer in the religion.
that was a problem from my nephrologists and transplant surgeons in AIMS. They used to say, Dr. Mehra, you always bring up something new for. I mean, what do we do? I said, I'm trying to help you guys. I don't want you to use those very expensive drugs. I want you to look for antibodies because today, for until 2010, the debate was whether antibodies are a cause or effect of the drugs. Because you would understand if you put something new from outside, if you put a kitty from outside, the host will make antibodies there, right? So the antibodies are a cause because of the graft. But the graft failure is effect. So cause or effect. And that took a lot of time. And now we know antibodies are bad.
Short and sweet talk. There will be one question. We are running out of time. So one question to a young soul. Dr. Mehra will be very happy if we took
Yeah, the president of the society comes at the end of my talk. अरे पीछे नहीं आगे बैठे हैं आगे
Rest can be discussed later.
Okay, anyway, meet me and we will talk about it. Thank you.
Dr. Mehra, thank you very much.
On the behalf of the organizing committee of Immunocon 2025, I extend our sincere gratitude to our respected chairperson, Professor Nibriti Das, for their valuable time, guidance, and for conducting this session with such grace and depth. As a token of appreciation, we would now like to present memento to our distinguished speaker, and I request our chairperson, Professor Nibriti Das, to kindly come forward and present memento to Professor N.K. Mehra. Can we have a huge round of applause? Now, I request Dr. Samir Srivastav to kindly present memento to our respected chairperson, Professor Nibriti Das, as a token of our heartfelt gratitude. We now move forward with a very special segment of the conference, the Women Scientist Session. This session celebrates the remarkable contributions, leadership, and scientific excellence of women in the field of immunology and biomedical research. It's an honor to showcase their inspiring work and groundbreaking ideas. With a great pride, we are heartily welcome our distinguished chairperson to kindly come forward and take their seats on the dais to lead this session. We are honored to welcome Dr. Amit Awasthi, professor at Translational Health Science and Technical Institute, Faridabad. Dr. Awasthi's pioneering research focuses on immune regulation, T cell biology, and host pathogen interaction, especially in infectious and inflammatory diseases. He is widely recognized for his scientific contributions toward understanding the immune mechanism that drive diseases, pathogenesis, and therapeutic responses. His work continues to shape modern immunology and inspire the translational innovations. Accompanying him, we have Dr. Santosh Kumar Kar, former professor at Jawaharlal Nehru University, New Delhi. Dr. Kerr is widely recognized for his significant contribution to vaccine development and immunological research, with impactful work spanning human and insect immunity, molecular biology, and translational biotechnology. He also served as the vice president of the Indian Immunology Society, reflecting his leadership and service to the scientific community. We are honored to have both with us today. We are requested to kindly come forward to lead the session and present the Women Scientist session and felicitation to our distinguished awardee.
Thank you. So we are here at a very important session, recognizing the contribution of our women scientist and a colleague on this very important occasion of annual meeting of Indian Immunology Society. And as we actually, I mentioned this little bit about my inaugural speech that when we took over in IISc Bangalore, and one of the things we really wanted to do is to bring women immunologists to lead from the front. I know they are leading from the front, but I think they need to lead front. I mean to say. And in this, for doing this, we started this uh, in February. We recognized 13 women scientists, and this recognition and this selection process has happened through a committee, a women education committee. We had five women who got these applications across India, and we really wanted to make sure that we are selecting these participants, or we are honoring from every part of the India. It's not necessary from the north only or the south. So therefore, we received many application, and we zeroed down to 13. And at that point of time, we were not really sure how this recognition will be given to them, the medal and the certificate and the honoring them. And subsequently, I discussed with Dr. Ambak Rai, who is organizing secretary of Immunocon, and I spell out that this is what we have done, and we are thinking to really call them here, and then they can have a special women's session where they will talk about their science, and then we can felicitate them on the stage in front of everybody. And Dr. Amma kindly agreed, and this is what we hear. So without wasting time, I request my co-chair, Dr. Santosh Kar, to start with the introduction of the first speaker. Thank you.
Hello, Dr. Arati Sundarajan, who is an accomplished immunologist and assistant professor at the Institute of Science, Nirma University, Ahmedabad. She earned her PhD in microbiology from the University of Tennessee, Knoxville, US, and pursued postdoctoral research in microbiology and immunology at the Emory Vaccine Center, Emory University, USA. Her research explores reproductive and viral immunology with a focus on understanding immune regulation during pregnancy, labor, and pregnancy associated complications. Dr. Sundarajan has received prestigious recognitions, including the Royal Society of Tropical Medicine and Health Early Career Grant, the ACRB Startup Research Grant, and the CSIR Aspire Grant, reflecting her continued contribution to advancing maternal and child health research. Dr. Sundaraj. So she is supposed to speak something? She has some time, No? 12 minutes. Okay.
Good evening, everyone. It's a pleasure to present my lab's ongoing research work today at IMMUNOCON. I am very grateful to IAS Executive Committee, the Women Education Committee, and also the conference organizers for giving me this wonderful opportunity today. So my lab basically right now it's trying to understand the role of maternal interferon alpha and dendritic cell subsets in pregnancy and specifically labor initiation. And we are also interested in exploring their role in pregnancy complications. So, specifically, preterm birth, pregnancy induced hypertension, and maternal anemia. Pregnancy is actually quite a remarkable immunological state. So you see that it's a very cleverly orchestrated sequence of pro and anti inflammatory processes. And it's kind of like a phased immune modulation. So you see very much at the beginning of pregnancy, the phase one, where implantation is involved and establishment of maternal fetal interface. So at this point, you see a very controlled inflammation. Then, as you are progressing towards phase two, which is involving immune tolerance and fetal growth, at this point, you have largely a predominance of anti-inflammatory environment. And then as the pregnancy is progressing forward, you see that there is a requirement of labor initiation and parturition. So the shift now goes towards a pro-inflammatory environment. So there is a lot of innate immune cell activation, cytokine and chemokine production, and many other immune cells are also recruited at this point. Okay. So, labor overall, the process of labor, it involves many things. So, they all culminate together and initiate the labor process. Some of them are listed here, but overall, the labor phase is generally divided into three stages. So, the first stage is this full 10 centimeter cervical dilation. Then you have the second stage, the birth of the baby, and the final stage, the placenta expulsion. So, but within this first stage of labor onset, you again have subdivisions. So, we have latent, active, and transition phases. So, and that division is largely based on the length of cervical dilation and also the frequency of contractions. And one more important point is that as the pregnancy is progressing within this latent active transition, there is also a inflammatory rise. So you'll see cytokines like IL-6 and IL-8, which are often, elevated as you progress from latent to the active phase of labor. So, in this context, we actually wanted to understand interferon alpha what is the profile of this cytokine in pregnancy, and what is its association with labor onset, and also how this is regulated. Okay, so this slide here shows our ongoing study, which shows the cross-sectional profile of plasma interferon alpha. Um, and it's basically normal pregnancy, a healthy pregnancy is what we are talking about. So you can see that as the pregnancy is progressing, very much at the initial phases, not a lot is happening, it's relatively low. But as soon as you hit that labor onset phase, um, right here, you can see this rise in the interferon alpha levels. And we are talking about only spontaneous labor onset here. So if we place it in context of other cytokines, which are well characterized, like the IL-6 and IL-8, the pattern is actually quite similar in the sense that as you move from late third trimester to latent labor to active phase, there is that rise in the cytokine pattern. So we went ahead and did some more work on this. So what we found was there is a significant increase in the interferon alpha levels in pregnant women who are in latent phase of term labor compared to a gestational age matched group of women who are in the non-laboring stage, right? And interestingly, the same pattern repeated in preterm labor also. So basically, the pregnant women who are in the latent phase of preterm labor onset also had significantly higher interferon alpha levels compared to the non-laboring controls. So this suggested to us that perhaps interferon alpha is very much linked with the labor process rather than with gestational age differences. Moving ahead, we also looked at hypertensive complications. So here we noticed something interesting. Yes. That is, so the hypertensive pregnant women who are not in labor, they are demonstrating higher, significantly higher interferon alpha levels compared to the normotensive pregnant women who are not in labor. So, in fact, these levels are quite similar to hypertensive pregnant women who are in the labor phase. And we have other ongoing prospective study also. So, there also we are finding that, in fact, women who have higher interferon alpha levels, they are going ahead and developing hypertension later. So we are seeing this pattern of early interferon alpha rise that is seen associated with hypertensive pregnancy. Okay, so based on these, observations, we, as part of our ongoing studies, we are now focusing on trying to understand what is causing this rise of interferon alpha in the labor onset. Our very preliminary data right now suggests that so we compared both interferon alpha and plasmacytoid dendritic cell numbers from comparing basically this early third trimester and the latent labor phase, where you see that distinct interferon alpha change. And what we found was, yes, interferon alpha is significantly more, but in contrast, the PDC numbers did not change much. So, I mean, it's still at the very preliminary stage, but we are working on expanding this. But we think that the blood abundance of PDCs has nothing to do with interferon alpha rise during labor. There could be some activation profile change, perhaps, or there are other cells which are making interferon alpha. So, our specific focus right now is also on understanding the decidual plasmacytoid disease. So we're working on characterizing these cells also. So, so we are looking at the basalis and periatalis portions of the maternal fetal interface, where we are able to identify the presence of plasmacytoid disease through the HLA-DR negative lineage, population, which are the dendritic cells. Following that, we are looking at the CD123 positive, CD11C negative, which is our PDCs. So we are interested in characterizing them a lot. So that would give us some insight, perhaps into their role in causing the interferon alpha increase as well, because they are very much positioned at that fetal interface where they may be receiving the signals as well. So, the other point that we are also examining is looking at cell-free DNA as a potential trigger of interferon alpha, because as pregnancy progresses, the cfDNA or cell-free DNA levels do rise as the pregnancy moves ahead. And we overlaid this pattern with our interferon alpha data, and we find that the pattern is quite similar. So we match the gestation phase, and it looks pretty much like a similar trend. So this is something we are exploring as well. And the second crucial question is this interferon alpha increase actually a participant in inducing the labor onset, or it is something as a consequence that comes after labor onset? So, given the dual nature of interferon alpha, we would, like to clarify further in future studies whether the pro-inflammatory role of interferon alpha is assisting in the labor progression, or the immunoregulatory role could actually help in bringing down the inflammation, which is crucial for postpartum recovery also. So that is something that we are interested in. I think time is up, but I'll just wrap up quickly last few minutes. A very just 3 slides. So we are also looking at the profile of dendritic cell subsets in a pregnancy-induced hypertension. So preeclampsia is a very, very well-known PIH disorder. Characterized by this dysregulated inflammation. So you see, contrast to normal pregnancy, a lot of imbalance in the immune cells is seen, which ultimately, affects spiral artery remodeling process, and eventually leading to hypertension and proteinuria. So one of the features that was very striking me was that people had found this imbalance in dendritic cell subsets. So there is an increased MDC PDC ratio. And this is also found in cardiovascular disease patients, specifically coronary artery disease patients, and also primary hypertension. So we designed a prospective study. So we took blood samples from pregnant women who were all normal. They were normotensive in early third trimester, and we followed up to see, some of them developed hypertension later, and some of them were normotensive. So we looked into the DC subset profile, and what we found interestingly, although these are early observations, is that this imbalance in DC subsets is evident even before the clinical appearance of hypertension. So we are quite interested and excited about this. We also found BMI nulliparity also as significant predictors for this. So I would like to conclude with some key points with the interferon alpha, changes that you observe in labor onset, and the MDC PDC imbalance, And we think finally that interferon DC axis may actually link labor initiation and also hypertensive related pathophysiology. So this like to thank my research team and also my clinician collaborator, Dr. Rohit Jain, and all the funding agencies. Thank you so much.
So dendritic cells again are involved in pregnancy. So as far as I know, dendritic cells are mostly important to present antigen naive T cells.
Yes.
Do you relate the presence and activation of dendritic cells during pregnancy?
Yeah, so that is, I think, one of their primary roles is, working to induce those T cells. But there is, some studies done where the large part of pregnancy we have Th2 predominance and T regulatory cells are keeping this immune tolerance. So DCs are crucial for priming them in that sense, for differentiating them into Th1 or Th2 cells. So I think that is their critical importance in pregnancy.
The interest of time, maybe one question from the audience, if anybody is having any question. So it looks like no. So thank you very much. Thank So this is my privilege to invite the second speaker of this session is Dr. Sajia Ali. She's from faculty member Sher Kashmir, and she's also associated with the Advanced Center of Human Genetics. And when I read her journey, she has vast research experience. From IISC, she worked in IISC, then she worked in NCBS with Professor Jeetu Mayer, and then she did her PhD from Jawaharlal Nehru University, and then multiple experience. And then she moved finally to Kashmir, where she is working on various cancer and molecular biology experiments and helping designing new therapeutic for cancer. Okay. So stage is on for you, Sazia. Thank you.
Uh, Good evening to all of you. Uh, I'm Shazia. So I'm thankful to IMMUNOCON and Sir Professor Santosh Sir, who has been there when I was doing my PhD from JNU, and Dr. Amit Sir, he has been there also when I was just joined my PhD, and to all my mentors, to God. And I think I started my work from IIC Bangalore and JNU and back to my place. So I'll give a brief overview of what I've been working on, how we can revive tumor heterogeneity, which is a therapeutic opportunity and challenge. This was my introductory slide, which has been already given by Dr. Amit sir. So, a brief overview of what I worked on in my PhD. I worked on breast cancer, and I was working on tamoxifen resistance in breast cancer therapy, where we made a stable cell line TAMR, and we could work with some molecules like F7 and see the cell cycle profiling, which was getting arrested in G1S. And we worked on some of the ROS inhibitions, and we could show that how these molecules can overcome the tamoxifen resistivity in breast cancer, and how these molecules help in increasing the reactive oxygen species, then DNA damage and P53 regulation, and finally cell cycle and arrest and cell death. So there are various type of mutations which occur at DNA level, at protein level, and at chromosomal level. So I'll be dealing with some of the mutations which are called missense mutations, and and their effect on protein level. So this is the normal DNA, and these are the protein. How the substitution, insertion, and deletion affects the protein structure. So, in my postdoc, I had a grant from Young Centers GST. So, I worked on acute myeloid leukemia, the patient samples, and I could see the IDH1, isostrate dehydrogenase, and KRAS mutations. So, what we could observe that IDH1, 2, IDH2, and KRAS genes, there were most of the mutations which occurred were missense. And in some of the patient samples, there was protein alteration which was occurring. So, this is the distribution of SNPs in context of the coding regions. There were frame shift, stop gain, stop loss, and in frame insertions also, but most of the mutations were missense. So, I'm sorry for the slide. So, this is the type 1 IDH1 mutation which we have identified in patients acute myeloid leukemia, how the nucleotide substitution occurs, what kind of variations was there, and these were the amino acid substitutions, and these are the IDH2 mutations which were occurring. And here I have summarized IDH2 and KRAS mutations which were identified in acute myeloid leukemia, and this is the percentage of the variants and the coding region variants, that what are the consequences, as I could see that missense variant was more prominent in such patients. So this is the heat map where we could see what are the different kinds of codings and the regions which were involved. And IDH1 being the highest, like mutated, was having IDH1 gene was having more mutation in the acute myeloid acute myeloid patients than IDH2 and KRAS. So this is the structural missense mutation, which I've shown at protein, how the amino acid change has happened and the substitution and the protein level alteration happened. So acute myeloid leukemia I could summarize, which is a heterogeneous group of population, which involves basically the change in myeloid cells, which we call blast cells. And it's a heterogeneous disease with available prognosis, which affects mostly at the age of 60, and it has 37% survival there. In the younger ones, I think the lymphoid is more, or promyelotic is more prominent than myeloid. So these are changes which happens. This results from the genetic change. Like I showed you the chromosomal translocations or the missense or the other kind of mutations. So the main aim for choosing oncogenic mutations ID which 1, 2, FLT3 also or KRAS was identified in these type of cancers was to guess, can we put them as an biomarker for the myeloid disease, and such molecular variation at the level of SNPs and special distribution across the gene, it may help us to inside the high spots or the functional impact variation of different locations in gene, and we could use them as a biomarker for AML prognosis. So, my present work involves the evaluation of CSR, where I'm presently working in the pharmacology department, PARP inhibitors, which is a pan CSR project for breast and ovarian cancer. So, this is the introductory slide. As we all know, how PARP inhibition prevents PARP repair. In normal cells, where there is homologous rejoining repair, but in the BRCA mutant cells where there is synthetic lethality occurs, and how we can resolve this, how the unresolved DNA damage we can be treated by PARP inhibitors, which leads to cell death. So this is just a primary slide because we are working on it and the trial is going on. Like I have used CD34, which is a hematopoietic stem cell precursor cell line, and how these molecules, the FDA approved olaparib, rucaparib, and we have synthesized some of the IM molecules, and how we could do the hematological toxicities, and some of the molecules were having good fold comparison as compared to the FDA approved molecules or drugs. So this is the PARP inhibitors which we are working in breast and ovarian cancer, and how we are comparing these, that how CSR inhibitors are better than the standard drugs. They have high cancer cell specificity, they have lower hematotoxicity, better enzymatic inhibition, and can be a promising candidate or drug for further development. So this is a bit of rationale of this work, that how we could identify and validate the Q mutant genes or proteins in the cancer as a new target, and how we can evaluate the mechanism driven by mutation or driven drug resistance, identify patients most likely and unlikely which can benefit from such therapies, and give a personalized medicine or a precision medicine for such types of cancers, and how we can target KRAS mutants or IDH1 2 mutants, and how these environmental risk factors or by the antibiotic resistance or gene mutations or by chemoradiotherapy, how all these subject affect this subject, and how we could use our approach. So finally acknowledge Skims Hospital while I had this SERB DST PDF project and DBT DST. Professor Dr. Delafrose, who has been my mentor in one of the postdoctoral program, Dr. Javed Rasool, who has provided the myelite samples. CSR, I am for the present pan CSR cancer project, and Dr. Zuhur, Dr. Anand Goswami, Dr. S. Shashank presently working in the PKPD pharmacology, and all the students and fellows and all the funding agencies. Thank you.
Thank you very much for being on time. We can have quickly a question if audience have one or two questions. If not, then no question. Thank you, Sazia.
Thank you, sir.
Thank you.
The next person to be presenting is Dr. Pragya Acharya. He's an assistant professor in the Department of Biochemistry at the All India Institute of Medical Sciences. Sorry? Medical Sciences, New Delhi. Her research focuses on the mechanism of signaling and biomarkers of organ dysfunction, with a special emphasis on immunopathogenesis of liver diseases such as acute on chronic liver failure and non-alcoholic fatty liver disease. Her laboratory integrates clinical studies with mechanistic investigations using omics-based approaches to identify novel biomarkers and therapeutic targets for liver and kidney dysfunction. Recipient of multiple competitive research grants, including the SERB Power Grant 2022, IUS External Grant 2021, DBT BioCare, and DST Early Career Award. Dr. Acharya has been recognized with the Inshallah Plenary Session Awards 21 and 22 and the NIH Fellow Award for Research Excellence 2015. She serves as the editorial board of peer journals and is an active member of the several scientific societies, contributing significantly to translational research in immune liver interactions and biomarker discovery. Dr. Acharya.
Thank you, sir, for that kind introduction. And I would like to thank Indian Immunology Society from the bottom of my heart, for this honor and this opportunity to talk about my work here. So, let me jump right into my work. The title of my talk today is From Metabolic Collapse to Immune Rewiring. Do pathogenic lipids drive neutrophil dysfunction in acute on chronic liver failure? You can see the name of a very complex disease here, acute on chronic liver failure. Everything that can possibly happen to the liver is in this name, right? So let's see what this complex disorder is all about. My lab in the Department of Biochemistry at AIIMS New Delhi works practically on all aspects of liver disease. Liver disease can be silent, as you very well know, because it's the largest solid organ in the human body, and any damage to the liver in small amounts can be buffered very effectively by the remaining hepatocytes and the remaining liver, right? You also know that liver can regenerate itself. And so, when liver disease progressively creeps up, it is silent and goes unnoticed in maximum number of times. If the liver disease is quite substantial, chronic liver disease is silent and substantial, and on top of an underlying chronic liver disease, you have an acute insult to the liver, like if you have an infection, if you have drug induced liver injury, or an activation of viral hepatitis that rapidly damages a large proportion of your hepatocytes. The liver can no longer buffer that damage because the chronic liver disease has already damaged a substantial proportion of the liver. And that is known as acute on chronic liver disease, an acute insult on underlying chronic liver disease. That is a very, very complex and dangerous situation. Now, liver disease silently progresses through these stages. You have healthy liver. You must have heard of fatty liver, which deals with incorporation of fat into the liver. Then you can have liver fibrosis, steatohepatitis, which is inflammation in the liver, and ultimately liver cirrhosis. From this point onwards, the liver has two directions to take. Either it can do something known as acute decompensation, which we don't need to go into right now, or if there is an acute injury on top of all this chronic progression, you get acute on chronic liver failure or ACLF. Now, mortality in ACLF is rocket high. If a patient who is diagnosed with ACLF comes into the clinic, then within 28 days, he has a risk of mortality of about 50%. That means that 50% of the people who are diagnosed with ACLF on a particular day will die within a month. And that's a very high mortality. And mortality in ACLF is thought to be because of the associated multiple organ dysfunction that also happens subsequent to the liver failure. Now, in ACLF, mortality is definitely linked to organ failure, and this organ failure is believed to happen because of an dysfunctional innate immune activation in ACLF, which occurs due to this massive necrosis of hepatocytes and damage to multiple organs and tissues, releasing high levels of DAMPs and PAMPs. And many times is also associated with sepsis or bacterial infections, and then with a lot of DAMPs and PAMPs, which activate the innate immune system. In most cases, suppose you have acute liver failure, that's another syndrome where you don't have a chronic liver disease, but you have some amount of acute damage to the liver. You also have an activation of the innate immune system, but you do not have as much of a mortality as in case of ACLF. Why? Because you know that any kind of a systemic immune response activation is also then succeeded by a compensatory and inhibitory response. And in most cases of acute liver failure or limited tissue damage, this process goes on as expected. But a lot of studies in innate immune profiling in ACLF and follow up studies show that in ACLF patients, this compensatory inhibitory immune response is defective and does not kick in at the right time. And it's been shown that, so this is a sort of a graph which shows that in normal individuals in the red line, you have activation of SIRS and then compensatory activation of CARs at the right time.
in ACLF survivors, now there are some people who do survive the diagnosis of ACLF beyond 28 days, and those survivors have a delayed activation of CARs and a little bit reduced activation of CARs. But in ACLF non survivors, the CARs response is almost completely inhibited, it doesn't kick in at the right time. And a lot of players, innate immune cells, are implicated in this process, like neutrophils, which have been shown to have phagocytic defects, monocytes, DR loss, NK cells as well. But nobody really understands what triggers this innate immune dysfunction and how these PAMs and DAMs actually orchestrate this process. When I joined our department and at Ames, we were initially, my all these a lot of these studies came out of the undergraduate teaching classes that we take. And I was taking a class on innate immunity and trying to teach the role of macrophages and neutrophils. And while I was doing that, in parallel, I was studying ACLF, and suddenly we saw that there are a lot of papers that came within a very short span of time, which showed that there is an expansion of neutrophil to lymphocyte ratio in ACLF non survivors, which means already neutrophils are the most abundant circulating immune cells. But in case of ACLF, the neutrophil population expands exponentially and the lymphocyte population goes down, thereby leading to extremely high NLR ratios or neutrophil to lymphocyte ratios. That made us wonder if neutrophil expansion is actually an important feature of ACLF and could it be mechanistically involved in acute on chronic liver failure pathogenesis. You remember that neutrophils are your classic first responders in innate immune cells, and they have multiple types and functions: phagocytosis, cytokine production, release of ROS, etc., degranulation, etc. And a lot of recent literature also shows extreme heterogeneity in neutrophil responses and phenotypes. So there are neutrophil subpopulations with different subpopulation markers, much like you have a spectrum of macrophages, M1, M2, you have a spectrum of different types of immune cell populations. In neutrophils, also recently, these have been shown. Transcriptomic profiling of ACLF neutrophils revealed the presence of such neutrophil antigenic markers, and a predominant one that we found in ACLF was CD177, which is a cell surface CD marker, and that was associated with an increase in ELANE and MPO classic neutrophil degranulation signatures. And the phenotype looked as if ACLF neutrophils are kind of poised for active degranulation much more than in chronic liver disease or any other ALF or healthy controls. We confirmed this using flow cytometry, and truly, all ACLF patients actually had almost 90% of their neutrophils expressing CD177 on their surface. And that was distinct and significantly higher compared to all the disease control as well as healthy controls. And ACLF also had a higher representation of netosis pathways, suggesting a very characteristic phenotype. Now, CD177 is a cell surface glycoprotein, and it's a GPI-linked protein, and it essentially, okay, I'll have to be faster. It essentially interacts with endothelial cell surface receptors, and we showed that CD177 positive neutrophils migrate into liver tissue using post-mortem tissue biopsies from ACLF patients. While all these characterization on CD177 was going on, my student Shanali showed that ACLF neutrophils and ACLF immune cells in general have a very characteristic TLR expression pattern as well. Other than TLRs 2 and 4, these neutrophils and monocytes do downregulate most of the other TLR expression. Now, one of the things that our lab has been focused on is understanding how these phenotypic changes come about and can we control them. We have been looking at different aspects and systematically characterizing the serum and tissue profiles using proteomics, lipidomics, metabolomics approaches. Today, I'll just show you a little bit about what we have done here. And because lipidomics was interesting to us, because lipids have immunomodulatory roles. So one question we asked is whether these lipids are actually involved in modulation of innate immune responses in AC. So using mass spectrometry based approaches, we showed that ACLF survivors and non-survivors have altered plasma lipid profiles, and they have very specific, we have been able to identify very specific lipid mediators, specific tag species and phospholipid species, which are different in ACLF survivors and non-survivors. These also correlate with clinical markers of organ dysfunction, and we have also created a CTP score, which is a combination of these lipids with clinical scores, which predict mortality in these patients with almost 90% accuracy. So the question, are lipids immunomodulatory? We did a very simple assay. We took ACLF non-survivor lipids, took donor, leukocytes from healthy uh, individuals, and created an ex vivo system to study them. We incubated these healthy cells with ACLF-derived lipids or healthy lipids. And what we found is that the TLR expression pattern in ACLF patients, ACLF derived leukocytes, could be partially mimicked by lipid incubation of healthy donor cells, suggesting that these lipids could um, regulate gene expression and therefore phenotypes in these healthy leukocytes, and could be mechanistically involved in creating a mortality like situation or mortality like immune response in healthy cells. So we also did a lot of proteomics from postmortem renal and liver biopsies, and looked and found that they had a very significant neutrophil degranulation signature in ACLF patients, suggesting that neutrophil degranulation was a major pathway. Just one more minute. So an analysis of these pathways also revealed to us multiple transcription factors, particularly PPAR gamma, which using molecular docking we have shown can have the potential to bind ACLF lipids, which was significant and discriminatory between survivors and non-survivors, thereby suggesting that there may be a lipid-mediated rewiring of innate immune cells in ACLF through such transcription factors. PPAR gamma may not be the only one, but this is something we have started with, and we'll be looking at others in the future. So in summary, we found that in ACLF patients, you have upregulation of pathogenic lipids, which have the potential to convert healthy neutrophils into a partial ACLF-like phenotype. And right now we are looking at different kinds of approaches to restore this immune balance and restore function of these leukocytes. So with that, I'll end my talk. I thank you for your kind attention. And I also want to thank all these people listed on this slide. First of all, my very, very vibrant student group. They keep me on my toes. They do more experiments than I can handle. And all my collaborators, without which this work would have been impossible. And the funding agencies, of course. Thank you very much.
Any questions? Yes.
Yeah. Thank
data from this adipose-derived lipotoxicity. So the data you have shown in your patients. So do you think there is any association? I mean, the lipid is de novo lipogenesis or it's adipose-derived lipotoxicity?
So that's a fantastic question, Priyanka. So that is a very complicated answer. The reason I have only shown you comparisons between ACLF and healthy controls and completely avoided the CLD group is that CLD in themselves, their immune responses are altered. So comparison between CLD and ACLF, it's almost like having a different disease. So there is definitely a priming from adipose-derived lipids of immune cells. But how that links to ACLF further is a question we'll have to answer later. But it almost looks like a different diseased group in terms of its immune profile. And that is a little complicated to address. So yes, we have identified 4 specific lipid species which are very, very highly associated with mortality, independently validated in a different cohort of patients. And there is a specific triacylglycerol glycerol which we are very interested in exploring. So we are going to do that. The ones that we docked. Okay, here, some of the LPEs, plasmalogens, and this PI, phosphoinositide, the specific PI, and one of the tags.
Thank you, Dr. Acharya. So the next speaker Rikua Nazir is a senior assistant professor at the Microbiology Research Laboratory, Center for Research for Development, University of Kashmir. Her research encompasses medical and environmental microbiology with a focus on antimicrobial resistance and microbiome, whatever. She has published extensively in reputed international journals and contributed book chapters with Springer, Elsevier, Wiley, and CRC Press. A recipient of the Distinguished Microbiological Award 24, Dr. Nazir has led DBT, DST, and ICMR funded projects addressing microbial applications in environmental sustainability and biomedical innovation.
Thank you, sir. Thank you for the kind words. Myself, Turkaya, I have, I'm highly thankful to the organizers for giving me opportunity to present my some work here, and and also to the IIS for nominating us for the felicitation of the Young Women Scientist Awards. And I have done my PhD from University of Delhi uh, under Professor Madhu Khanna. And since then, I have been the member of Indian Immunology Society, and I know you both, sir, since then. After that, I went back to my place and did my postdoc there on outer membrane proteins of Diclobacter nodosus, there where I worked on the vaccine development and other things. And then when I joined University of Kashmir, then I started my work in different fields there. And this is one of the work that we are working on antimicrobial resistance and what are the alternatives to the antibiotics, basically. We all know the mechanism of resistance because of the dearth of novel antibiotics, and these are the different mechanisms of developing resistance as antibiotics when antibiotics enter into the bacterial cell. So we have an activation of antibiotics by enzymatic degradation. We have decreased influx due to altered cell membrane. We have activation of antibiotic efflux pumps. We also have plasmids with antibiotic resistant genes, then no binding of antibiotics due to target gene mutations, and even the modification of antibiotic target site. So then with this idea, like MDR pathogenic microbes, when they are treated with the conventional antibiotics, they develop the resistance. And to overcome this resistance, we looked for some native lactic acid bacteria, and where the bacteriocins are being treated as the antimicrobial peptides. So therefore, these probiotic labs, they are the therapeutic antimicrobial agents, and which is right now an urgent command to work with. So why lactic acid bacteria? Like it's these bacteria has been worked out since ages, like since the fermentation of foods have started, and even these probiotic labs, they produce AMPs in the form of bacteriocins, and they have tremendous medical importance, and their exploitation in medicine is a novel approach to treat with the AMR. And due to their antimicrobial activities, these labs are efficacious in reducing the global emergency directly and even in the form of secretions, that is antimicrobial peptides. So now the bacteriocins as the alternatives to the antibiotics, these are highly valuable AMPs and which are ribosomally synthesized in both gram-positive and gram-negative bacteria. And the majority are known to originate from gram-positive bacteria. And due to their high stability and the low toxicity, Bacteriocins are very much beneficial for human pathogenic infectious diseases, and several bacteriocins have been in focus, particularly those from the labs. So this is the classification of the bacteriocins. I'll move on the slides so as to save the time. So now, how bacteriocins work? Bacteriocins, they have the antimicrobial role of antimicrobial role, and which basically occur due to the inhibition of pathogenic microbes via the quorum sensing, through even pore formation in the membranes, or through signaling the immune cells, and thus they modulate the host immune system. So this slide is showing us the two ways, like for the gram positive and another of the gram negative. As the gram negative are basically having effective barrier against some of the gram positive bacteriocins, which are secreted by the gram positive bacteria. So here we have the pore formation, what happens, and cell disruption. Then they can even dither in the cell wall formation. Outpouring of ions and metabolites can take place. Then even the depolarization of the membrane bilayer and disorientation in the septum formation also. So on the other hand, gram negative, they have interrupted replication and transcription, inhibition of ribosomes with delayed and disturbed translation, even blocked or congested chaperones, improper protein folding also. So role of bacteriocins is not limited. It plays a role in various ways. Like it is they are anti-hypersensitive, they are anti-diabetic. They have a role in microbial modulation as well, and even the cholesterol lowering, and on the whole immunomodulation. So bacteriocins, they directly either kill or reduce the colonization, and while stimulating the production of AMPs, and they also activate dendritic cells, neutrophils, mast cells, macrophages, and then thereby also lead to the secretion of more cytokines, chemokines, and other factors. So there are certain these are certain bacteriocins with the anti-inflammatory and the anti-modulatory properties, which have already been worked out. And the antiviral and the antiparasitic action of bacteriocins, here we have the disruption and inhibition of viral glycoprotein synthesis, and inhibition of intracellular viral multiplication and assemblage also. And on the other hand, when it comes to the antiparasitic action, depolarization of mitochondrial membrane and increased production of reactive oxygen species in parasitic microbes take place. So further, when we look for the anti-cancer and the anti-inflammatory inflammatory action of bacteriocins, so they increase the fluidity of membrane by forming the ion channels on cancer cell membrane, and thus increase the levels of LDH, ROS, and pro-apoptotic genes like BAX and BCL2, and and then increasing the expression of anti-apoptotic genes. So they also lead to the increased apoptosis and necrosis, thus reducing the migration and proliferation by inhibiting the mitochondrial energy metabolism and glycolysis. So on the other hand, these bacteriocins, they modulate the immune system via anti-inflammatory and immunoregulation, and also through the anti-inflammatory cytokines and antimicrobial substances. So this slide is showing the potential bacteriocins that are preclinically that have been tested against several pathogenic microbes, and these bacteriocins, they have been successfully given the clinical trials. So our lab is basically we have isolated the probiotic isolates from different sources, like cold adapted labs from the fecal matter of the endangered animal that's there, and labs from chicken gut and the fish. So we have collected from even from the rhizospheric soil. So we have collected uh, these labs from different sources and looked for different mechanisms. Like we have first isolated these labs, and then went to the DNA extraction, amplification, and the sequencing process, where we have mm, isolated 45 isolates from the native. This work is basically belongs to one of the PhD scholars of my lab, who is at the submission point right now, and he has worked basically 45 isolates from this native chicken, and then from boiler he has isolated 35, and among them we have got two isolates, NC10 and BC3. So NC10 has shown a very promising result, which we have further proceeded for different probiotic properties and other safety assessments. So, probiotic properties, high acid and bile tolerance survives the GI conditions. Strong autoaggregation and co-aggregation have been found with better gut adhesion and pathogen exclusion. And even the powerful antimicrobial activity across multiple pathogens have been seen. Non-hemolytic and mostly free of virulence genes uh, have been found. And then further, we have carried out the purification and characterization. We have purified NC10 bacteriocin 7.2 KDA stable at 100 degrees Celsius, proteinaceous in nature and highly active. And for on LCMS-MS confirmed the peptide sequence and 90% homology with known bacteriocins have been identified. The Legilactobacillus salvaris NC10 showed highest probiotic score in PC and heat map analysis, and a promising antibiotic that has an ability to help fight AMR, enhances the health support, supports sustainable farming in poultry, and indigenous chickens have are valuable source of novel potent lab strains. Another study, just give me 2 minutes only. So that belongs to the another scholar of my lab, and she has worked on rhizospheric soil with the same parameters, almost probiotic characterization and in vitro safety evaluation. And we have identified 26 novel AMPs and with the different probiotic characteristics, and then with the LCMS and other ultrafiltration, Amicon ultrafiltration cell. So, where we have further moved with the in vitro model infection with the bacteriocin and given the treatment in the cell lines and then infection model. And then in vivo, have done with the certain parameters, and some of the work is still going on in these models. So, the possible mechanisms here, we have the downregulation of NF-kappa B during infection, upregulation of the anti-inflammatory cytokines, reduction of infectious inflammation mediators, TNF alpha and IL-6, modulation of TREG and TH17 axis, and protection of mucosal tight junctions and barrier integrity. So the expected significance we have that they can serve as antimicrobial, anti-inflammatory, immune balancing, potential applications, gut infections, sepsis, systemic inflammatory disorders. It can be an adjunct therapy alongside the antibiotics. And so here we introduce a new candidate postbiotic with a therapeutic relevance.
Thank you very much, Rukaiya. So, in the interest of time, we will not have questions. If anybody is having any questions, you may directly ask at the time of coffee break or the dinner time. So, thank you very, very much.
The next speaker is Dr. Yasmin Chowdhury, and the assistant professor in the Department of Biochemistry, Biotechnology, with a research focus on cancer biology, biochemistry, and molecular mechanism underlying disease progression. Her work explores signaling pathways involved in cancer and other pathological conditions to identify novel molecular targets for therapeutic intervention. A graduate and doctoral student of Northeastern Hill University, Shillong, Dr. Chaudhary also completed postdoctoral research at the Betson Institute for Cancer Research, Glasgow, United Kingdom, under the DBT Overseas Associateship for the Northeastern region. She has been recognized with several awards, including the University Gold Medal from NEHU, the Young Scientist Award by the International Society for Radiation Biology, and the Best Poster Presentation Award from the Indian Science Congress Association. Her research combines biochemical and molecular approaches to advance understanding of cancer signaling and traditional strategy for disease amelioration. Dr. Chaudhary.
Thank you, sir, for the kind introduction. I am grateful to the Indian Immunology. Yes. I'm grateful to the Indian Immunology Society for this honor. I'm also very grateful to the organizers for giving me the opportunity to present my work today. I would like to present a part of the work we have been doing on drug repurposing for targeting metabolic disruption during carcinogenesis. We are specifically interested in betel nut induced carcinogenesis because of the strong association of betel nut use with increased cancer risk and incidence in India as a whole and Northeast India in particular. And while the role of betel nut and its constituents as carcinogens is very well established, less well known is the fact that betel nut use is also associated with a wide variety of metabolic disturbances like increased abdominal obesity, high levels of cholesterol, and hyperlipidemia among betel nut users worldwide. This point is of significance because of the role played by reprogrammed lipid metabolism in fueling carcinogenesis. We looked at this particular aspect of betel nut induced carcinogenesis in our study. We are all aware of the merits of drug repurposing in terms of saving time and investment required for drug development. And among the quite large number of drugs that have been repurposed for cancer therapy in experimental as well as clinical studies, the two which were of interest to us were metformin and berberin. Metformin, we know, is one among a class of different types of antidiabetic drugs, including the dipeptidyl peptidase 4 inhibitors and TZDs, which have shown very good anti cancer effects in epidemiological studies as well as through experimental evidence. And another such class of drugs are the lipid synthesis inhibitors, including berberin and sorafenib, which have been shown to be very beneficial in cancers involving upregulated levels of lipid synthesizing enzymes and lipid synthesizing genes. Both classes of drugs, antidiabetics and lipid synthesis inhibitors, they converge upon the AMPK signaling pathway, which is the central pathway for regulating cellular metabolism, at the crux of which is the AMP activated kinase, which is as the name suggests, activated by very high levels of AMP and low levels of ATP, indicating low levels of cellular energy. So, in terms of its downstream effectors, the ones which we were interested in specifically were acetyl CoA carboxylase, ACC, which is the rate limiting enzyme for fatty acid synthesis, HMG CoA reductase, HMGCR, which is the rate limiting enzyme for cholesterol biosynthesis, fatty acid synthase, which is also required for fatty acid synthesis, the tumor suppressor P53, which is a very critical tumor suppressor, and of course the AT-mTOR signaling pathway, which is involved in very close crosstalk with AMPK signaling. So, with this information in mind, the two research questions we asked were whether betel nut disrupts AMPK signaling during betel nut induced carcinogenesis, and whether betel nut induced carcinogenesis in a murine model can be treated with antidiabetic drugs or lipid synthesis inhibitors. To answer the questions, we employed a very well established model of betel nut use, whereby we exposed mice to an aqueous extract of betel nut at a daily dose of 2 mg per ml over a period of 24 weeks. From the 16th week of exposure onwards, we also administered, along with AABN, either metformin at 2 doses of 75 or 150 mg per kg body weight, vildagliptin or pioglitazone at 5 or 10 mg per kg body weight, and orlistat. So we used orlistat. This is a direct inhibitor of the fatty acid synthase enzyme. We used it at 20 mg per kg body weight, and berberin at 10 mg per kg body weight. So our very first observation was that the ABN, aqueous extract of betel nut, exposed mice were dyslipidemic.
They had highly elevated levels of triglycerides, cholesterol, disturbed LDL, HDL ratios, and much higher levels of palmitic acid compared to oleic acid. So, this abnormal lipid profile was ameliorated when the mice were treated with metformin, vildagliptin, as well as pioglitazone. In terms of lipid metabolism, AABN reduced the expression of PRKAA, which is the gene coding for the alpha subunit of AMPK. It could also very efficiently reduce phosphorylation of AMPK at threonine 172, which is the crucial activating phosphorylation event for this enzyme. So essentially, the aqueous extract of betel nut could inactivate AMPK. And all three antidiabetic drugs, metformin, vildagliptin, and pioglitazone, could restore phosphorylation of AMPK at threonine 172, as we can see in the data. All three drugs could also restore phosphorylation of ACC downstream of AMPK at serine 79, which is very crucial for regulating triglyceride biosynthesis. They could reduce the expression of fatty acid synthase, again required for fatty acid biosynthesis, and they could also reduce the expression of HMGCR, which is crucial for cholesterol biosynthesis. In terms of the cancer studies, we found that ABN exposed mice developed prominent nodules in the liver from the 16th week of exposure onwards up to the 24th week. Treatment for 8 weeks with metformin, vildagliptin, and pioglitazone could reduce the number as well as size of the liver nodules. All three antidiabetic drugs could restore the tissue architecture which was disrupted by ABN. We found that all three antidiabetic drugs could significantly reduce cellular proliferation, which we measured by immunohistochemistry for Ki67 as well as by cyclin D1 expression. They could restore apoptosis in the liver of the ABN exposed mice by downregulating the expression of anti-apoptotic BCL2. In terms of oncogenic signaling, all three antidiabetic drugs could reduce the expression of the AKT1 gene as well as phosphorylation of AKT at serine 473 and a phosphorylation of mTOR at serine 2448, which are crucial for oncogenic signaling. All three drugs could also restore the levels of PTEN, which is a phosphatase tumor suppressor that regulates AKT phosphorylation, and they could restore phosphorylation of P53 at serine 15. This phosphorylation event of P53 comes downstream of AMPK, and it is crucial for the tumor suppressor activity of P53. It was lost in the avian exposed mice, substantially reduced, and it could be restored by all three antidiabetic drugs. So this part of the work we published in these three articles. The results that we got with the lipid synthesis inhibitors were similar, so I will not go through all of them again. What I would like to focus on are some very interesting results. When ABN exposed mice were treated with cisplatin, they exhibited fur shedding and loss of body weight, which are the symptoms of cisplatin induced toxicity. But when cisplatin was combined with berberin, the fur shedding was significantly reduced, body weight was restored. The combination of cisplatin and berberin could restore kidney architecture of the cisplatin treated mice. It could reduce the serum creatinine levels, indicating that the combination could basically reduce cisplatin induced renal toxicity. The combination also reduced lactate dehydrogenase secretion, indicating that it could give protective effects from hepatotoxicity. The combination of cisplatin and berberin could reduce cell proliferation more efficiently than cisplatin alone. It could also induce apoptosis more efficiently than cisplatin alone. So again, the study with the lipid synthesis inhibitors we published in another 3 articles. To summarize, aqueous extract of betel nut induces carcinogenesis in a murine model. by inactivating the AMPK signaling pathway, and through inactivation of AMPK, it could activate AKT mTOR oncogenic signaling. These alterations could be reversed by treatment of the mice with anti-diabetic drugs and lipid synthesis inhibitors. So I would like to conclude by thanking our funding agencies and the three students. All three are now doctorates who have done the work, Jenny, Momita, and Kasturi. Thank you everyone for your patient listening.
Thank you very much for nice talk.
Thank you, sir.
In the interest of time, we will not have questions.
Thank you, sir. Yes.
And we can have questions if somebody later. Thank
Yes.
you.
Thank you.
खत्म करते हैं. Hello. Next speaker is Durba Pal. Durba Pal is an associate professor in the Department of Biomedical Engineering at the Indian Institute of Technology, Ropar, and a DBT Wellcome Trust India Alliance Intermediate Fellow. Her research integrates molecular biology, regenerative medicine, and tissue engineering to develop cell-based therapeutics for metabolic and degenerative diseases. A PhD graduate from Vishwabharati University with postdoctoral training at the Ohio State University, Wexner Medical Center. Dr. Pal's pioneering work has been featured in leading journals such as Nature Medicine and Nature Nanotechnology. She has received several national recognition, including the Nasi Young Scientist Platinum Jubilee Award and the Serve Women Excellence Award, with over 2,100 citation and the H index of 20. Dr. Pal continues to advance innovative biomedical research with a strong translational focus. Dr. Pal.
Thank you, sir. Good evening, everyone.
We are not putting on that.
Thank you, sir, for the kind introduction. I'm very thankful to the organizing committee, the Women Educational Council, and also the Indian Immunological Society, all the executive chairpersons, to consider me as one of the women scientists and giving me opportunity to deliver my work in this platform. So I will start my work with the topic of my title is adipose tissue macrophage, which is playing a very important role in obesity-induced adipose tissue inflammation and insulin resistance. So, as we all aware about the diabetes, because it is a ghar ghar ki kahani, because every house, I think, every some members is having a diabetes. So, nothing to say more that about the prevalence of diabetes, which is how to remove this one. Um, something blocked. Somebody you there? Okay, sorry for the disturbance. So, so what I'm saying that as you all know that 1 in 10 adults having a diabetes, and the prevalence of diabetes is very high, particularly in India, because India is often called the diabetes capital. And as you see here in India, mostly the states of the south and Punjab, the upper belts, these are all having a high rate of diabetes. So why we are interested in obesity? Because the obesity actually leads to this or is a major risk factor for the type 2 diabetes. So obesity means when you talk about the fat cells are having a very important role in our body. So it is depending upon the location and their number of the cells, it is actually comes to the some dysfunctional, and then it leads to the diabetes. So here we are showing that how adipose tissues are located. There are majority of two different types, the white adipose tissue and brown adipose tissue, and there is another third that is called big adipose tissue. So we are here interested to understand the white adipose tissue because they are actually more playing important role in the metabolic disorder, having a role of storing the energy. But when these fat cells when they store in high amount of fats, and then it undergoes a hypertrophic adipocytes, and that hypertrophic adipocyte will ultimately lead to a low grade of chronic inflammation, which ultimately lead to the onset of type 2 diabetes. So basically, the distribution of fat is in our population is either in the pear shaped or apple shaped. So what it is found that the abdominal fat, the intra-abdominal fat, or intrahepatic or intramyocellular lipid content, or pancreatic fat, these are the visceral fats which are majorly the risk factor for developing the type 2 diabetes, rather than the lower fat, the gluteal fat, which is usually found in the pear shaped bodies. So, in this adipose tissue, the visceral adipose tissue, which is composed of basically the fat cells, adipocytes, but there are a large number of stromal vascular fractions of cells, which is including the macrophages, immune cells, dendritic cells, endothelial cells, progenitor cells, stem cells, they all are involved. Within that, the macrophages, which is actually playing a very important role, because the proportion of that macrophages, as you can see here, it is in normal, it is usually there in 5 to 10%, but when due to the cause of the chronic obesity, it reached to the 50% of in the visceral adipose tissue. Adipose tissue macrophages having a good role there in because it helps in the clearings of the dead and damaged adipocytes. It also helps in adipogenesis, angiogenesis, lipid metabolisms. But once it get inflamed or inflammation happen in the macrophages, when then it release a lots of pro-inflammatory cytokines, and which ultimately helps in the onset of type 2 diabetes. So, what our lab is looking into the fact that how this during the period of the obesity, the chronic obesity, we found that the two major player in the microenvironment on the adipose tissue. One is, since there is a large laden of fats when they decompose on your abdominal layer, there the vascularity is very very poor, and that's why there is a zone create where there's a low oxygen tension is there. So that is called hypoxic zone. So there is an adequate amount of oxygen or nutrients are not reaching there into the cells, as well as these hypertrophic adipocytes, they undergo a release of okay the release of a large amount of free fatty acids from their adipocytes, adipose tissue. So that release free fatty acids will ultimately really activate the TLF4 signaling pathway and activates the adipose tissue inflammation that we published in Nature Medicine in 2012. Later, we found that when we are trying to understand the adipose tissue macrophages, their number. So what we found that the adipose tissue macrophages are quite high in HFD models, the high fat diet models, when you compare with the ASPC, which is a adipose stem cell precursor cells, adipocytes, and macrophages. Now, trying to understand these macrophages, we have taken from the diabetic mellitus human patient data and from the non-diabetic patient, where we found that its macrophage load is very high in adipose tissue, which is 32.4%, and which goes to 81.4 is the poly M1 markers. So it is CD80 positive markers. So that means they are highly M1 or pro-inflammatory macrophages compared to the M2. Similar observation we found in the high fat diet mouse model, which is a diabetic animal study and model which used in our study. Later, we've tried to find out that what are the hypoxia, because this is hypoxia and high free fatty acid content that upregulates lots of different microRNAs. When we have done the geo data set, and from there we found that the mid to 10 3p is the important player which is augmented in the adipose tissue, in the visceral adipose tissue particularly. So which we also validated in the human patient as well as in the mouse model. And what we found that in those isolated macrophages, the phospho-NF-κ is also high. That means the NF-κ signaling pathway is on, and that's why the signaling proliferative cytokines is also on. Now, trying to understand that how this microRNA activating the NF-κ signaling pathway, we found that mid-to-10 3P is actually targeting a negative regulator of NF-κ signaling pathway. One such important molecule is a SOS1, that is the suppressor of cytokine signaling factor. SOS1 mediates the proteasomal degradation of NF-κ 65, that having the seed sequence at the present in the 3-UTR where mid-to-10 can bind. And we have shown that how mid-to-10, when is augmented, the SOS1 gets um, the mid-to-10 inhibitor when you have given the SOS1 is again get high, and in case of H plus L it is getting low. So microRNA 3P actually downregulates the SOS1, and that's why the phosphoinhibitor signaling pathway is on. That's why we have administered the anti-mid-to-10 3P LNA delivery, and that's trying to rescue the obese mice from AT inflammation. And we are successful in that. That we found that the macrophage count, you can see here the M1 macrophages in the mid-to-10 LNA delivered mouse. We found that the M1 macrophages load the proliferatory macrophage that goes down compared to the and there's augmentation of more M2 type of anti-inflammatory macrophages. So this book is published in Diabetes, that and we have also shown the SOS1 how the SOS1 marker mRNA expression get high in this particularly treated mouse study. Not only this, macrophages which are shorted from the visceral adipose tissue, and we have cultured them, and then we have separated out the media, and we isolate the exosomes from this, and we found that this exosomes is actually laden with high amount of mid-to-10 3P. So you can see the mouse in the human patient data, as well as and they have a high amount of EVs, and in that EVs, the mid-to-10 3P level is very high. And this ATP with the macrophage, which is released these EVs, which is laden with the mid-to-10, they are actually uptaken by the nearby adipocytes. So we have characterized those EVs. So we have characterized those EVs, and then we have shown that how this EVs, when we have when the uptake this EVs was taken by the adipocytes, the glucose uptake level goes down. So this mid-to-10 is actually again downregulating the glucose uptake, or downregulating the insulin signaling pathway in the adipose tissue, and it makes a dysfunction in the adipose tissue in the function. So there's lots of what we have done, but I'm in the time of interest, I'm not going, because in the EVs there are a whole lot of microRNAs are there. So how do we know that B210 is also doing? That's why we have used the dicer knockout and dicer knockout mice, that and then from there the EVs will received, and from there we have given the B210 mimic and showed that how this is actually uh, playing a role in insulin sensitivity. So what we found that ATM derived this mutated EV worsened the insulin sensitivity by increasing the HOMA-IR score. So we'll lastly, just 1 minute. Lastly, we found that this mutated 3P is actually also targeting and silencing the DIT4 expression in adipocytes, and that's how they are actually prevent the insulin signaling pathway to work. So these are all studies that we have done, and so better to have go to the last page, that where we found that Homaya scores is recovered. So these are all improves. It shows that the mutagen 3P inhibitor improves the insulin sensitivity in HFD mice. So this work is also published. So whoever are interested, they can look into those pages. But we have shown that how EVs also playing an important role in maintaining the systemic insulin sensitivity in. So this is the last graphical abstract that we have shown that in this macrophages, due to the obese hypoxic lipid environment, there is a high rise of beta-3p, which is blocked the SOS1. That's a phosphorygata gets activated. And side by side, they're also releasing a large of EVs. They are reaching to the nearby adipocytes, and they are actually blocking the GLUT4 and IRS1, and that's how they are creating the insulin resistance. So that's how we connected the mutant 3p and their role in adipose tissue dysfunction, as well as we also developed the anti-mutant 3p LNA. If you can deliver, or their EVs, lead and mutant 3p, if you deliver, then it can be rescued. Thank you. This is the all my funding agency and the listener. Thank you for everyone, and I'm happy to take any question.
Thank you. Thank you. So, there is no question because we are running late. Although you can, if somebody is interested, can discuss with you directly. Thank you so much.
So the last speaker is Dr. Moumita Bhomik, is a scientist D at the ICMR National Institute for Research in Bacterial Infections, Kolkata, specializing in molecular immunology and gut biology. Her research focuses on understanding how RNA binding proteins regulate immune response, gut barrier integrity, and epithelial mesenchymal transition, such as human organoids and mouse systems. She explores post transcriptional mechanisms that influence host defense and disease progression. A recipient of the CF, CEFIPRA Women Fellowship and the IIS Women in Science Recognition, Dr. Bhomik's work bridges fundamental molecular analysis insights with translational strategies for infection driven and inflammatory diseases. Dr.
Thank you, sir. Good evening, all. So, thank you, IIS, for selecting me for this award and giving me this opportunity to present my research. So, I belong to this institute, which was earlier called ICMR NICED, most famously known as Cholera Institute of Kolkata. Now it is renamed as NIRPI. So, as Sir mentioned, I work in inflammatory bowel disease. To briefly, if I tell about this disease, these are classified, can be classified into two types. One is ulcerative colitis. As you can see, the inflammation is from the rectal going upwards to the colon, and the Crohn's disease. Something appeared. Excuse me. So sorry. And this is another one is a Crohn's disease where you get patchy inflammation in any part of the alimentary canal. So though this disease do not cause mortality, but it causes a huge amount of morbidity, as you can see in this figure. So what we do in our lab, we tend to believe, again it is coming. We tend to believe that the RNA binding plays a role in dictating the pathogenesis of these disease. So we work with two kinds of RNA binding proteins. One RNA binding protein, when binds to the RNA, causes the decay of that mRNA, and another RNA binding protein that binds to the RNA and causes stabilization. So we believe that there is a balance between this decay causing RNA binding protein and the stabilizing causing RNA binding protein, which leads to the disease. So, if we consider this elephant as the major problem of IBD, what my team does in each part, we look into the regulatory cell function, TH1, TH2 balance, the metabolic balance, and the tight junction protein, and try to understand through RNA binding protein biology. So when I started my lab, what we did, we didn't have much fun. We analyzed the publicly available data, public available data of transcriptome, and we looked for the RNA binding protein regulations that was given between the normal and the colitis mice samples. And we found lot of RNA binding protein was dysregulated. And from those, we chose HNRNPD1 or AUF1 for our further study. Why I particularly chose this, I will be explaining in my next slide. And then when we got funding and all, and we proved that in the colitis, in IBD patients, there is a downregulation of this IBD of AUF1 expression. So we took the biopsy samples and we checked the AUF1 expression from those biopsy samples of IBD, and for the control, we took the patients who were having no colitis or no inflammation as shown by the colonoscopy study. So what is this? Why we took this AUF1? This particular RNA binding protein is fascinating because it plays this dual role. In some cases, it binds and causes decay. In some cases, it binds and stabilizes. So if you look into the literature, there are a lot of examples that it causes either death or life of mRNA. And this AUF1 protein has four isoforms: 45, 42, 40, and 37, having RRM domain, which is the RNA recognition domain, in a singular form or in multiple form. So what we did, so to know the function of any protein, we need a system where the protein is absent. Then only you can study what is the major functional role it plays. So we don't have any commercially available AUF knockout. So we collaborated with Dr. Professor Surajit Sinha of ISES in the Cultivation of Science Kolkata, who is known for developing this morpholinos. He develops the cell penetrating morpholino, which is already a FDA approved drug for Duchenne muscular dystrophy. And we took this technology and he developed morpholino targeting the AUF1 gene. And with his collaboration, we have established that we can develop an AUF knockdown, although it is not equivalent to a knockout, but it can serve our purpose. So while starting with this AUF knockdown mice, we were already working with DSS colitis mice, and we started comparing the features of this colitis mice and the AUF knockdown mice. And what we found, this AUF knockdown mice develop spontaneous colitis. As you can see by the shortening of the gut length, and the erosion in the histology, as you can see in the and infiltration of immune cells. The disease activity index goes hand in hand with that of colitis. And also the gut permeability changes, means the gut becomes more permeable, like the FITC that we done with the FITC dextran experiment, which is a common way to look for the gut permeability. So, taking this, why AUF1 was developing colitis, we started approaching in different angles. One of my students was looking for the tight junction protein regulation, which can cause a barrier function disruption. So, to cut the long story short, what he showed that when we knock down the AUF1 in the mice, he showed that the occluding protein expression, which is a tight junction protein and is responsible for its expression is needed for barrier function, it is shown that it is the half-life of these mRNA, occludin mRNA, is decreased. And on the contrary, claudin 2, whose expression increase in expression causes increased gut permeability, showed that it was the half-life was increased. Another student, Aheli, she showed that IL-27 can polarize the regulation of IL-27 by AUF1 and how it polarizes the B1A cells to B10 population. And Oishika is studying on the ROR Treg and Th17 cell balance, and she showed in a similar like occludin and claudin 2 that ROR gamma T and FOXP3 just behaves in an opposite manner, thus dictating the Treg and Th17 balance in this. Sorry. So I will just schematically show how the dysregulation occurs. We took the example of occludin mRNA, and we can generalize this to any other protein, any other mRNA which causes stabilization by AUF1. So say the occludin is a microRNA 122. It's the target of occludin mRNA. When it binds, we know that with binding, it calls for the RISC complex, and with the RISC binding, the occludin mRNA is degraded. This is a normal phenomenon. Now, if AUF isoforms are present, what happens? AUF1 has an affinity on the microRNA 122, and it takes away the microRNA 122 from the site of the occludin mRNA, and thus relieves this occludin mRNA to produce the occludin protein, and thus the protein is thus we get an overexpression of this protein with AUF1. Now, what happens with the claudin 2? The opposite behavior. Now, we chose microRNA 195 because microRNA 195 is the target for claudin 2. Now, as in the normal phenomena, it binds when microRNA 195 binds, it causes degradation through REST complex. And when AUF1 binds, now AUF1 does not have any affinity for these kind of microRNAs, which targets these mRNAs, which causes a decay in this curve. So it binds to some other place in the mRNA of claudin 2. And now when it binds, it doesn't call off and when it binds, it calls for the ubiquitin, and through ubiquitination, and along with the ubiquitination, the AUF1 protein is degraded along with the mRNA. So next we thought whether we can put on the AUF1 into the tissue to produce any translational benefit we can get or not. So Oshika was working on the organoids, and she has developed this technology working with the human biopsy samples, developing organoids from there. And she took developed organoids from the mice, normal mice, and transfected these organoids with AUF1 expressing, having EGFP along with it, and transferred those organoids to DSS colitis mice on different days and checked for the recovery from the colitis. As from the histological analysis and the gut length, you can see there is a recovery. So I will move fast for the sake of time. So this is what, since it was cloned with EGFP, you can see that the organoids is it present in the histological sections, and with Ki67 we see that there is a regeneration of the tissue. Now I will briefly talk about Aheli's work. She was working on the P1A cells, how it is being polarized to B10, and she showed that butyrate increases the AUF1 expression, and this AUF1 sits on IL-27 mRNA and increases its expression, means stabilizes the mRNA producing the protein. And this IL-27 can be produced from the B1A cells or from the macrophages. It binds to the receptor and polarizes the B1A cells to B10. So with this concept, we thought if we can adaptively transfer the B10 cells. Now, B10 cells transferring is little difficult, so we thought of uh, polarizing it with butyrate and then transfer to a colitis model. Now, here we took another different model of colitis to emphasize the function of B1. We took rat knockout mice, and with the transfer of T cells, we developed a transfer colitis mice, and to this mice, we have adaptively transferred P1A cells, butyrate treated P1A cells from the wild type and AUF knockdown mice. While the AUF knockdown mice did not show any recovery from the disease, but the wild type mice showed recovery from the disease, as evident from the body weight and also the colon length. So we checked for the P1A cells in the MLN, and we showed that these P1A cells were transferring to B10 cells in both in and spleen and the MLN in the rat knockout mice, and the T cells that were being introduced to develop colitis, it was also getting transferred to T regulatory cells in the wild type mice, but not in the AF knockout mice, knockdown mice. Sorry. So, what are the future prospects? Because we are trying to develop a holistic treatment for IBD. In both of the cases, in first case, we try to restore the gut epithelium, but we have not touched on the immune cells. In the second study, we have transferred the beta cells. But our total, we believe that there is a holistic treatment is required for IBD, and we are working on that. So, with this, I will thank you. Well, thank you all and acknowledge my students, my collaborators, Dr. Sinha Shoman Boshak, with we have done the Ragnacout work, and my funding agencies.
Thank you very much. So, uh, now we request all the speakers to come on the stage for felicitation and getting certificate as well as the group picture. And may I also request Professor Raj Mehra to come on the stage, as well as Dr. Manisha Medaikar, Madam, to come on the stage to distribute the certificate to the speakers. Thank you. I just wanted Dr. Mehra and Dr. Manisha to say a few words about this initiative. We started this this year only. So your words, madam, about the recognition of the women speaker and the scientist by Indian Immunology Society.
Very nice. It is a very good initiative they uh, started this year, and it's very encouraging for the female scientists, particularly. And it was really appreciative that we have not that many men are not good, but females also are competing equally. So thank you all of you for your best presentations.
Thank you, madam.
Think I've seen this for the first time in one of these conferences. Very interesting and actually very good to see young people presenting work and excellent work by all of them. I thoroughly enjoyed all the presentation. Congratulations to all of you once again. And thank you for this session. Thank you.
Thank you, madam. Thank you. So with this, we close this session. Thank you.
Thank you, Dr. Santosh Kumar Kar and Dr. Amit Avasti, for leading this meaningful ceremony and for continued guidance, inspiration, and unwavering support to the immunology community. Now, I'll hand over the stage to Dr. Amit Avasti and Dr. Santosh Kumar Kar for the Dr. Kostav for the general body meeting of Indian Immunology Society. Please, sir.
Tasmai Shri Gurave Namaha. In a long lasting tradition and duty to remember all our mentors who have always been handholding us, the IIS continues to do this by honoring the senior immunologist. And this year, as we know, we have honored three of our senior immunologists. And the last two 50th meeting we had done, and during the meeting at what you call THSTI also, we had done the same. Felicitation of women scientists. In alignment with the International Day of Women and Girls in Science, 11th February 2025, IIS launched an initiative to recognize outstanding contribution by women scientists in immunology, our Stri Shakti. The first announcement also coincided with the International Day of Women and Girls in Science. The International Day of Women and Girls in Science is an annual observance adopted by the United Nations General Assembly to promote the full and equal access of participation of women in science, technology, engineering, and mathematics field. We conceptualized this idea, and we had a team of 5 women scientists who were a committee, and as Amit had already told, and the result you could just before, the 13 scientists we selected, out of which 7 could make it to the meeting, this meeting. And we would continue to do this another cycle. We have given IIS awarded 14 student members registration fee reimbursement of rupees 5,000 each to attend the FIMSA advanced course, and we have given 9 travel bursaries to student members to what you call support their travel and attendance to this meeting. And we give a travel award of 10,000 rupees, and I mean, many students use this for most of the immunocons we have been up in the past. Yep. Thank you. Over to Dr. Deepak for his.
Wait for इसको यार कौस्तुभ
ये कहाँ से आ गया? हाँ, इसको बंद कर दीजिए. अपना ट्रेजर.
So I believe that most of the members, or most of the people are sitting here are the IAS member, because this is the meeting is only for the members who are the IAS member. I request non-members may please excuse us, and they have to leave because this is the affair of the IAS. So therefore, only members are allowed. Thank you. My apology. I should have announced this in the beginning, but thanks to Dr. Roma, she just reminded me. Thank you.
Good evening, all. So, as a treasurer, I am going to submit that financial report of the last financial year 2024 to 25, and for this year also up to that 15th of November we have done. So first of all, I would like to thanks to all contributor who have contributed in that extra money from their Immunocon savings to make a increase that corpus of the Indian Immunology Society. And this is the, I think we can say that we have increased our corpus money is at 61 lakh rupees in the last finance year, and this finance year we have 69 lakh rupees. So we have made 8 lakh rupees new FDs, and thanks for that savings received from that 50th Immunocon, Dr. Roma have organized in his team. So we have received that 5 lakh rupees from that. And last Dr. Nandi also contributed 3.6 lakh rupees for their as a registration fees of 10%. So on that surplus money, we will possible to increase that IAS corpus in this year. Yeah, that 50th Immunocon teams. Yeah, 50. Yeah, organizing committee. 50th organizing committees. Thanks to all the members of organizing committee. for their contribution for that society. They have given the surplus money apart from that the 10% registration fees. So that thank you for this 5 lakh rupees extra from that apart from that registration fees. Yeah, I think this is the last to last finance year. So that time cost to buy is the se- uh,
नहीं नहीं
no,
वो जो एक्स्ट्रा से उसका एफडी बना है उन फ्रॉम उनका टोटल कंट्रीब्यूशन
that is your time.
नहीं नहीं वो कितना वो तो 5 लाख 10% मतलब 50th इम्यूनो
back to the society because we already saved 6 lakhs. So the seed money was not then utilized from the society funds. So around 8.3 was that contribution. And we also got some funds from Immunology Foundation. We also returned 1 lakh to Immunology Foundation back because it is being used for some students and the fellowship. So we thought it will be good, put to good use.
Yes.
Thank you, everyone. Thank you. It was a joint effort.
Bangalore given is yeah. So in that last finance year, because that AMS 50th Immunocon meeting is the 2023 to 24 financial year. So in the last financial year, this is the statement of last financial year. So in that we got that income of 12.3 lakh rupees, in which 6.72 lakh rupees in the last finance year we received as a membership fees. This is the around 160 members we have registered for that. This is the highest money in the last 5 years we got in the one financial year as a registration fees money. And another things we received that last contribution from Immunocon 2024 is the Bangalore Immunocons conducted by Dr. Nandi and his team. So we received the registration fees amount is the 3.6 lakh rupees. This is the 10%. So this is the also is contribution is appreciate well for that society.
तो हिम वो कहाँ है नंदी कहाँ है अरे सामने आके शक्ल दिखाओ यार तुमको जरा थैंक्स करेंगे वेयर आर यू
So as usual, every year we receive that partial money for that pay our UIS fees. So we receive the 2 lakh rupees from that Immunology Foundation. This every year we will receive for this IUIS fees. So total we have and we received the around 4.28 lakh rupees as a interest of our 61 lakh rupees FD. So total income of the last financial is 16.6 lakh rupees, and unfortunately we are not able to spend more money, so we have spent in that last finance year is 9.64 lakh rupees. So we have that net profit is that around 6 lakh rupees, so we have to pay that tax for that 6 lakh rupees. Now so we have in that in this year we have increased fellowship and increased that travel money, so we are in on the track in this year. So we are spending more money in this year.
Did we get any savings from Bangalore conference?
No, only registration fees of 10%.
How much are we paying to IOIS every year now?
So last year we paying 3.12 lakh rupees, and this year that IAUIS increased 15% of that registration fees money.
Yeah.
So this year we have paid 3.7 lakh rupees.
Are they still giving you rebate? Because during our time we impressed upon them you should charge us only half the fee. How many members they charge from us?
So in that our that we have sent that
देखिए
1200
इनकी जो 3,12,000 है It will come on the members that you show to them. We
Yes.
have so many members. How many members have you shown to them?
So we have shown 1200 members.
1200.
Yeah, because
And
we have
how
shown
much
the
were
active
they?
members.
They used to charge us $2.5 per member. Earlier, they used the full fee was $5.
Yes.
I don't know how much is the fee
Yes,
now.
yes. 3 point something.
So on 2.5, we will have to pay 15% more.
Yes. This year we have paid already.
Okay. So
Around
our
3 euro.
contribution to iOS will increase.
Yes.
Has increased.
Has increased.
Yeah, I mean from next year it
Uh-huh.
will
Yes,
increase.
yes.
So from the Immunology Foundation, you still get 2 lakhs.
That's
Yes,
correct.
we also request that Immunology Foundation if they will increase. So this year we have not increased. Again, we will write to Immunology Foundation if they will
Yes,
increase.
yes.
Yeah. Again, this year we will also write for that. So this is that audited statements of last financial year and this current financial year because that last year we have. So this current this is our corpus money that we have already 69 lakh rupees FD and we have some saving accounts around 8 lakh rupees. And this year, if you look at that from that April to November 15th, so we have earning around 9.67 lakh rupees and we have expenditure around 12 lakh rupees. So in this year, we will cover and we will not pay more taxes for this year. So we are on that process. Thank you. Thank you.
Thank you, Deepak, for. Uh, so I must really congratulate Dr. Deepak because he's very particular about the finances. And the way he's presenting, you get a sense. If you ask me to about the finance, I think I will come zero because I will be more focusing on other things, not the finances, which I need to learn. But thank you, Deepak, because every once in a week he comes and say, this saving is coming, this interest is coming, and he often visit AIMS headquarter. He sits down with the AIMS headquarter person who looks after various activity, and he travels from THSTI to AIMS. We don't pay him anything. So we are not paying anything to him for taxi or anything. He goes by himself because from AIMS he can go take a metro to go to his home because he lives in Delhi. So thanks very much, and we really appreciate your contribution. So now we first of now we can go through the GBM agenda, which has been circulated to all the members. We will go through one by one, and the very first agenda is the approval of the GBM minutes. Sir,
These people will just read. And then minimums करके I'll have to open one log.
So now this 10% you got from Bangalore, you'll be getting from here. I was wondering if you could have a separate fund, call it Immunocon fund, and help it, for student travel to Immunocon and things like that. So if you could, keep it a little separate and do that, that might be useful because often while last year's organization, I found a lot of students writing about, travel funds and things like that. If you could do something like that, I think it would help. You reserve that and you increase the numbers. So that's something that for you all to consider.
Constitution.
thank you, Deepankar, for your suggestion. But so I just